lot of different problems that can occur in this system. And so again, DLBCL is the most common type, and it has the worst prognosis of the high-grade lymphomas. And as can be seen by this image, it develops from abnormal B cells that spread out rather than grouped together under a microscope. So let's zoom in a bit on some of the common oncogenes. So we have MYC, BCL2, and BCL6, as I previously mentioned. MYC is mainly associated with cell proliferation. BCL2 is an anti-apoptotic gene, and BCL6 is a transcription repressor, and they're found on chromosomes 8, 18, and 3. So BCL6 is, again, a proto-oncogene and a transcription repressor that is mainly required for germinal center formation and antibody affinity maturation, which has different mechanisms of action specific to the lineage and biological function of the cell. And so it forms complexes with different co-repressors and histone deacetylases to repress the transcriptional expression of different subsets of target genes, and it represses its genes by binding directly to this DNA sequence 5' TTCC TAGAA3', which is also known as the BCL6 binding site. It can also indirectly repress the transcriptional activity of transcription factors, and then it represses genes that function in differentiation, inflammation, apoptosis, and cell cycle control. We zoom in a bit on the MYC gene. It becomes clear that it's tightly correlated to cell proliferation. It functions as a transcriptional regulator and is capable of inducing or repressing the expression of a large number of genes, which are thought to mediate its biological functions. So the downregulation or inactivation of MYC results in the impairment of cell cycle progression. As can be seen here, it's reported in 10% of DBCL cases, so it is something that we need to pay attention to. Zooming in finally on B-cell lymphoma 2 or BCL2, this is an oncogene and it's primarily plays a role in regulating apoptosis. It works to promote cell survival by inhibiting apoptosis, which can accelerate lymphoma growth and induce resistance to chemotherapy. So the expression of BCL2 is related to a poor prognosis, just like with the other two genes, and it's normally expressed transiently during B-cell maturation. So BCL2 protein expression can indicate the deregulation of the BCL2 gene caused by translocations, mutations, or gene amplification. BCL2 also suppresses apoptosis in a variety of cell systems, including factor-dependent neural cells or lymphopoietic cells, and it also regulates cell death by controlling the mitochondrial membrane permeability, and it appears to also function in a feedback loop system with these caspases by inhibiting their activity, either preventing the release of cytochrome C from the mitochondria or by binding to the apoptosis activating factor.
So moving on, we're going to go over to classifications. A lot has gone on, and a lot has changed since 2008, switching over to 2016. So in 2016, the World Health Organization turned the classification HGBL, high-grade B-cell lymphoma, to replace the classification of BCLU, B-cell lymphoma unclassified, from 2018 or 2008. These B-cell lymphomas have features that are intermediate between DLBCL, diffuse large B-cell lymphoma, which is really common, the most common non-Hodgkin's lymphoma actually, and BL, Burkitt's lymphoma, a highly aggressive NHL with MYC translocation. HGBL consists of two subcategories, as seen right here. We have HGBL-R and HGBL-NRS, which we'll get into shortly. But before that, it's important to note that although this seems like a clear spectrum that we laid out here, in actuality, DBCL and BL are almost indistinguishable in adult patients specifically, but they do need to be categorized due to having very different regimens when it comes to treatment. Now let's get more into the specifics pertaining to the distinguishing between the two subcategories. So first, the categorization of HGBL-R. R stands for rearrangement. These are HGBL that have MYC, BCL2, and RBCL6 rearrangements. These common oncogenes were presented by Melody earlier. The rearrangements of these genes can vary from double hit, MYC BCL2, or MYC BCL6, and possibly even triple hit, which is just all three of them all together. So one distinguishing factor of these is those who have this type of categorization is the presence of MYC translocation. So that is actually required for this classification. On the other hand, we have HDBL-NOS, which stands for not otherwise specified. These are rarely encountered and have no MYC, BCL2, and or BCL6 rearrangements. This is what makes it indistinguishable, or actually distinguishable from HDBL-R. Without these, it's going to be NOS, non-otherwise classified and specified. Just like HDBL-R, though, HDBL-NOS present morphologies between BL, TL, BCL, and BL.
Sí, adelante.
Another important side note is the difference between double hit and triple hit expressors. Hits or rearrangements are classified under HDBL-R, whereas double expressors are the overexpression of these proteins. So you can actually have double expressors in both, but it is not an indicator of either. So moving forward, we do have epidemiology right here. Based on clinical data, HDBL has a slight male predominance as opposed to HBGL-NOS, which seems to occur equally in both genders. The age range of which these lymphomas occur in and are reported at are in the 6th and 7th decade of life, so when you're around 60s and your 70s respectively. Though there's no clear evidence that the diagnosis of HDBL-NOS occurs at a particular age range, there seems to be a trend of increasing occurrence with age. The clinical features. When reported, it is most common for patients to be in the later and more advanced stages of the disease. With HDBL-R, more than half are reported with stage 4, and with HDBL-NOS patients, as you can see here, they actually report at stages even later to 3 and 4. Other clinical features pertaining to HDBL-R is the high IPI. This consists of showing elevated lactate dehydrogenase serum levels, which indicate tissue damage. There's also the ECOG, the blood performance statuses of patients, which tend to be 2 or 3 and even 4. By 4, that means they're bedridden. HDBL-R patients have more than one extranodal site, which can occur in the bone marrow and or the CNS central nervous system. Similar to HDBL-R, the NOS version is involved in the bone marrow and CNS with lymphadenopathy and extranodal mass. It is common to see B symptoms with these patients as well, and in rare cases, the patients can actually have tumor cells circulating in the peripheral blood. Now moving forward, we're going to go with diagnosis and how can we prevent all these things or make it a lot better, because as you can see right here, B-cell lymphoma gets really bad really quick. In terms of diagnosis, HDBL methods are used are very similar to methods that are for other lymphomas as well. So for example, for number 1 here, we have DLBCL. It can diagnose from an excision, biopsy of the lymph nodes in suspicion. These are biopsy sheets that are large cells that support the structural integrity of the follicular center. When looking at these cells, we're trying to look for positive CD20, PAX5, CD10, BCL6, BCL2, and of course, CMYC. As mentioned earlier, with extranodal mass-associated BCL, diagnosis can also be made with bone marrow biopsies due to the often infestation of such around 59 to 94 in BCL and, of course, HDBL patients. Due to the various similarities between lymphomas, various methods are divided in order to identify the classifications and the degree of the disease. So we have up to 4 types of diagnosis here. I'm going to go through them really quickly and really briefly. But first we have FISH. So this is Florensis in situ hybridization. It is used to address specific gene loci of interest with probes. An example of this usage is having MYC break-apart probes as well as BCL2 translocation. Both are evident in HDBL. Then we have cytomorphology. Cytomorphology is a guiding principle for downstream diagnostic. So for example, an example of bone marrow smear contributes to the determination of whether there is a lymphocyte or even lymphoma infiltration. And it can even tell you the possibility of such occurring in the future. Cytomorphology and histology can also be helpful in assessing the degree of the lymphoma maturity. In terms of classification of the disease, when you look at the common morphologies associated with it, it can help a lot. So in terms of HDBL-NOS, the typical morphology seen is very similar to BL with a monomorphic population. These cells will have a less strong basophilic cytoplasm and a lack of vacuoles. In contrast, HGBL-R has a majority of DL-BCL morphology. So that's the difference between them. Morphology of the cell provides a lot of information, but is limited to variable morphologies presented in HDBL patients, especially in double hit patients. Therefore, there's a need for various methods and diagnostics to tell us more. Moving forward, we do have molecular genetics. This type of molecular genetic mutations that we tend to observe are the TP53 and the MYC. Looking at these mutations can help with diagnostics because TP53 mutations are actually more common in MYC and BCL2 than it is in MYC and BCL6. In BL, TP or TCF3 and ID3 are very common and would be the main targets you want to look out for to identify it. Whereas in HGBL-NOS, in terms of molecular genetics, there's no rearrangements between MYC, BCL2, as we mentioned before, along with 20 to 30% cases showing a single MYC rearrangement. Then we have immunophenotyping. So a general breakdown of what to look out for this diagnostic measure is what is expressed and what isn't expressed. So what is expressed as a mature B-cell lymphoma, they will express CD19, CD20, CD59 or 79A, and PAX5. There's a lack of CDT, CD34, and cyclin D1 expression. Then, furthermore, we do have an absence of Ig expression, but this is not proof or a precursor of whether or not you can determine the diagnosis. CD10 and BCL6 expression is found in most lymphomas, and IR4 and MUM1 is expressed in approximately 20% of the cases. Okay, so there's a lot that I threw, and I know there's a lot that's going on, and the expression gets really complicated. So we're looking at morphology, we're looking at phenotype, we're looking at cytogenetics, but how can we diagnose this? So the diagnosis approach to HBCLs, lymphomas are potentially fall in the HBL categories, be morphology to resemble these type of things. So there's a lot of things that it does resemble, but these distinctions can be very subjective as well, since there's a lot of interwebbing between them. The orange arrows are the ones that indicate that there is a single hit, and of course the red arrows are the ones that indicate that it is a triple hit.
Okay, perfect. So now that we have diagnosed this and we now know what's happening, now we can find ways to treat it. And so because high-grade B-cell lymphomas grow so quickly, the first treatment is often chemotherapy, and it's usually a regimen of 4 drugs and 1 monoclonal antibody. And so we're going to use cyclophosphamide, doxorubicin, vincristine, pyridoxine, and rituxan. And so the regimen is known as R-CHOP, and it's often given in cycles 3 weeks apart, depending on the stage that an individual is at for HGBL, depends on how many cycles they'll go through. And so people in stages 1 to 2 will usually undergo 3 to 6 cycles of R-CHOP, and then that might be followed by radiation therapy to the infected lymph node areas. Another new, and it's becoming a more popular treatment, is stem cell transplants. It's also known as bone marrow transplants, and so it lets doctors give higher doses of chemotherapy, sometimes along with radiation therapy. And so stem cell transplants are used to treat lymphoma patients who are in remission or who have a relapse during or after treatment. And so we have 2 main types of stem cell transplants. In an autologous stem cell transplant, the patient's own stem cells are used, versus in an allogenic stem cell transplant, we use a donor. Donor stem cells, usually a family member or someone who has the same type of cells that the patient. And so the donor's tissue and the patient's tissue in both situations are frozen, and then they are then inserted into a patient via an IV. And so autologous transplants are the most common because they use the patient's stem cells. This one may not be an option for a lot of patients. And so allogenic transplants are used a lot less often. They also have a lot of severe side effects that a lot of patients can't tolerate, especially those who are older, as with a lot of HGBL patients, or who have some other type of medical problem.
You have 1 minute.
these are our references, and we're wrapped up. Thank you so much for listening. This is such an interesting topic, and me and Caitlin had so much trouble narrowing it down because it's so broad and there's so many different directions we could have taken it in. So yes, thank you so much.
So if there aren't any questions, up next we have Dr. Jorge Aquino from Australia University, the Laboratory of Developmental Biology and Regenerative Medicine. He will be presenting on the contribution of the neurocrest and glass-positive WNT1 positive parasites within healthy and diseased livers. If there are any questions for the presenter, please write them in the chat at the end of the presentation. Thank you.
So I was, okay, thanks for the invitation.
Jorge, perdón, Jorge, tienes que ponerlo en presentation, por favor.
Presentation. Sí.
Gracias, disculpa.
Es in English, ¿no? The presentation.
No, en español.
En español, perfecto.
Bueno,
Bueno,
hablo en español.
perfecto, muy bien. Bueno, entonces le hice igual, la presentación la puse en inglés para que lo pudieran seguir los que no hablan muy bien español. Bien, entonces ya habiendo introducido el título, voy a pasar a exponer el tema que me toca. A ver si logramos, ahí está. Bueno, la cresta neural es una estructura embrionaria que se origina en un estadio embrionario temprano, ¿no? En donde el embrión tiene tres láminas y se origina en la parte del ectodermo, en la parte dorsal del embrión, cuando es un disco. En esa parte, cuando se induce la formación del tejido neural, que es el primer tejido que se diferencia en un embrión, vamos a encontrar la cresta neural en los bordes de la placa neural. Luego se pliega esta estructura porque se produce un surco neural y se forma el tubo neural que será posteriormente el cerebro o la médula espinal. Del techo de este tubo hay un grupo de células que se delaminan, se desprenden del epitelio y migran, y son capaces de migrar grandes distancias, de modo tal que contribuyen con distintos tipos de células. En el caso de las células mesenquimales, sobre todo sucede en el cuello y en la cabeza, mientras que en el tronco suelen originar neuronas, glía, células secretorias de glándulas y también las células pigmentadas de la piel. Nosotros en nuestros estudios desarrollamos algunos modelos de fibrosis hepática. En uno de esos modelos inyectamos una droga que se llama tiocetamida, que después de unas 8 semanas genera una fibrosis hepática importante establecida. Posteriormente puede generar una cirrosis. Otro modelo distinto que no es hepatotóxico, sino que es una ligación, digamos, del conducto biliar, genera también una fibrosis hepática posterior a dicha lesión que es originalmente en el conducto biliar. Entonces son dos modelos distintos con distintas etiologías, corresponden a distintas etiologías de la fibrosis hepática. Luego obtuvimos el hígado, la médula ósea y la sangre periférica de los animales tratados. Y para poder identificar el origen en cresta neural, lo que hicimos fue utilizar unos ratones transgénicos que expresan un gen reportero en rojo bajo un promotor del gen de la proteína Wnt1. Bien, entonces lo primero que estudiamos fue la glía en el hígado, porque también en el hígado hay nervios, hay tejido nervioso y hay glía. Entonces normalmente las células de la glía se disponen alrededor de la vena porta en el hígado y tienen típicamente dos procesos. Nosotros en el caso de la fibrosis hepática, en los dos modelos, observamos un aumento significativo en la cantidad de células de la glía en el hígado. No solo eso, sino que la morfología de las células pasa a ser mucho más compleja. Analizamos esta complejidad de las proyecciones de la glía mediante un método que se llama análisis de Scholl, que consiste en medir la cantidad de veces que los procesos de esta glía atraviesan estos círculos concéntricos, desde la zona más cercana al soma del cuerpo celular de la glía hacia la zona más lejana. Es lo que graficamos acá. O sea, de 0 micras a 100 micras desde el soma. Y así podemos observar que en los animales con fibrosis, la complejidad de los procesos, o sea, la cantidad de veces que se intersectan estos círculos, aumenta con relación al control, en los dos modelos estudiados. No solamente eso, sino que también la longitud de los procesos gliales es mayor, la longitud total es mayor en los dos modelos de fibrosis. Bien, nos interesó también ver que había otras células distintas que eran trazadas, o sea, que expresaban el gen reportero, o sea, que derivan de células que expresaron en algún momento Wnt1 en el hígado. Y estas células tenían aspecto de células endoteliales y expresaron lectina, que es un marcador de célula endotelial. La célula endotelial es la célula que recubre los vasos sanguíneos y la observamos tanto en los vasos pequeños o capilares hepáticos como también en las venas cortas. La cantidad de células endoteliales que eran positivas para el gen reportero con la fibrosis aumentó significativamente en los dos modelos de fibrosis. Sin embargo, cuando estudiamos durante el desarrollo el origen de estas células, vimos que no era en la cresta neural, sino que eran células que están en la médula ósea, como les explicaré después. También observamos hepatocitos que eran positivos para el gen reportero. En el caso control era una frecuencia rara de estas células positivas, pero con la fibrosis aumentó muy significativamente, siendo encontrada a veces en grupos de células que expresaban el gen reportero. Bien, y vemos acá la cuantificación. Los dos modelos de fibrosis, con tiocetamida hepatotóxico y con la ligadura del conducto colédoco, cómo aumenta la cantidad de células tipo hepatocitos en el hígado, que son tomeitopositivas, o sea que derivan de células que son Wnt1 en el momento del desarrollo del animal. Y estas células nuevamente se vio que no eran derivadas de la cresta neural. Cuando quisimos analizar la médula ósea de estos animales, para ver el posible origen de estas células hepáticas, que veíamos que era raro que tuvieran origen hepático por la frecuencia normal en que se encuentran y cómo aumentaban tanto y en tan distintos lugares del hígado, pudimos analizar la médula ósea para ver si se originaban ahí y qué tipos de células expresaban el gen reportero en la médula ósea. Y vimos que las células de la glía, de la médula ósea, que acompañan más abundantemente las venas, las vénulas y las arteriolas en la médula ósea, eran positivas para gen reportero y para GFAP, que es un marcador de glía. Sin embargo, también había algunas células que eran GFAP negativas, y estas células vimos que tenían procesos largos, complejos también, probablemente rodeando células hematopoyéticas, que por ese motivo probablemente formen parte de nichos de células hematopoyéticas en la médula ósea. Estas células expresan fibronectina, que es un marcador de células estromales. Con lo cual, cuando cuantificamos las células tomeitopositivas de la médula ósea, vimos que la mitad eran células de la glía, en verde, y la otra mitad eran células estromales. Y de las células estromales había dos fenotipos. Unos que eran de células perisinusoidales, que están alrededor de capilares pequeños en la médula ósea, y otra fracción muy pequeña de células periarteriolares, que están alrededor de vasos un poquito más grandes. Y típicamente había dos células trazadas por campo de microscopio de 200 por de los dos tipos más abundantes. Quisimos analizar otro ratón distinto que expresa el gen reportero bajo un promotor que se llama GLAST, que nosotros analizamos y vimos que también estaba expresado en células de la glía, en estos ratones, el gen reportero. Entonces lo que vemos en este caso en la médula ósea es que trazan células perisinusoidales, que son estas que ven aquí en el parénquima de la médula ósea, y trazan también periarteriolares, con la misma frecuencia que veíamos antes, como les voy a mostrar después. Estas células rodean a vasos que son CD31 positivos y expresan fibronectina, los dos tipos, las perisinusoidales y las periarteriolares, expresan desmina, expresan viventina, como se ve acá en los dos tipos, y únicamente las periarteriolares expresan actina de músculo liso, que es un marcador típico de estas células y que no está presente en las perisinusoidales. Y la frecuencia es de vuelta, la gran mayoría de las células que son tomeitopositivas son perisinusoidales y hay unas pocas periarteriolares. Y en general, por campo de microscopio encontrábamos 20 células positivas, a diferencia del caso de los ratones que habíamos visto antes, que encontrábamos solo dos. O sea que el 10% digamos, de las células que encontramos aquí son derivadas de células Wnt1 positivas. No hay contribución con la glía, en la médula ósea ni en el hígado de células trazadas con estos ratones GLAST. Cuando analizamos los tipos celulares que encontrábamos en el hígado lesionado por fibrosis, vimos que aumentaban la cantidad de células endoteliales también que eran trazadas significativamente a 8 semanas de tratamiento. Tanto en los animales que inyectábamos con tamoxifeno AP2, en que indujimos la recombinación del transgen, como cuando lo hicimos a P60. En estos ratones tienen la especificidad de que tienen una variante de CRE, que es una enzima que permite la recombinación y la expresión del reportero, que necesita de la presencia de tamoxifeno para estar inducido. Con lo cual, solo cuando inyectamos esta droga, que puede ser en este caso AP2 o AP60, o sea, estadio postnatal 2, o en el adulto AP60, vamos a ver células que son tomatopositivas. Con lo cual, AP60 ven que baja mucho la fracción de células trazadas, y eso se debe a que hay menos células en este estadio en el adulto que expresan o que tienen activo el promotor de GLAS. Bien, y en cuanto a los hepatocitos, también trazados con estos ratones GLAS, vimos que en el caso control había muy pocas células trazadas, mientras que estas aumentan después de la lesión, ¿no? A una semana, o a 8 semanas de tratamiento, en que es mucho mayor la cantidad de células y es significativo que son tomatopositivos. Entonces, para corroborar que podían migrar estas células de la médula ósea hacia la sangre periférica y después al hígado, analizamos los compartimentos, la cantidad de células que eran tomatopositivas, que expresaban GLAST, que expresaban CD44 en sangre periférica, en el caso de los paneles de abajo, o en la médula ósea. Y vimos que en la médula ósea se reducía la cantidad significativamente después de la lesión en los dos modelos, y aumentaba significativamente en la sangre periférica, lo que habla de un cambio en el compartimento en que se encuentran ahora estas células. Quisimos saber si tenían propiedades de células progenitoras, entonces las pusimos en un frasco en cultivo y vimos que generaban colonias, algunas eran positivas y otras eran negativas para el gen reportero. Las positivas las ven acá en gris. O sea, más o menos un 10% a 15% de las colonias expresaron el gen reportero. Y con la lesión se reducían la cantidad de colonias positivas. Acá lo vemos en los frascos y acá está la cuantificación. Y la reducción era similar en las fracciones totales de colonias o en la fracción de colonias que eran tomatopositivas, o sea que derivaban de estas células Wnt positivas que están en la médula ósea. Sin embargo, a pesar de que no hubo una diferencia en el número de colonias que eran tomatopositivas con la lesión con relación a las que eran negativas, sin embargo, la abundancia de células en cada colonia, dentro de la colonia, se redujo mucho en el caso de los animales tratados con tiocetamida, lo que sugiere que esta lesión lo que hace es que induce que las células progenitoras que permanecen en la médula ósea tienen mucho menor capacidad de proliferación que en una condición normal. Es como que se agotaran producto de la lesión. Quisimos cambiar de modelo, utilizamos en este caso un modelo que se llama de hepatectomía, que es un corte del 66%, del 70% del hígado, de la masa hepática, con lo cual el 30% remanente se regenera en unos 7 días. Es un buen modelo para ver cómo estas células eran capaces de contribuir con la regeneración del hígado. Primero analizamos la movilización y vimos que estas células específicas de la médula ósea se movilizaban desde el compartimento de médula ósea, donde se reducían después de la hepatectomía a los 7 días posteriores a la hepatectomía, a los 5 días, perdón, y en la sangre periférica aumentaban. Analizamos el hígado de ratones GLAS-CRE y vemos que la cantidad de hepatocitos en el caso de la hepatectomía aumenta significativamente los que son trazados por el gen reportero, o sea, por la expresión de GLAS. Bien, y no solamente los hepatocitos, sino que también las células endoteliales aumentan significativamente después de la hepatectomía. Con lo cual eso sugiere que estas células ayudan a la regeneración del hígado. Quisimos analizar si podría haber alguna contaminación con otros tipos celulares que podrían fusionarse con células en el hígado, o sea que se sabe que hay células hematopoyéticas que se pueden fusionar con hepatocitos en el hígado. Bien, y vimos que ninguna de las células que eran tomatopositivas expresaban CD45, tanto en el control como en el animal con hepatectomía, y tanto en la fracción de células de médula ósea como de la sangre periférica. Tampoco expresaban CD31, que es un marcador de progenitores de células endoteliales. No expresaban CD11B ni c-KIT, que son marcadores de progenitores de granulocitos y monocitos, que son células que típicamente podrían llegar a fusionarse después de una lesión, como se ha visto en el cerebelo y en otras regiones y en el hígado también. Y tampoco expresaban SK1, que es un marcador de células de médula ósea, que está en algún tipo de células estromales de médula ósea, y también está en células hematopoyéticas, células madre hematopoyéticas, mieloides y linfoides. O sea que ninguno de los progenitores de otro tipo, otras ramas, digamos, ni algunos tipos de células estromales correspondían el fenotipo de estas células que estamos estudiando nosotros. Fue interesante ver que estas células coexpresan marcadores estromales, como vemos en este caso, ¿no? Fibronectina, vimentina y desmina, en el hígado. O sea, una vez que se diferencian hepatocitos, siguen expresando la mayor parte de ellas estos marcadores estromales. Y lo que hicimos fue, para corroborar que estas células pudieran diferenciarse en células del hígado, es trasplantar células a partir de estos ratones Wnt1-3 en ratones atímicos. Estos ratones atímicos fueron inyectados con las células que expresan el reportero, con células de la médula ósea, después de haber hecho la hepatectomía en estos ratones atímicos, ¿no? Luego de 6 días de la hepatectomía. Y lo que observamos es que con la hepatectomía el reclutamiento de células Wnt positivas aumentaban en el hígado, ¿no? En la fracción parenquimática. Acá estamos viendo solo las células que son hepatocitos. Bien, y no solamente eso, sino que había células que eran CD44 positivas, tomato positivas, que, digamos, después de la hepatectomía expresaban albúmina, o sea que eran hepatocitos. Y eso aumentaba significativamente con relación al control sin hepatectomía. Y había el doble de células que expresaban fibronectina. Esto es al día después del trasplante. A los 5 días después del trasplante de las células, seguíamos viendo un mayor reclutamiento de las células en el hígado, que eran tomato positivas. Estas células, en este caso, del 20% de esta fracción, de todas las que son Wnt1 positivas, el 20% coexpresaban albúmina y fibronectina. Quiere decir que hay células que siguieron diferenciándose en albúmina, que probablemente expresaran fibronectina antes. Y estas células también, en una fracción, expresa vimentina. O sea, estamos analizando la misma subfracción de células que son Wnt1 positivas. En el caso de la fracción no parenquimática, para estudiar las células endoteliales, vimos que también había un mayor reclutamiento de células tomato positivas después de la hepatectomía en los ratones atímicos en el hígado, y estas células con mucho más cantidad que expresaban CD31, o sea que eran células endoteliales. Estas células de la médula ósea eran capaces de expresar marcadores de células endoteliales. Y a los 5 días esta proporción también se mantiene, más significación, o sea, el mayor número de células CD31 positivas en el caso de la hepatectomía. Bueno, este resultado nos resultó interesante. Nos preguntamos si podría haber alguna droga, sustancia pequeña que pudiera inducir la expresión, la movilización de células de médula ósea y la contribución de estas células con el hígado. Y el tipo de molécula pequeña que nos interesó, que es un oligonucleótido, se llama IMT504, que se sabe que induce la movilización de células mesenquimales, células madre mesenquimales de médula ósea, con lo cual podría ser un buen candidato para usar. Y lo que observamos es que después de inyectar en los animales IMT504, obtuvimos muestras de médula ósea y de sangre periférica, y vimos que había un aumento de 20 veces la cantidad de células tomatopositivas en médula ósea y en sangre periférica en todas las condiciones, con o sin lesión por tirocetonida, después de unas 18 horas de haber inyectado la droga. Ninguna célula es capaz de dividirse 20 veces en tan poco tiempo, en 18 horas. Entonces esto sugiere que puede haber una inducción de la expresión de Wnt en algunas de estas células que estamos observando en médula ósea o sangre periférica. Entonces, para estudiar qué sucedía con la fracción de células progenitoras, realizamos cultivos, ensayos de unidades formadoras de colonia para ver las propiedades de progenitoras de células estromales que tenemos en las células tomatopositivas y el efecto que tiene IMT sobre esas colonias, formación de colonias. Y lo que observamos es que había un aumento significativo por la aplicación de IMT in vivo en los animales en la cantidad de colonias tomatopositivas y en la cantidad de colonias totales que teníamos en cada placa. Este aumento fue a expensas de la fracción tomatopositiva. Como puede observarse acá, el porcentaje de colonias que eran positivas aumentó al doble o más en el caso de los animales que recibieron la inyección 18 horas antes de poder obtener las muestras para sembrar los cultivos y generar este ensayo de progenitoras de células estromales. Esto quiere decir que de alguna manera afecta específicamente la fracción de células que estamos estudiando. Y no solo eso, sino que también las colonias recuperaron la capacidad normal de proliferación y generan colonias densas nuevamente. O sea que este oligonucleótido es capaz de recuperar, de restaurar las funciones características de las células progenitoras en la médula ósea, lo que puede ser interesante porque quizá en varios ciclos de aplicación, en una enfermedad crónica prolongada, podría permitir un mucho mayor reclutamiento y contribución con regeneración de tejidos. Bien, entonces, como se sugiere un efecto en la proliferación celular, estas células lo que hicimos fue obtener y expandir los cultivos de colonias positivas hasta pasaje 4. Y analizamos el tiempo de duplicación de los cultivos para ver si el tiempo que requiere una célula en dividirse y generar la misma cantidad de células a un cultivo, duplicar la cantidad de células es menor. Y fue lo que observamos, o sea, de 6 días bajó a 2. No solo eso, sino que la fracción de células que estaban en división celular era muy significativa, un 71%, 72% de las células estaban en división, lo que es bastante poco usual en un cultivo de este tipo. No solo eso, sino que por real-time PCR, PCR cuantitativa, analizamos la expresión de Wnt1 y de ciclina D1, que está bajo, río abajo, digamos, ¿no? De Wnt, porque es inducida ciclina D1 por efecto de Wnt, y vimos que el tratamiento in vitro de las células induce en un aumento de un 50% en Wnt1 y ciclina D1. La movilización es otro factor que nos interesaba analizar in vitro, el efecto que podía tener esta droga, IMT-504, y para eso usamos un modelo de ensayo de herida in vitro, ¿no? Herida, o sea, generando un espacio por, digamos, un tip que lo corremos sobre la superficie de cultivo, después analizamos cuánto tiempo tardan las células en repoblar este espacio que se generó por la lesión in vitro. Y lo que vimos es que a las 8 horas de haber hecho la lesión ya hay cambios significativos. Y después posteriormente se mantuvieron, incluso se mejoraron la cantidad de espacio que era recubierto en la zona lesionada, digamos. Y no solamente eso, que es una medida de la movilización celular, sino que también expresaron las células por PCR, vimos que había una reducción en la expresión de caderina, lo que se asocia a la movilización celular. Bien, uno podría pensar que IMT podría estar induciendo Wnt, la activación de Wnt y la expresión de tomato en células que antes no eran tomato positivas. Y para eso, corroborar que no era así, lo que hicimos fue generar ensayos, digamos, de unidades formadoras de colonia, estromales, y analizar con el tiempo, después de un pulso de IMT-504, si aumentaba la proporción de células que eran de colonias que eran tomatopositivas en este caso, ¿no? Y lo que vimos es que no, el porcentaje se mantenía igual en los primeros 2 días. Bien.
Dr.
Entonces,
Aquino, we have three more minutes.
okay, perfect. So, and then for the recruitment of cells, el reclutamiento de las células, vimos que había mucho mayor reclutamiento en los animales que eran inyectados con IMT-504 en vivo y que había más contribución con células albúmina positivas, o sea que aumentaba la cantidad de hepatocitos que eran tomatopositivas por efecto de la aplicación en vivo de IMT-504. También fibronectina aumentaba en esta condición. Y en el caso de las endoteliales, esto es un día post-trasplante, vemos que se reclutan más células tomatopositivas por efecto de IMT-504, había más células endoteliales por efecto de IMT-504, con la cual concluimos que la cresta neural solamente contribuye con células de Schwann en el hígado, y estas células de Schwann son activadas y generan un fenómeno de biosis durante una fibrosis hepática. Hay una subpoblación de células sinusoidales estromales de médula ósea que expresan BLAST y que son trazadas con Wnt1. Esta subpoblación celular es capaz de originar hepatocitos y células endoteliales durante procesos regenerativos hepáticos. Y por último, esta fracción celular de células progenitoras se movilizan hacia sangre periférica y una lesión crónica hepática causa un fenotipo hipomórfico en estas células que puede ser recuperado o revertido por aplicación de IMP504, posiblemente a través de la reactivación del promotor de Wnt1. Bueno, termino agradeciendo a los que hicieron gran parte de este trabajo. Romina Sierra, que fue becaria doctoral mía, se doctoró hace poco, el año pasado. Tesinistas, estudiantes de grado de medicina de Federal Austral. Un grupo de Karolinska, con el que colaboramos sobre todo para analizar el origen en la médula ósea de estas células hepatocitos endoteliales. Y después nuevos actores del grupo que están investigando en tejido nervioso, en lesiones nerviosas y también en lesiones en infarto cardíaco, y otros grupos que nos proveyeron con IMP504 y colaboraron con los cultivos celulares o con algunos de los ratones del estudio. Nada más, muchas gracias.
Muchas gracias, Jorge. Nos vemos en Perú el próximo año, en vivo.
Bueno, perfecto. Bueno, muchas
Gracias,
gracias.
gracias. Gracias, Jorge.
Thank you, Dr. Aquino, for that wonderful presentation. And if anybody has any questions, feel free to message in the chat. Up next, we have Andrew Reyes and Alexandria Wang. They are UCLA students and members of the International Circle of Genetic Studies. They will be presenting on myelodysplastic syndrome, also known as MDS. If there are any questions for the presenters, please write them in the chat at the end of the presentation. Thank you.
Hi everyone, today Andrew and I will be discussing myelodysplastic syndromes, also known as MDS, for our workshop presentation today. The purpose of our presentation is to summarize the latest literature on MDS. We plan to introduce MDS, its diagnosis, risk stratification, cytogenetic profile, and adapted risk therapies. So MDS is a heterogeneous group of myeloid malignancies with distinct natural histories. It is detected by the presence of cytopenia during routine analyses of peripheral blood. In the United States, MDS occurs in every 3 to 4 individuals per 100,000 people, and this risk increases with age. The majority of MDS patients die from intrinsic causes of the disease and not due to its progression to acute myeloid leukemia, AML. Cytogenetic analysis is used to assess risk and to choose the most effective treatment. The diagnosis of MDS is usually received upon abnormal complete blood count, CBC, and dysplasia. It is confirmed through a bone marrow aspiration and biopsy. The aspiration evaluates cellular morphology and blast percentage, while the biopsy evaluates bone marrow cellularity and structure. One additional test is the analysis of bone marrow cytogenetics. This test determines prognosis and is used to select therapy. MDS has a heterogeneous cytogenetic pattern with 20 different alterations under 5 different subgroups as seen in the diagram. This diagram shows 5 different cytogenetic classifications according to prognosis or overall survival. Very good prognosis is predicted by a single alteration of chromosome 11q deletion or the loss of the Y chromosome. Good prognosis is predicted by a single anomaly on the following chromosomes and also double abnormalities, including a chromosome 5q deletion. Poor and very poor prognosis is also predicted by 3 or more abnormalities. Many MDS patients experience additional cytogenetic changes associated with progression to AML or worse survival. There are 3 more commonly used prognostic scoring systems for MDS, and they are ordered from oldest to newest. So the International Prognostic Scoring System, also known as IPSS, uses a score comprised of blast percentage, number of cytopenias, and cytogenetics. It is simple and reproducible. However, it is not precise for lower risk patients, and it has less of an emphasis on cytogenetics. The IPSSR, which is a revised edition of the first, has more of an emphasis on cytogenetic analysis by including the new cytogenetic MDS score. Both IPSS models are developed for newly diagnosed patients and excludes those with proliferated MDS, chronic myelomonocytic leukemia, and those who have received prior treatment. The third model, the Global MD Anderson Cancer Center MDACC model, accounts for these limitations as it evaluates MDS patients at any stage of the disease. It also accounts for the heterogeneous nature of natural history and prognosis along lower risk individuals by including a prognostic scoring system just for this group. And it found that lower risk patients with poor prognosis accumulate more mutations. However, the 3 systems do not consider the impact of comorbidities on MDS. MDS is more frequent in older patients with comorbidities. A study used the Adult Comorbidity Evaluation 27 on a cohort of 500 MDS individuals and found significant impact of comorbidities on MDS survival, and this indicates a need for a prognostic system that is inclusive of comorbidities. So cytogenetic alterations can reveal information regarding prognosis, treatment, and the outcomes of allogenic stem cell transplantation. So in terms of prognosis, mutations in RUNX1, TP53, EZH2 are associated with adverse prognosis. Mutations in the splicing factor SF3B1 is associated with good prognosis and prolonged survival. As seen in the graph, SF3B1 is the most common mutation among MDS subtypes. In terms of treatment, MDS patients with TET2 mutations correspond to higher response rates to azacitidine. And looking at the graph, TET2 is one of the most common mutations in MDS. And MDS patients with TP53 mutations correspond to high response rates to decitabine. Information about cytogenetic alterations can also predict the outcomes of allogenic stem cell transplantation. For example, MDS involving TP53, RUNX1, ASXL2, JAK2, and RAS pathway gene mutations are associated with shorter survival or relapse-free survival, and TP53 mutations are particularly adverse. Molecular alterations also aid the development of clinical trials for MDS with mutations in splicing genes such as SF3B1, SRSF2, etc. Some treatments targeting these mutated genes have already been FDA approved for AML, and we expect to see the same for MDS in the future. However, more evidence is needed to use mutation analysis to evaluate prognosis and select treatment in the clinic.
So hi everyone, I'm going to be talking about a generalized or a summary of risk-adapted therapy. We're going to generally categorize between lower risk and higher risk and newly diagnosed MDS. And there's also cases of relapse or refractory lower and high risk MDS. However, given time constraints of today's presentation, we'll be mostly focusing on the first two. So this table here presents a summary of the generalized risk categories that I just discussed, as well as the treatments and recommendations for treatment. So in case of newly diagnosed and also refractory lower risk MDS, current treatments include erythroid stimulating agents, which is recommended for patients presenting with significant anemia, but without cytopenia. There's also lenalidomide, which is recommended for patients presenting with anemia, good platelets, deletion 5q, but without thrombocytopenia. For patients who fail either ESA or lenalidomide, azonucleosides are recommended. However, in the event that the above three treatments do not induce a favorable patient response, there are also the options of immune therapy and allogeneic stem cell transfer. However, this is generally reserved for younger patients. In the case of newly diagnosed high-risk MDS, azonucleosides are actually the standard frontline treatment. In the event that patient response is not optimal, then there are options of AML-like chemotherapy and, again, allogeneic stem cell transfer. But similarly, these are reserved for younger patients, especially those presenting without poor cytogenetics, particularly the TP53 mutation. So to sort of summarize the information that was just discussed in the previous table, this diagram here summarizes the treatment algorithm recommended by the Department of Leukemia at the University of Texas MD Anderson Center. So following diagnosis of MDS and determining the stage of disease, patients are categorized as either lower risk or higher risk MDS. For lower risk MDS patients, initial treatment options include erythroid stimulating agents or growth factors, lenalidomide, and azonucleoside, again, based on the recommendations that we discussed in the previous table. And for patients not responding to those treatments, then they do have the options to enroll in a clinical trial or undergo allogeneic stem cell transfer. For higher risk MDS patients, allo-SCT is marked in this first box here just because they recommend that, that you begin the donor matching process as soon as possible in the event that allo-SCT is recommended in the future. However, as mentioned, the azonucleosides are the standard frontline treatment for these patients. And when that fails, options include AML-like chemotherapy, enrollment in a clinical trial, and again, allo-SCT.
About 3 more minutes, Andrew.
Thanks, Wilson. So at this point, we kind of had a general overview of the treatment algorithm and available treatments, but I would like to go into a little bit more detail on allogeneic stem cell transplants, just because they are very significant in that they're only potentially curative treatment for MDS patients. However, given that, you may be wondering, based on this previous diagram, LSCT is depicted as a last resort treatment. And the reason for that is that because it is severely counterbalanced by a high incidence of treatment-related mortality, as well as the potential and vulnerability of developing graft versus host disease. And this is especially important for older patients who are less able to tolerate invasive procedures such as allo-SCT. And so there are 3 main factors really that go into considering a patient who's potentially undergoing allo-SCT. Again, that's age. Younger patients are preferred over older patients, as well as the availability of an optimal donor. There's 3 types of donors: matched sibling donors, haploidentical related donors, as well as unrelated donors. These are categorized based off of the degree to which their human leukocytic antigens match the patients. And lastly, there's also consideration of the presence of high-risk mutations, as these allogeneic stem cell transfer might not be worth the risk in these cases. So to give you a little bit more of a graphical perspective, right, When comparing the development and the cumulative incidence of GVHD between different donor types, you see that matched sibling donors and haploidentical related donors fare much better than unrelated donors. There's a stark difference, between approximately 15% and 30-35% for the unrelated donors. So it's generally recommended that allo-SCT is reserved for patients who do have an optimal donor. And this graph here is showing the differences in survival, patients undergoing allo-SCT with and without the high-risk TP53 mutation. You can see patients with a high-risk TP53 mutation have a far lower overall survival than those without. Right. So in these instances, it may not be worth the risk of treatment-related morbidity in these cases. So just to conclude, MDS comprises a heterogeneous group of myeloid malignancies. There's 3 existing risk stratification systems: the IPSS, IPSSR, and MDACC. And again, allo-SCT is the only curative treatment of MDS, and the considerations that go into whether or not treatment is underwent is age, donor availability, and high-risk mutation. So with that, thank you for listening to our presentation, and we'd be happy to accept any questions at this time.
Tu micro está malogrado, Bruno, por favor.
Ok, las disculpas del caso. El siguiente expositor es el estudiante... Un momento. Disculpen. Las siguientes expositoras son las estudiantes Grace Young y Bilal Hamid, estudiantes de la Universidad de California, miembros del Círculo Internacional de Genética, capítulo de Estados Unidos, presentando el tema denominado leucemia linfoblástica de células T. Si hay alguna pregunta para los expositores, por favor escribirla al final de la presentación. Muchas gracias.
Hello, everyone. Grace and I will be presenting on T-cell lymphocytic leukemia, and we'll be focusing on the past decade and changes in research.
My name is Grace, and I'm a third-year human biology and society major at UCLA.
And I'm Bilal, and I'm a second-year microbiology major at UCLA. So let's dive into some background. TPLL, which is the T-cell prolymphocytic leukemia, and what we'll be talking about today, was recognized as a distinct disease from chronic lymphocytic leukemia in 1973 due to its unique clinical presentation and very poor clinical outcomes. Initially, there was a small cell variant called T-cell chronic lymphocytic leukemia, which is mentioned in a lot of literature in the past 15 to 20 years, but that was incorporated into the TPL diagnosis due to the genetic and clinical similarities seen in the disease. Recently, a lot of research has been focused on understanding the molecular and genetic characterizations of the disease in order to figure out treatments and figure out avenues of therapeutics. So a little overview of the disease, T-cell prolymphocytic leukemia. The word prolymphocytic in the name of the disease is actually a misnomer because the leukemia is from post-thymic T cells and not the pre-thymic immature cells. And it appears mainly in the blood, spleen, bone marrow, and lymph nodes, just like a lot of other leukemias. There are 3 main variants: a typical medium-sized tumor cell variant, which accounts for approximately 75% of cases, a small cell variant that was initially called TCLL, which has smaller tumor cells with small condensed chromatin and nucleolus, and the cerebriform variant, which has very irregularly shaped cells and is slightly more aggressive and has worse prognosis. Overall, this disease is very, very rare, and it accounts for just about 2% of the 300,000 leukemia cases worldwide. For risk factors, there's actually only one main risk factor due to the rarity of the disease, and that's pre-existing ataxia telangiectasia, which is a recessive genetic disorder which results in neurological development or degeneration, chromosomal instability, and immunocompromised issues. And the risk factor is pretty significant because it reduces the median age of symptom onset from about 60 to 30, and it results in a large increase of translocations, resulting in more aggressive forms of the cancer. It also increases likelihood of developing other cancers other than T-cell prolymphocytic lymphoma, and it includes other T-cell lymphomas and B-cell lymphomas. So the clinical features of this disease, overall, it's a very aggressive disease. 73% of cases have splenomegaly, which is the enlargement of the spleen. 53% show lymphadenopathy, which is the irregularity, tenderness, and enlargement of the lymph nodes throughout the body. 40% of cases show hepatomegaly, which is the enlargement of the liver. And 20 to 30%, depending on the literature, show skin lesions, primarily on the arms and torso. A small proportion of patients are asymptomatic and are denoted in the literature as inactive TPLL. But their median time before the disease becomes aggressive is 33 months, and after their disease becomes aggressive, they have a similar disease progression beyond that point. And overall median presentation occurs at 63 years old with a 1.3 male to female ratio. So the important cytogenetic factors that characterize the disease is the inversion of the 14th chromosome, a translocation between 2 points of the 14th chromosome, and a translocation between the X chromosome and the 14th chromosome. These primary cytogenetic variances occur in 90% of cases and result in the TCL1 protein and a rearrangement of the T-cell receptor locus, both of which affect T-cell proliferation and vastly increasing the chances of a mutation and other leukemias. There's also other notable events, which is the isodicentric 8p1 TA8 translocation and the trisomy 8q translocation, which appears in 70% of cases, and the deletion or missense mutation in ataxia telangiectasia mutated locus in 65% of cases. The second mutation causes issues with the AT kinase, which results in issues with the growth and synthesis portion of the cell cycle, resulting in irregular cellular development and mitosis.
Great. So I'll be taking us through diagnosis and treatment. And so first with diagnostics, similar to many of the cancers mentioned previously, for diagnosis, you would need an immunophenotype, specifically for TPLL, and in order to differentiate it between other T-cell leukemias, you need a presence of CD26 and TCL1 protein expression, which makes sense given the mutations that are often present in TPLL, as Bilal has mentioned previously. Similarly, we also usually get a blood count, T-cell analysis, and karyotype in fish, which makes sense, especially for TCL1 and MTCP1. And with a lot of cancers that involve tumors and active tumors, imaging such as a CT are also needed to measure the burden and organ involvement. And so on the screen here, you can see the criteria set aside by the WHO in 2019, now recognizing TPLL as its own leukemia. And so in order for diagnosis, you need either all 3 major criteria or the first 2 major criteria plus 1 minor criterion. So as you can see on the left, the major criteria often involve the T-cell clonalities, as well as on the 3rd bullet point, abnormality of 14q, which involves TCL1, the 2 dimers. And on the right, for the minor criteria, a lot of those involve smaller chromosomal abnormalities that can also affect the clinical course of the disease, as well as organ involvement on the last bullet point. And for treatment, because it's such a rare and also aggressive disease, there is unfortunately typically only one treatment option for patients experiencing an active disease, which is alatuzumab. And alatuzumab-based chemotherapy is most common, and it is sometimes paired with pentostatin. So Jane et al. In 2017 reviewed medical records from 120 consecutive patients with TPLL who presented to MD Anderson between 1990 and 2016. It is one of the most recent comprehensive studies, or longitudinal studies, with this amount of patients. So it was really a really prominent study that came out when it did, and so they found that the remission rate is pretty high, around 80%, with a complete remission rate a bit higher for when alatuzumab is paired with pentostatin. But still, even those achieving complete remission can also relapse as well, which is why it often requires additional treatments, including hematopoietic stem cell transplantation, which we've heard a lot of recently, especially applied towards aggressive cancers. And ultimately, a lot of TPL patients have to resort to clinical trials as researchers are still trying to figure out what new therapeutics can really combat this disease. And Bilal also mentioned that there is a possibility that a patient may present with an inactive disease, which usually had a 33-month point until it became active. And so for those patients, observation is the most recommended clinical course. It is minimally invasive, and you really want to wait until you have such an aggressive chemotherapy.
I'm sorry for the interruption. I just wanted to let you know you have 5 minutes left.
Oh, thank you. And so for prognosis and relapse, the median survival Jane et al. Found for their cohort was 19 months from the time of diagnosis, which agrees with the literature that says that the median survival is between 1 to 3 years. And so if you have mutations involving TCL1 and AKT, as Bilal mentioned earlier, that is also associated with a worse prognosis, as is lower hemoglobin count, higher lactate dehydrogenase levels, and white blood cell counts at the time of presentation. And Bilal also mentioned that there are additional subtypes, most of which are pretty aggressive and unresponsive to treatment, although we kind of are unable to risk stratify these patients because of a lack of general knowledge and just because this cancer is so aggressive. So this warrants a close observation of response because it has extremely high remission rates, and the fact that small cell and pseudoform variants have a worse prognosis. And for future directions, this lack of knowledge in this area generally points to warranted research investigating long-term survivors, especially with the hematopoietic stem cell transplants, since it's a new technology that has really blossomed over the last 10 years. We also need to identify clinically tractable pathways in order to produce target therapies, as alatuzumab is also used in other mature cancers. It's generally used for really aggressive cancers, and there still needs to be targeted therapy specifically for patients. And because TPLL also presents with a lot of complex karyotypes, it's really hard to kind of have a general response when the disease itself is so heterogeneous. Going to my next point about molecular tools to predict prognosis, as Bilal mentioned, there's really a lack of risk stratification in TPLL, which is a problem when a lot of patients present with marker chromosomes, or they have additional chromosomal abnormalities that is really hard to track. And interestingly enough, with marker chromosomes, usually a greater number of marker chromosomes indicates a poor prognosis, but in TPLL specifically, that hasn't been found to be the case. So it really goes to show how there's so much more to this disease than we know on the surface. And even with the last 10 years of research, we had a lot of gains in understanding the specific abnormalities that are associated with the disease, but further research is needed to determine what other abnormalities can be paired, especially in complex cases. So with that, thank you everyone for listening, and here are references, and we are more than happy to answer any questions in the chat. Thank you.
Ok. El siguiente expositor es el estudiante Franklin Calderón, estudiante de la Universidad Nacional Pedro Ruiz Gallo, presentando el tema denominado Huntington Disease. Si hay alguna pregunta para el expositor, por favor escribir en el chat de la plataforma al finalizar la exposición.
Y un momento voy a comenzar a compartir pantalla. ¿Se puede apreciar?
Sí.
Sí, pero tienes que ponerlo en presentación, Franklin.
Se ve.
¿No se escucha?
Sí, estoy tratando de presentar, solo que tengo un problema aquí. Por favor. Ok. Bueno, buenas tardes entre todos, doctor y estudiantes también profesionales que están aquí. Bueno, en esta oportunidad me toca exponer acerca de la enfermedad de Huntington. Mi nombre es Franklin Calderón, como ya lo mencioné antes, estudiante de la Facultad de Ciencias Biológicas de la Universidad Nacional Pedro Luis Gallo. Y bueno, vamos a comenzar sin más preámbulos. Lo que sí le voy a pedir, voy a apagar un poco la cámara porque está un poco lenta. Realmente se va a estar haciendo mi audio, por favor. Pues, no importa. Bueno, vamos a comenzar. La enfermedad de Huntington. Bueno, la enfermedad de Huntington se va a transmitir, es una enfermedad mayormente de autosómica dominante. Quiere decir que solamente basta un alelo para que se pueda transmitir al hijo. Y en este caso, esta enfermedad de Huntington se localizó en la región del genoma que está involucrada como en el cromosoma 4, específicamente en el cromosoma 4p1.6.3, en la cual fue identificada en el año 1993 aproximadamente. Y este gen responsable de la enfermedad, que es el gen IT15, este gen se va a extender en una región del ADN que aproximadamente tiene unos 180 kilobases, en la cual va a contener 67 exones y va a aportar una amplificación génica que va a estar producida por la repetición del trinucleótido, citosina, enina, guanina. Este gen lo que va a codificar lo que es un ARN mensajero de 10 kilobases que se va a traducir en una proteína. de 350 kilodaltons, que es la proteína huntingtina. Esta se va a encontrar en todas las neuronas del cerebro y cuya función normal, bueno, era desconocida. Ahora esta mutación génica va a provocar lo que es en la enfermedad consistentemente, justamente en lo que es la amplificación del trinucleótido de citosina en guanina, en un segmento del ADN que es inestable y que está localizado en el exón 1 del gen. Mayormente esta enfermedad de Huntington se va a caracterizar por trastornos cognitivos, una alteración motora, sí, hasta psiquiatra. A nivel celular, la huntingtina mutante va a producir una disfunción, lo que es neural, y una muerte a través de varios mecanismos, que va a estar incluida la alteración como la proteástasis, la transcripción y la función mitocondrial. Asimismo, también la toxicidad directa de la proteína mutante. Como podemos observar en la imagen de aquí, en donde podemos observar que el cuadro, las imágenes, los iconos con los cuadros rojos, quiere decir que está el gen afectado, y los azules, los iconos azules, quiere decir que está normal, ¿no? Es un individuo normal, en la cual va a poseer 1, 2, 3, 4 y 5 hijos, en donde a su vez la mayor parte de los hijos van a estar con la enfermedad, 50%, 50 aquí, 50 aquí, 2 hijos mutantes, perdón, 2 hijos con la enfermedad y 2 hijos sanos. Al igual que en esta imagen de acá, podemos observar lo que es un individuo con la enfermedad y un individuo sin la enfermedad, en donde a sus descendientes va a tener un 50% de descendientes con la enfermedad y 50% de descendientes sin la enfermedad. Bueno, el número de repeticiones, quiere decir el trinucleótido citosina y guanina, va a ser normal cuando va a presentar una cantidad menor a 28, en donde su resultado es que no va a desarrollar esta enfermedad de Huntington. Asimismo, tenemos de 29 a 34, en donde su resultado no tendrá la enfermedad de Huntington, pero la siguiente generación está en riesgo de desarrollarlo, como sus hijos o como sus nietos. Después está dentro de 35 a 39, en donde el resultado va a ser que algunos individuos puedan desarrollarla de manera tardía, siendo una generación en riesgo. Y mayor a 40, cuando su resultado específicamente va a desarrollar la enfermedad de Huntington. Bueno, acá tenemos el cromosoma 4, en donde prácticamente este cromosoma va a presentar lo que es la enfermedad de Huntington, con las repeticiones de 35 nucleótidos de citosina, adenina, guanina, en donde su resultado va a producir la proteína no mutante, y por consecuencia su neurona va a ser una neurona saludable. Pero también vemos aquí el cromosoma 4, en donde va a presentar un incremento del trinucleótido citosina, adenina, guanina, en donde va a contraer la enfermedad y va a producir una proteína mutada, y por consecuente una degeneración de una neurona, como podemos apreciar acá. Bueno, acá tenemos lo que es nuevamente el cromosoma 4, prácticamente lo que es la parte del gen de la enfermedad de Huntington, en donde vamos a poder extraer una parte o amplificar esa parte, como podemos aquí ver en la imagen, y a través, acá podemos ver lo que es un triplete de codón, lo que va a codificar lo que es un aminoácido en concreto, y podemos ver acá los trinucleótidos de citosina, adenina y guanina, en donde este triplete va a codificar lo que es el aminoácido de glutamina. Pero al haber un triplete de citosina, adenina y guanina mayor a 36 repeticiones, se va a decir que es un gen mutado, como podemos apreciar en la imagen. Bueno, en la siguiente imagen es donde van a ser los mecanismos celulares patogénicos de la enfermedad de Huntingtina, en donde en la figura número 1, en el número número 1, podemos apreciar que esta Huntingtina se va a traducir para producir lo que es una proteína huntingtina. Esta a su vez va a estar unida a lo que es el fragmento del exón 1 del amino terminal, en donde prácticamente va a ser resultado de un empalme aberrante. La longitud del tracto también de la poliglutamina, que es el poliQ. Mayormente lo que es en el número 2, vamos a apreciar lo que es la huntingtina nativa de longitud completa, en la cual se va a dividir mediante proteólisis para generar fragmentos de proteínas adicionales. En el número 3, podemos observar lo que son los fragmentos de proteína que va a ingresar al núcleo. Y en el número 4 podemos observar que los fragmentos se van a retener en el núcleo a través de lo que es la autosecesión, la oligomerización y lo que es la agregación. Lo que va a conducir a lo que es a la formación de inclusiones, como podemos apreciar en esta imagen. Asimismo, es un proceso que va a causar lo que es una desregulación transcripcional a través de lo que es el secuestro de otras proteínas y a través de otros mecanismos definidos como de manera incompleta. En el número 5 podemos observar lo que son fragmentos de huntingtina que se van a oligomerizar y se van a agregar en el citoplasma, como podemos ver aquí. En el número 6 vamos a ver la agregación de la huntingtina que se ve agravada mayormente por la alteración de la red de proteástasis, que va a estar relacionada con la enfermedad, lo que también va a producir alteraciones celulares globales.
5 minutos, Franklin.
Ok. En el número 7 tenemos las formas aberrantes de la huntingtina, que van a dar resultados lo que es alteraciones globales, lo que va a incluir lo que es disfunción sináptica, lo que es también una toxicidad mitocondrial y lo que es un transporte axonal malo, ¿no? Deficiente. Muy bien. Tenemos la expansión de la poliglutamina, que va a conducir a una alteración del plegamiento y de otras modificaciones post-traduccionales de la proteína Huntington y acumulaciones nucleares de fragmentos de la proteína mutada. Como podemos apreciar aquí, vemos el trinucleótido de citosina e guanina, en la cual va a tener una cantidad de 10 a 26 repeticiones. Esto quiere decir que va a producir una proteína de huntingtina, pero va a producir la normal. A diferencia, en esta imagen podemos apreciar de las repeticiones del trinucleótido de citosina e guanina, que va a ser mayor a 35 repeticiones y va a dar como resultado una proteína mutada, como podemos apreciar en la imagen. Bueno, dentro de las manifestaciones clínicas tenemos lo que son síntomas extrapiramidales, como la coria, que se va a dar mayormente dentro de los 45 años a 55 años. En un 10% de los síntomas aparecen antes de los 20 años, mayormente predominando la variedad de rígido asténico, que es la forma de Westphal, y síntomas parkinsianos y tardíos. Asimismo, los trastornos oculomotores, que son los trastornos de motilidad ocular, miradas lentas e inexactas, dificultad para iniciarlas y sin acompañar movimientos de cabeza, movimientos de seguimiento, son lentos y convergencia está disminuida. Y también tenemos los que son trastornos cognitivos, como problemas de atención, cálculo, afaxia, apraxia, agnosia y demencia. Y bueno, y las manifestaciones psiquiátricas, que son los trastornos del ánimo, la depresión mayor y un trastorno obsesivo compulsivo. Acá podemos ver diferentes personas con la enfermedad de Huntington, en donde se puede apreciar a un señor, una niña y a una mujer anciana. Bueno, y eso sería todo de mi parte en esta oportunidad y espero que haya sido un pequeño aporte sobre esta enfermedad de Huntington. Muchas gracias.
Thank you, Franklin, for your presentation. Our next presenter is Dr. Victor Pereira Sanchez. He is an advisor of the International Circle of Genetic Studies and comes to us from the Department of Child and Adolescent Psychiatry at New York University's Grossman School of Medicine. He will present the topic of neuroimaging of attention deficit and hyperactivity disorder, ADHD. If there are any questions for the exhibitor, Please write them in the platform chat at the end of the presentation. Thank you.
Thank you very much, and good afternoon from New York City.
Víctor, perdón, ¿cómo estás, Víctor? Puedes hablar en español si quieres, no hay problema.
Okay.
It's whatever you want to, okay? Whatever you prefer.
Okay. I prepared this in English, so.
Okay, that's fine. Good. Go for it. Thank you.
Okay. Perfect. Thank you. I'm gonna share my screen. Y cualquier preguntita también me la pueden poner en español en el chat. Ok. ¿Sí ven mis diapositivas?
Sí, doctor.
Tito. ¿Ven las diapositivas en grande, no?
Sí.
Perfecto. Muy bien. Muy bien. Okay. Yeah, again, good afternoon from New York City. I am a psychiatrist, originally from Spain and trained in Spain in general psychiatry. Now I'm at the second year of a child and adolescent psychiatry fellowship training at NYU and in the Department of Child and Adolescent Psychiatry. And I'm also a PhD candidate at the University of Navarra in Spain. And what I'm going to talk about refers more to my PhD research that has been in trying to study the biological basis of attention deficit hyperactivity disorder. So I presented some of this data last year to some of your colleagues, but I have also thankfully updated some material and also with publications of mine. So just a summary. So ADHD is the attention deficit hyperactivity disorder. So this is a neuropsychiatric disorder. It's a neurodevelopmental disorder. So its origin is not fully understood, but it has a very strong genetic component. So we know that the heritability of ADHD is around 75%. So it's a very strong genetic component, although the transmission of ADHD is polygenic, and also on the one hand there are many genes. There's no any gene that is necessary to be mutated to cause the disorder, and also it's multifactorial because also a component of ADHD seems to be environmental and related to possibly to early adverse factors or exposure during pregnancy or during birth to tobacco, to drugs, to other toxics. But there are still, we don't know still there's no any single factor that causes ADHD. So okay, and this disorder is the most common disorder in children and adolescents. So around 5 to 10% of children and adolescents from all over the world have this disorder. And the main symptoms are inattention, hyperactivity, and impulsivity. And this is a disorder that causes a lot of trouble to these kids. In half of them, the disorder persists into adulthood, and well, during childhood. So the main problems are especially at school. So this lack of attention, with this impact in their grades, and also this hyperactivity and impulsivity that makes them to get into trouble with other colleagues, with teachers. During adolescence, this impulsivity can be really problematic because it would make many of them more prone to use drugs or to take to fall into risky behaviors in terms of drug abuse, sex, driving, get into more trouble. And in adulthood, for those who have persistent ADHD, the main symptoms are especially inattention, although they may also have some hyperactivity and impulsivity. And adults with ADHD, they have a higher risk of involved in car accidents, in being fired from jobs and have a job instability, and also have instability in their intimate relationships. So they have higher chances of divorce, also substance use, and of problems with justice. Okay. And the treatment of ADHD is, here in the talk, I'm going to talk more about the brain imaging, but I wanted to give you an overview of ADHD before. And the treatment for ADHD has 3 pillars, but the most effective is medication. So there are different kinds of medications, especially stimulant medications that are methylphenidate or derivatives of amphetamine. So stimulant medications try to increase the levels of dopamine in children, in the brain of children and adolescents with ADHD, increase the transmission of dopamine, and they are very effective in most of the patients. Okay, and so just a spoiler about brain imaging in ADHD current state. You can search this paper in the journal Current Opinion Psychiatry. It's open access. It was published last month or this month. This March. So here, my supervisor in New York, Dr. Castellanos, who's an expert in the field of brain imaging and ADHD, and I, we surveyed the field of research on ADHD in 2019 and 2020. And the spoiler is that brain imaging in ADHD also is true for all psychiatric disorders, is not yet ready to be applied for clinical management. So it's only useful in research, but we cannot still diagnose people or any psychiatric disorder with brain imaging, nor use it for other clinical purposes at an individual level. Okay, so brain imaging in psychiatry is currently the most probably one of the most exciting areas of psychiatry research, because we know that the mental disorders are mediated by damage of the brain, but this damage we don't still understand well, because it's not like an Alzheimer's disease where you can more easily do a PET scan or other brain imaging and you can see that the brain is having changes, or it's not like a stroke where you can see the lesion, the vascular lesion in the brain. In psychiatric disorders, brain imaging is being helpful in research, but we are trying to find, and we have not yet found any reliable marker in the brains of people with mental disorders. And brain imaging in psychiatry evolved a lot from first there was no imaging, so they were studying the brains post-mortem, the brains of people with lesions like this guy, Phineas Cage, that had this very important injury, and got a personality disorder after this injury. Also, earlier imaging techniques, so X-ray, were not very useful because X-ray is very good to see the bones, but the brain is a soft tissue. So there are different techniques for brain imaging. I want to highlight that the main types are 2, so functional and structural. So in structural brain imaging, we want to see the anatomy of the brain with high resolution, and there the best technique is MRI, the magnetic resonance imaging. And then the functional functional imaging. So the functional imaging, we don't only want to see the anatomy of the brain, but we also want to see how different areas of the brain are being activated and are how different parts of the brain are connecting to each other. So yeah, here are different examples. This is a computerized tomography, so this is based in X-rays. So here, yeah, we see the bone very clearly. We see some of the brain, but the definition is not very high. But for anatomy, yeah, structural MRI is the best. And also for functional imaging, especially for neurodegenerative disorders, these PET techniques are very, very useful because PET is based on positrons and so in nuclear medicine, and they can find the radioactive signals associated to specific structures of the brain or to specific functions of the brain. But the MRI is by far the most used because it's relatively simple and is safe because it's based on magnetism that doesn't have any radiation. And especially for child and adolescent research, we prefer not to use radiation, so we prefer MRI. And there are different kinds of MRI. I talked already about the structural MRI, so very good to see the anatomy. In this case, we'll see this guy had this elongation of these ventricles, and this guy had psychotic symptoms. And when these ventricles were drained, so the symptoms of the patient improve a lot. There's another way of using a structural MRI, but called DTI, that will try to identify rather than the gray matter, rather than the neurons, it tries to identify the axons and to see how axons, where are the axons located, and how the axons are connecting different parts of the brain and in which direction. There's also another anatomic form of MRI called the spectroscopy. So here, this is a structural MRI, but it can measure different chemicals through a spectroscopy. It can identify different elements in the brain. So, for instance, the levels of glutamate or glycine or other neurotransmitters in different parts of the brain. And this is especially useful to study brain tumors because you know that some brain tumors are rich in a specific chemical component, and you know if you identify that component in a brain tumor like this will be a marker of prognosis and help you diagnose also the tumor. And then and this is very much used in psychiatry is the functional MRI and resting state functional MRI. So in the functional MRI, well, here we have an anatomic MRI, but what we are seeing here is activation. So we are seeing we are having the patient in the scan for a time, and we will ask the patient to do certain activities. For instance, to clap their hands, or to memorize a list, or to do other cognitive or physical tasks. And we are seeing. We are measuring the flow of oxygen in the brain. So we are trying to see what parts of the brain are activated when the patient is performing a task. So you will say that this task is related to this area of the brain. Okay, and then there's a variant called the resting state of MRI, in which rather than asking patients to do any task, we would ask them to just stay still and to be bored for 10, 20 minutes. And what we are trying to see there is not peaks of activation because there's no activity. Well, there's an activity that is rest. What we are trying to see is the connectivity. So in a resting state, how the brain networks are working, are connecting with each other, because that seems to give a lot of information about how the brain how the brain networks are functioning in patients, in healthy patients and patients with mental disorders. So more specifically for ADHD, so in terms of anatomic ADHD, on the one hand, we have findings from the previous decade that of ADHD with children and adolescents. So they recruited children and adolescents at the NIH in the US, and they follow them for 10, 15 years, and they were doing MRIs every couple of years. So if this video works, well, doesn't work. Don't worry. But the thing here is that this group found that children and adolescents with ADHD, as a group, they tend to their brain development is slower. So there's a delay of around 2 years. So that points out at ADHD as a neurodevelopmental disorder. So a disorder in which the brain development goes slower. And in some cases, it not only goes slower, but there are some parts of the brain that don't develop, don't get to fully maturate. Okay. More recent studies, they are cross-sectional, they are not longitudinal, but they are in this group, the NIGMA. It's an international consortium, so it tries to bring big data from multi-centers, from multi-center studies. So many different hospitals and universities, they contribute with data. So they have thousands of patients and thousands of control individuals. And what they have found is that also as a group, children with ADHD have lower volumes in subcortical areas, especially the accumbens, amygdala, hippocampus, and also they have lower total brain volume, and also lower cortical thickness, so their gray matter is less thick, and also lower cortical surface area. So this points out ADHD as effective as it supports the idea that ADHD is a neurodevelopmental disorder, and the brain of children and adolescents with ADHD as a group is different, is less mature, is smaller. But again, these are group level differences. There are no individual differences, and the differences are very small in side effect. Sorry, in effect size. So there are differences, but they are very subtle. And then also, studies with DTI have also suggested and have also found differences. So it seems also that the axons of the white matter in children with ADHD is also immature as a group. There are disruptions there. And then as for functional MRI, so on the one hand, with task-based fMRI, so giving the patients specific tasks and seeing what areas of the brain are activated. So there have been many studies, but most of these studies have been very small in sample sizes, and their findings are not very, they don't converge across the studies. So when you do a meta-analysis of these brain imaging studies, you don't find that, and you correct for multiple comparisons, you don't find areas of the brain where these findings do converge. So we are still pretty much ignorant about what are the real differences in fMRI in children and adolescents with ADHD. Although some of the, there are some studies that have found areas of the brain that are differently activated or deactivated with medications in children with ADHD. And this is the area where I have been doing research, is the resting state fMRI. So in the resting state fMRI, what we are trying to see is the brain in a default state, so in a resting state without doing any task, and we are seeing how the oxygen flows in each part of the brain, and we are doing basically correlation analysis. So we are trying to, well, we correlate, so we see how oxygen flows here, and we see, well, and we calculate which other areas of the brain have a pattern of oxygen oscillation that is similar to this area. So with that, we get functional connectivity maps. And these maps mean that if their oxygen fluctuations are very, very correlated, are very synchronous, so to speak, that means that these areas of the brain, even if they are distant, you know that they are probably connected. They are talking to each other.
Dr. Victor, 5
Yes.
minutos.
5 minutes. Yes, perfect. Okay. So, and with that, many previous research has been able to identify the networks of the brain in resting states. So how different areas of the brain are correlated to other areas of the brain and to create networks. And one of the hypotheses here with ADHD is that children and patients with ADHD, they have this balance in how brain networks work. So the network that is the default one network, that is the network that it should be more active when patients or when people in general are bored, is in children with ADHD is overactive in moments where they should be paying attention to tasks. So when they should be, for instance, in the classroom doing a task, they should activate their attention networks and deactivate the default mode network. But ADHD makes them more difficult to deactivate the default mode network. Okay, so we are trying to study as well. And this is my second study. We are trying to see if medications have an effect and change the pattern in which brain networks function. But again, so the field here, the research field is still very immature, and it needs a lot of improvements in methodological improvements and also increasing sample sizes. Okay, but yeah, we need more transparency as well in this research. We need more big data, and also, well, there's a promise, and there are many of these big groups with multicentric data. They are combining genetics and brain imaging and trying to find what is the relationship between genetics, brain imaging, and behavior. Because it seems that brain imaging seems to be the mediator between genetics and symptoms. And yeah, this is the first GWAS in ADHD. The Manhattan plot is like where I live. Okay, so in summary, there are differences in the brains of children and adolescents with ADHD, with healthy controls, people without ADHD, but these differences are subtle and are at a group level. And right now, we are the field needs to grow and to improve the methodology, their samples, and also to incorporate other data as genetics. And we are still, I think, far away from using brain imaging in psychiatry. Okay, thank you very much.
Thank you, Dr. Sanchez, for your presentation. If there are any questions, you can feel free to leave them in the chat. Our next presenters are students Justin Yi and Alaire Zhao. Justin and Alaire are students at the University of California, Los Angeles, and members of the International Circle of Genetic Studies, USA chapter. They will be presenting on the topic of ring chromosome. If there are any other questions for the exhibitors, please write them in the platform chat at the conclusion of their presentation. Thank you.
Justin, you good?
Can you, Justin, please?
What's going on? Okay. Hi, I'm Alaire Zhao. I'm a second-year computational science and biology major, and we're going to here present a thing on ring chromosomes.
Can everyone hear me now?
Okay, so you were muted. I was so confused.
Oh, my bad. I think there was something wrong with my computer and microphone. All right, look. Yeah, so my name's Justin. Sorry for the technical difficulties. I'm also a student at UCLA, and I'm working with Alaire, presenting about ring chromosomes.
Okay, so just to start with some background about ring chromosomes. A ring chromosome is any chromosome where the distal ends are fused into literally a ring. Usually this results in a net loss of genetic material. So far, there have been cases reported for a ring variant of every chromosome. These are typically de novo and not inherited from parents. And it has a level of mosaicism that can vary from not at all mosaic to like a low level mosaicism. And it's rarely seen in a high level mosaicism, like across the entire patient. Ring chromosomes themselves are fairly rare. Up to about 1 in 30,000 births is the most common within like ring chromosome 6. The nomenclature in karyotypes is denoted with an R parentheses N for the number of the chromosome, with a PAQB after that, where A indicates the breakage on the P arm and B on the breakage at the Q arm. Ring chromosome causes typically severe phenotypes in infants. However, the mechanism of its formation and the elucidation of how exactly it causes these phenotypes is not fully understood yet. And in many cases, it's observed that the phenotypes arise mainly from the deletions and not from the ring itself. Okay, cool. So there are a few proposed mechanisms of formation. Justin, if you could, yeah, cool. And click again, please. And again. Awesome. So ring chromosomes usually result from 2 terminal breaks in both chromosome arms, followed by fusion of the broken ends, or from the union of one broken chromosome end with the opposite telomere region. The 2 main types are, as described here, a double-stranded break, where there's a break on the P arm and the Q arm. And then the loss of protective proteins causes the 2 ends to fuse. There's also a telomere dysfunction, which is more rare, where no deletion happens. The telomeres themselves have no protective proteins or a lack of protective protein ends. Sorry, my mouth is right. And the 2 telomere regions fuse. There's also subtelomeric fusion, which is even more rare, where one breakage happens and the other side doesn't have a breakage. And this leads to a semi-loss of genetic material. On the right, there are some like examples of a double-stranded breakage and a telomere dysfunction. Excuse the low quality of the GIF. And figure 2 shows a quick, like what it looks like in a real karyotype.
So, understanding how ring chromosomes replicate in the cell during cell division is important for understanding the general effects of having ring chromosomes. In general, the stability of ring chromosomes depends a lot on how the ring chromosome interacts with its sister chromatid. In the diagram, we show that 2 or 3 scenarios lead to unstable products. And now I'll just quickly go over this diagram. If a ring replicates itself with no sister strand exchange in prophase, the ring chromatids separate at anaphase, resulting in equal-sized rings. If a crossover event occurs, the 2 daughter chromatids form a dicentric double-sized ring. As the centromeres get pulled to opposite poles, and depending on where breakage occurs, deletions and duplications can result. 2 sister chromatid exchanges would result in interlocked rings with subsequent asymmetric breakage, leading to unbalanced products. Since ring chromosomes have a high potential to become unstable at the end of cell division, leading to various chromosomal aberrations like the creation of ring derivatives, this would lead to cells with partial deletions, duplications, fragments, etc. And these cells are also likely to be non-viable. The continual generation of aneuploid cells and the loss of these cells lead to significant growth deficiencies. Pote et al. Coined the term ring syndrome to describe the phenotype of growth retardation and mild cognitive impairment for those with autosomal ring chromosomes, as these phenotypes are present regardless of which number chromosome is in the ring.
So most ring chromosomes appear de novo, which means that they are formed after conception of the infant or the fetus. However, there are some ring chromosomes that are inherited, from mostly maternally inherited. This accounts for about 1% of all ring chromosomes. And this makes sense because ring chromosomes themselves, as stated earlier, are quite unstable. So usually the ring themselves will be lost during meiosis. Not only that, but carriers of ring chromosomes are often infertile. So not similar as the mechanism, the inheritance of ring chromosomes is also not fully understood. There's usually normal dysfunction with a 1-1 segregation where half the conceptuses of like this daughter of the mother cell will have the ring chromosome. And the dynamic mosaicism also could vary depending on the degree of the phenotype. So far, there have been published cases of ring chromosome inheritance in chromosomes 8, 13, 14, 17, 18, 20, 21, and 22, but not of the other chromosomes.
Great. And now this slide details miscellaneous observed effects of ring chromosomes. Ring chromosomes have been observed in every chromosome, so this slide is by no means an exhaustive list. Instead, we would just like to show you the similarities between the different phenotypes of different ring chromosomes. Despite different ring chromosome numbers, the characteristic phenotypes of those with ring syndrome are physical and mental growth retardation. These are the characteristics that define ring syndrome. To point out some common characteristics from the table, the cases presented regarding ring chromosome 1, 2, 6, 22, and 23 all have body dysmorphisms. The cases in ring chromosomes 1, 3, 22, and 23 have some form of mental retardation. For more information on these cases, the last column of the table provides a link to the paper detailing each case, and we can send the PowerPoint to those interested. We'll now move on to 2 different case studies regarding ring chromosomes for a deeper look into the unique effects of ring chromosomes.
Sorry for the interruption. You have 4 minutes remaining.
Okay, so the first case is about a patient who was an infant-born patient, about 13-month-old when the study began. The patient demonstrated phenotypes along the lines of injured uterine growth retardation and having meconium staining of the amniotic fluid. A few months after birth and conception, at about 18 months, the patient also demonstrated severe talipes equinovarus, also known as clubfoot, hypotonia, and low height, weight, and head circumference, patent ductus arteriosus, asthma, delayed speech and motor skill development. The case was put through standard cytogenetic and molecular cytogenetic processes, namely array CGH, to determine the karyotype to be 46XXR10, P15.1, and Q261. So the phenotype of the patient was compared to other patients in the Decipher database. Decipher is a interactive web-based database which incorporates a large host of tools to aid interpretation of genomic variants. And what it does is it retrieves information from other relevant cases, and the patient variant is displayed alongside those cases to understand the context of both the normal variation and pathogenic variation reported at the locus, and then that can help interpretation of the phenotypes. So once comparing the phenotypes of this patient to other patients with similar or identical deletions, but not necessarily ring chromosome formation, we find that the 10q terminal deletion is responsible for the majority of the phenotypes observed in this patient, whereas the 10p terminal deletion is responsible for slightly less, but also some. And here we can see a picture of the patient at 7 months. We observe a down slanting and narrow palpebraal fissures, a broad nasal ridge, stubby nose, smooth philtrum, thin upper lip, averted lower lip, microstomia, and low set ears, and a very short neck. All demonstrations of phenotypes of this ring chromosome and/or deletion.
In the interest of time, I'll just briefly go over this case study number 2, which is an observed ring chromosome 8 and trisomy 9 in a patient with AML. And I just chose this case just because it was interesting because this is the first occurrence of trisomy 9 and a ring chromosome in a patient with AML. Now onto some tools that are commonly used to study ring chromosomes. There are various cytogenetic tools that are utilized to study ring chromosomes, and a few important ones include G-banding, which provides a visual karyotype that can easily confirm the presence of ring chromosomes. FISH is also extremely important for detecting ring chromosomes and understanding the portions at each end of the ring chromosomes that are deleted when the ring chromosome is formed through fusion. The image on the left shows a FISH probe hybridized to chromosome 3 close up and at normal size. And the image to the right is one of our ICGS case studies regarding the presence of ring chromosome 7 in a patient with TL.
Okay, and just very quickly, some future directions for the research of ring chromosomes is that since ring chromosomes are very rare, there are often not many reported cases of ring chromosomes. So continued categorization of ring chromosome cases is important to understanding and delineating specific treatments for ring chromosome patients. And also studying long-term effects of patients with ring chromosomes would also further elucidate what the symptoms are later in the life course of the patient.
Thanks everyone for listening. You can accept questions either live or through the chat.
Gracias, Justin y Alair. Bueno, la siguiente expositora es Milagros Pérez, estudiante de la Facultad de Ciencias Biológicas de la Universidad Nacional Pedro Ruiz Gallo, quien nos expondrá acerca de poliposis adenomatosa familiar. Si hay alguna pregunta, la pueden escribir por el chat.
Buenas tardes con todos. Como ya mencionó, mi nombre es Milagros Pérez. El día de hoy voy a hablarle acerca de la enfermedad.
Sube el volumen, por favor, no se escucha.
¿No me escuchan? ¿Ahora sí me escuchan?
Sí, pero muy bajito, muy bajito.
Para hacer un poco más alto. Bien, buenas tardes con todos. El día de hoy voy a hablar acerca de la enfermedad, que afortunadamente es poco frecuente, pero es de suma importancia. Esta es denominada la poliposis adenomatosa familiar. Bueno, esta es una enfermedad autosómica dominante que se va a caracterizar importante lo que es en el desarrollo de cientos a miles de adenomas, que son como unos pólipos en el recto y en el colon. Y también además de ello se puede encontrar en cualquier sector de lo que es el tubo digestivo. Ahora, mayormente se diagnostica, esto es diagnosticado durante la segunda década de la vida. ¿Y esto es por qué? Porque la mayoría de los pacientes van a permanecer asintomáticos durante los primeros años. ¿Por qué? Porque conforme va pasando el tiempo, los adenomas van a crecer y esos van a ser más numerosos, ¿no? Lo que va a causar sangrado rectal e incluso también va a poder desarrollar lo que es el cáncer. Ahora, casi todos los pacientes van a desarrollar lo que es el cáncer colorectal. Y algo muy importante de esta enfermedad es que es causada por una mutación germinal del gen APC. Ahora, con respecto a la epidemiología, el cáncer colorectal está cocinado por casos esporádicos de 70 a 75%. En el 15% es por historia personal o familiar de pólipos o cáncer colorectal. Entre el 5 y 10% es el cáncer colorectal hereditario no asociado a poliposis. Y entre el 1% tenemos lo que es la enfermedad inflamatoria intestinal. Ahora es aquí donde tenemos esta enfermedad que es menos de 1% la poliposis adenomatosa familiar. Bien, ahora para poder entender mejor esta enfermedad, tenemos que recordar quién es el que produce. Esta es el gen APC. Este es muy importante, ¿no? En situaciones normales como en patológicas. En condiciones normales va a favorecer el desarrollo y lo que es la homeostasis, pero en lo que es las condiciones patológicas va a causar lo que es la carcinogénesis. Ahora, si nosotros nos vamos aquí en el modelo de lo que es el cáncer colorectal, y se sabe que el cáncer de colon derecho e izquierdo tienen diferentes molecularmente hablando, y, y por eso el pronóstico. Ahora, el cáncer de colon derecho se debe a mutaciones, tenemos al gen, por ejemplo, KRAS, BRAF, y también por metilaciones. Mientras que en el colon izquierdo, aquí es donde se va a ver el modelo de lo que es la carcinogénesis, y es aquí donde se va a encontrar esta enfermedad. Ahora, también dice que en el 60% de lo que es los casos de mutaciones del gen C en lo que son las células somáticas son las que nos van a dar el modelo típico de lo que es la carcinogénesis, que han sido diagnosticadas en por lo menos hacia los 60 años. Pero si las mutaciones van a ocurrir en células germinales, es ahí donde se va a desarrollar la enfermedad, que es poliposis adenomatosa familiar. Bien, ahora hablaremos del gen. Está el gen APC. Este está ubicado en el brazo largo del cromosoma 5 con las bandas 2,1. Tenemos. Y aquí nos dice que hay una región codificante que se van a dividir en 15 exones y va a codificar una proteína grande. Esa proteína es la proteína APC, que además esta tiene múltiples dominios que contienen sitios de unión para numerosas proteínas, ¿no? Incluidos los microtúbulos, tenemos también a los componentes de la vía WNT, la beta es la beta-catenina y axina, y los reguladores citoesqueléticos como por ejemplo EB1, tenemos a EB1 y también a IQGAP1. Ahora, este gen es pequeño, como se puede observar, pero tenemos que identificar en esta imagen que las líneas rojas, estas son, se refiere a la activación, y las celestes son de inhibición. Ahora podemos entender que en condiciones normales, el gen APC favorece desde el citoesqueleto de la célula la estabilización de lo que son los microtúbulos, a pegarse a lo que es la beta-catenina, que va a regular a beta-catenina. ¿Y la beta-catenina qué hace? Bueno, normalmente cuando ya está regulada hacia abajo, va a hacer que haya un control de lo que es la proliferación y la diferenciación celular. Y eso es en condiciones normales. Vuelve a repetir, pero cuando el gen está mutado, este gen produce una proteína trunca, ¿no? Y entonces esta proteína trunca ya no va a poder trabajar, ¿no? No va a poder trabajar de forma adecuada y no va a poder, no va a poder inhibir lo que es la beta-catenina. Por lo que las
3
concentraciones de
minutos.
esta beta-catenina se van a alta, entonces va a hacer que es una sobreactivación en lo que es la proliferación, incluso una inhibición también de lo que es la diferenciación. Y también va a ser una inestabilidad cromosómica, y también puede ser que una estimulación en lo que es la asimilación celular. Es por eso la importancia de que este gen, si se encuentra mutado, va a ocasionar lo que es la carcinogénesis. Bien, ahora hablaremos respecto a las manifestaciones clínicas. Va a caracterizar por la presencia de cientos o miles de adenomas. Acá podemos ver que son como unos pólipos, colorectales, que van a estar en diferentes tamaños. Y además se pueden desarrollar una variedad de manifestaciones gastrointestinales extracolónicas. También en el estómago se dice que los pólipos de la glándula púbica se van a desarrollar en un 90% de los pacientes que presentan esta enfermedad. También van a existir pólipos adenomatosos en duodeno e intestino delgado. Ahora, con respecto a lo que es el diagnóstico, para el médico la identificación desde las manifestaciones de lo que son las manifestaciones extracolónicas, pueden llevar, por ejemplo, a realizar un examen endoscópico de lo que es el intestino grueso. Esto debe hacerse inicialmente tomando, por ejemplo, una historia familiar, ¿no? Una investigación genética del paciente y que tengan también sus familiares de primer grado. Ahora, eso debe iniciarse de tomar una historia familiar y realizando también una sigmoidoscopía o una colonoscopía completa. Dicho esto, también va a haber una secuencia directa de lo que es el gen APC. Respecto al tratamiento, se dice que la detección precoz de esta enfermedad es imprescindible para una intervención eficaz, es decir, para que sea posible poder prevenir lo que es el cáncer colorectal. Ahora, una vez que los pólipos ya se han desarrollado, el tratamiento definitivo consiste en lo que es la colectomía, y total también con la anastomosis ilional. Entonces, se va a recomendar que los pacientes con riesgo con esta enfermedad se sometan a una colonoscopía o una vigilancia endoscópica de lo que es el resto del aparato digestivo. Para poder concretar esta prevención es aconsejable también que los familiares se puedan realizar unas pruebas, ¿no? Unas pruebas moleculares para determinar su riesgo. Ahora, también para el tratamiento hay unos medicamentos antiinflamatorios, lo que son no esteroideos, que pueden inhibir la formación de nuevos pólipos. O sea, si aparecen nuevos pólipos son demasiado rápidos o la demasiada cantidad para poder resecarlos, se va a requerir una extirpación del recto o la ostomía permanente. Esas son las referencias y muchas gracias. Ha sido todo y cualquier pregunta la hacen al chat.
Gracias, Milagros. Ahora nos expondrá Kathleen Maloney, bióloga molecular del Hospital Baylor Scott and White Health System, que nos expondrá acerca de microarreglos cromosómicos. Si hay alguna duda, la pueden escribir por el chat de la plataforma.
Don't hear anything. We don't hear anything, Kathleen. It's still mute.
Okay, can you hear me now?
Yes.
Great. Okay, sorry about that. Okay. Just give me 1 second to share my screen. Okay. So, like I said, my name is Caitlin Maloney. I am a molecular technologist with Baylor Scott and White Medical Center in Texas. And today I'm going to be talking about chromosomal microarrays. So microarray technology starts with isolated nucleic acid that is tagged with a fluorescent dye or biotin. This image is showing a sample starting with RNA, but you can also start with DNA. It just depends on what your array is looking for. Those samples are going to be added into microarray chips. They're also called slides or wafers. And this is going to allow for the simultaneous detection of thousands of genes. Those chips are going to be scanned, and then digital files are going to be created reflecting the probe intensities. And then those files are going to be converted into interpretable data analysis files in order to visualize any abnormalities. So the principle of microarrays is comparative genome hybridization, also called CGH, which is the complementary hybridization between probe and target DNA. So in the image, you have your solid surface, which has thousands of DNA probes fixed to the surface. Those probes are specific sequences, and they are designed to allow complementary hybridization with your target sample. So the probes can represent a single gene, but you can also have probe sets in order to represent the multiple transcripts for a gene. So after the hybridization step, the chips are going to be washed, and any of the unbound sample is going to be washed away, and then fluorescence is generated and creates a signal intensity when those arrays are scanned. This is just another picture to show what an actual array looks like and how that little surface actually has millions of probes showing the hybridization, and then this is that probe intensity file. And so a comparison of karyotyping fish and CGH, just to show when you would use each and then what the pros and cons are. So for arrays, you have whole genome coverage similar to karyotyping. You have a higher resolution than fish or karyotyping, which is one of the reasons why CMAs are popular. They also pick up submicroscopic copy number changes like microdeletions and microduplications. However, arrays cannot detect balanced rearrangements or point mutations. So arrays might not be useful in some situations. There are lots of applications for microarrays. One of the most common ones is gene expression and epigenetics. They're also used in genome-wide association studies, pharmacogenomics, and then for the point of today's lecture, cytogenetic copy number analysis. And CMA or cytogenetic copy number analysis and chromosomal microarrays are the first line of testing for patients with congenital anomalies and neurodevelopmental disorders. So there are a lot of synonyms for arrays, chromosomal microarrays, but when we refer to CMA, we're talking about chromosomal microarray analysis, which measures gains and losses of DNA throughout the genome, including aneuploidies, other structural changes, and submicroscopic anomalies. And there are three types of chromosomal microarrays. We have ACGH, SNP arrays, and then a combination of those. And the main takeaway from this table, and the difference between both of these arrays, are that ACGH arrays are useful for detecting net gains or losses in genetic material, while SNP arrays can detect copy neutral changes, regions of homozygosity, mosaicism, and actually determine the genotype at a specific locus, as opposed to ACGH, which is more broad copy number variation. So this is just another picture showing the two for array CGH. You're comparing your patient and your control DNA in a single sample to detect those copy number variations, again, microdeletions and microduplications. In this example, which is, I think this is one of the most common pictures that people see for microarrays, the yellow areas represent equal copies of patient DNA and control DNA. The red and green spots represent unequal copies of your patient DNA compared to your control. So red would be a loss of genetic material, and green is a gain of genetic material. On the other hand, you have SNP arrays, which compare your patient phenotype in silico to the reference set. So you would use computer simulation in order to detect single nucleotide changes, also called SNPs. And SNPs are common genetic variations in their genomes, and they're defined by having a frequency greater than 1%. So your probes on SNP arrays are known nucleotide sequences that have multiple alleles, and your patient DNA will bind to allele A. If they have allele A, it'll bind to allele B if they have allele B, or if they are heterozygous, the sample will bind to both allele A and allele B. And again, SNP arrays are useful for genotyping, as you see here, and as well as figuring out the breakpoints of copy numbers, areas of heterozygosity, etc. And so the A's and the B's is a way to simplify SNP genotypes because there are over 10 million SNPs. So the AA and BB just represent homozygous for that allele, AB is heterozygous, and then these would be 1 copy, 0 copies, 3 copies. So the microarray analysis software depends on what assay you're using. The case examples I have today, we'll be using this software right here, which is called CHAS, but there are other ways to visualize microarray data. So the first case presentation is a 7-month-old female with heart defects, brain hemorrhaging, kidney problems, multiple congenital anomalies. And you can kind of see this large, obvious, like large duplication and then a couple of small deletions. So our analysis software gives us a karyotype. All of the red areas are deletions. The purple blocks are losses of heterozygosity, and then the blue regions are going to denote gains or duplications. So this is what our segment table looks like. We can see where our copy segment starts and ends, the size of it, what OMIM genes are present in that region, whether or not there are FISH probes for that region. There's a space for us to write our interpretation, and it also gives us the nomenclature for that copy number segment. So down here in this detailed view, we can see this copy number variation. They're all separate segments, but we have a gene, like an editing tool, where we can combine these copy number segments and it'll give us like the final size and gene content. And so we have a lot of different tracks to show us different things about this segment. So again, the blue represents duplications. It'll tell us how many copy numbers are present. If I was to hover over any of these tracks, I could see that this segment probably says 3. This segment will say 2, because this is what a normal segment looks like. And then this over here would say 1. The weighted log-2 ratio track is it just shows the relative probe intensities to our reference file. We use this smooth signal track in order to detect non-integer copy number variations that you would see in mosaicism. And then down here, the allele difference is actually a visualization of the SNP alleles. So again, we use a combination ACGH and SNP array. So there's a pattern for each copy number state. And I like this example because you can see that this is what a normal allele track looks like when you have 2 copies. This is what a gain would look like, and then you can kind of see at the end, this is what your allele tracks would look like for a loss. And so they can make these tracks because all of the probes are tightly squeezed together to form this hamburger track, where your top line is your homozygous AA alleles, your middle track is your heterozygous AB alleles, and your bottom track is your homozygous BB alleles. So if we scroll down a little bit, we can also see any of the patient data files that we have imported into the software. So we have about 8 years of copy number data saved into our system so that whenever we see new copy variations, we can compare them to what we've already seen in the region, or we can add new ones for future reference. And we color code them based on what they are. We just keep them green if they're likely benign, commonly seen, red for deletions, blue for duplications. So we also have a few more tracks here at the bottom. The chat software comes with the database of genomic variants, which is an archive of genetic variation. It also pulls in the OMIM genes, and OMIM is a catalog of genes and genetic diseases. And then we also have this pink track at the bottom for the same genes. You can just see how many transcripts there are, the gene length, what the intron and exon content is for that gene, and then just a side band at the bottom for reference. And so this first case was a partial trisomy 13 and a 13q34 microdeletion. It is pathogenic due to the size and the gene content. There were 86 genes in the duplication and 16 genes in the deletion. And some of the copy number variations are not as obvious, pathogenic or likely pathogenic. And so a little bit later, I'll show you the calculator and the scoring system that we use in order to arrive at that classification. So this case happened to have chromosome analysis that confirmed what we saw by arrays. Not all cases have chromosome analysis, but it is kind of nice to have that comparison. And the karyotype also found that there was a duplication and a microdeletion on chromosome 13. The second case presentation is a 1-year-old female with global developmental delay, low muscle tone, gait issues. It's not as obvious to see what would be abnormal here, but as I'll show you, chromosome 13 and chromosome 14 have some interesting variants that we actually reported out as variants of uncertain significance. And so this the first one was the duplication on chromosome 13 was 1 megabase pair, and it only included one gene, but that gene also wasn't associated with a Mendelian disease, which makes it a little bit trickier. So there are sparse probes. You can't really tell that there are 4 tracks for our duplication, but you can kind of see this bubble right here that this would confirm with a copy number segment duplication. So a gain of that size has also not been previously seen in our patient population. We had seen stuff in copy number variations in the same area, but nothing that was 1 megabase pair large. We also see, we have a track for candidate autism spectrum genes. And so this gene, there is some evidence that it might be a candidate for autism. And then on the very bottom, we have 2 transcripts, and it's showing the exons, which are the little tick marks, and the introns are just in between. And so you'll have multiple transcripts for a single gene due to alternative splicing, and that arrow represents the reading direction. So this gene happens to be transcribed on the reverse strand. I'm going to go fast just because the last case I think is the most interesting. This second case, we also found a region of a loss of heterozygosity, or what's called an LOH. And so you can kind of see these purple blocks are losses of heterozygosity that we'd called in the past. However, these areas are reported as variants of uncertain significance if there are autosomal recessive genes that might lie in the region that might fit the patient's phenotype. And so it's not diagnostic for a specific condition, it just raises the possibility of a recessive disorder. So the patient would have to have a mutation in an autosomal recessive gene that was lying in this area. And for this patient, there were multiple genes that were kind of spot on with the patient's phenotype. Last case presentation is a 10-year-old male with a history of Fragile X, consanguinity, ID, and developmental delay. And so you can see that those losses of heterozygosity, there are a ton of them in this patient's genome. And so there are actually 15 chromosomes with LOHs that were reported, but we're excluding the X chromosome. This is actually normal for a male to have two large blocks of heterozygosity because males only have one X chromosome, so they're going to be homozygous for that entire chromosome. And so this patient also underwent whole exome sequencing at another laboratory that confirmed a close degree of relationship or genetic relatedness with a common ancestor. It was actually calculated to be between 10 and 18% similar. And so whenever we see these long continuous stretches of homozygosity, it usually means that the patient has inherited identical genetic segments from related parents. So what's interesting about losses of heterozygosity is that we're going to see that it's a copy neutral event. So we're going to really have to look at the allele difference. And so just to kind of remind everyone what a normal allele difference looks like, this is the patient's 4th chromosome. I have the cytoband at the bottom for reference. And so we want to see 3 tracks to know that that area is normal. But when we start to look closer at these losses of heterozygosity, we see this huge gap in the middle. And so there are some areas of heterogeneity, as you can see the probes throughout this region, but it doesn't create a nice line like it does over here.
Sorry for the interruption. You have 5 minutes remaining.
It's perfect. Okay. And so your log2 ratio, your smooth signal, your copy number is all the same, so that doesn't really help you as much as your allele difference does. I think I just had 2 more chromosomes just to show what the normal area looked like and then what these losses of heterozygosity. You might hear AOH for areas of homozygosity or LOH for losses of heterozygosity. It gets kind of confusing. But to kind of wrap up, we use the ACMG ClinGen CNV calculator in order to determine, we come to a final classification based on a scoring system. And so this was developed in order to address the inconsistency with scoring CNVs. A lot of laboratories, they follow their own internal procedures. There's conflicting evidence for CNVs, and they just created these technical standards in order to alleviate that. So there's a calculator for gains, and then there's a calculator for losses. So if I come across a questionable CNV, I'll go through this calculator as well as Dr. Tirado. We kind of work together to figure out what the final classification is. And that just depends on whether or not the gene is a protein coding gene, does it overlap with haplosufficient regions or benign regions, how many genes are actually in that segment, is there literature or evidence in public databases or your internal system to support that copy number variation, and then do you have any inheritance information for the patient? Can you confirm if that CNV is de novo or if it segregates with the family? So after using that system, we arrive to one of these 5 classifications. And so I just want to draw attention to the variant of significance because that's a huge range. And a lot of variants are going to fall in between there because there's just insufficient evidence at the time of reporting to determine whether or not it's clinically significant. I think I'll finish up real quick. Anyone is welcome to take pictures of the slides for some videos, papers that I felt like explained arrays really well, and then a blog post at the end just to talk about chromosomal microarrays and then comparing them to other things. And I am also speaking at the talk in April. I'll have more case reports. We had a possible ring chromosome, so it was interesting to listen to the last presentation to kind of learn about that. And then we also have an interesting case of uniparental disomy. So that's my email, and thank you guys so much.
Excellent presentation, Kathleen. Congratulations.
So there are no more questions for Caitlin. Again, thank you again for the presentation. Our next presenters will be Emily Pang and Monsi Solanki. They are students of the University of California and members of the International Circle of Genetics Study, Los Angeles chapter. They will be presenting on Marfan syndrome. Once again, if there are any questions for the exhibitors, please write them in the platform chat at the end of the presentation.
All right, thank you, Andrew. I will share my screen. Okay. Can everyone see that? Yes, okay. So hello, everyone. Like Andrew said, my name is Emily, and I'm an undergrad at UCLA.
And I am Monsi. I'm also an undergrad at UCLA.
Yeah, and we're part of the Los Angeles chapter of the International Circle of Genetic Studies, and we will be presenting on Marfan syndrome.
So first, we wanted to give an overview of Marfan syndrome. So Marfan syndrome is a heritable systemic connective tissue disease that has a major impact on musculoskeletal, cardiovascular, and ocular health. So Marfan syndrome is an autosomal dominant genetic disorder, and it occurs every 1 case in around 3,000 to 5,000 cases. And it is known to be caused by a mutation in the FBN1 gene that encodes for fibrillin-1, which is a protein involved in connective tissues. This mutation in FBN1 has shown to cause an increase in the transforming growth factor beta, or TGF-beta for short, which often leads to issues with elastin and connective tissues overall. Because connective tissue is present throughout the body, Marfan syndrome affects many different parts of the body, and conditions of Marfan syndrome range from a few symptoms or no symptoms to serious and life-threatening complications. This largely depends on the location in the body that is affected and the severity of the condition itself.
Okay, so moving on to screening and diagnosis. So for diagnosing Marfan syndrome, we use the revised Gantt diagnostics methodology, and it's basically a criteria of major and minor clinical manifestations plus considering family history. So some examples of major diagnostic criteria include an enlarged aorta or a tear in the aorta, lens dislocation, family history, or at least 4 skeletal problems such as flat feet, curved spine, or a dural ectasia, which is the enlargement of the lining that surrounds part of the spinal cord. And some examples of minor diagnostic criteria are myopia, which you may be familiar with. It's short-sightedness, unexplained stretch marks, or loose joints. So as you can see, like the minor criteria, the symptoms are a lot more common than the major ones. So that's why they use like a combination of both to diagnose Marfan syndrome. So as you can see, the diagnosis remains essentially clinical, but as we mentioned before, FBN1 mutation is associated with Marfan syndrome. So FBN1 mutation screening analysis is also important to confirm the clinical diagnosis. And we will go into FBN1 mutation in more detail later. And this picture, as you can see, shows just a few of the many criteria used for diagnosis.
So now we'll be going over clinical features. So traits of Marfan syndrome include long limbs, long face, long fingers and toes, hypomusculature, and chest, spine, hip, and foot deformities. So as stated before, Marfan syndrome affects different parts of the body, usually where fibrillin is present. So for the cardiovascular system, it causes mitral valve prolapse and regurgitation, left ventricle dilatation and cardiac failure, and pulmonary artery dilatation as well. So along with these health issues, heart issues, aortic root dilatation is the most common cause of mortality among patients with Marfan syndrome. For the musculoskeletal system, there are often symptoms such as scoliosis, back pain, and that is often caused by dural ectasia, as stated before, and joint hypermobility. So the ocular system is also affected since connective tissue is also present there. And some typical complications include bilateral ectopia lentis, myopia or short-sightedness, and retinal detachment.
All right, so moving on to the genetics, like we mentioned before, Marfan syndrome is inherited in an autosomal dominant manner, but there have been some cases of recessive mutations, especially in sporadic cases, so we still have to keep that in mind, even though autosomal dominant is the main way it's inherited. So Marfan syndrome is associated with mutations in the FBN1 gene, which encodes for fibrillin 1, and it is found on the on chromosome 15 on Q21.1, as you can see in the diagram on the screen. It's 65 exons long, and it's found in 90% of Marfan patients. So it's found in most Marfan syndrome cases, but not all. So it's only associated with it. So more information about the fibrillin 1 protein. It's a glycoprotein found in the extracellular matrix that contributes to the final structure of microfibrils. So microfibrils give elasticity to the arterial wall and facilitates the artery to retain its initial form after expansion. So that's why the FBN1 mutation and Marfan syndrome in general has a lot of cardiovascular defects associated with it. And microfibrils also connect the ciliary body with the lens of the eye. So that also explains the ocular defects as well. And additionally, fibrillin 1 is also involved in the activation of transforming growth factor beta, which regulates cell proliferation. So fibrillin 1 normally inhibits the activation and release of TGF beta, but structural deficits in microfibrils are associated with increased activation of TGF beta, which causes deleterious effects on smooth muscle development and extracellular matrix integrity. And there are two main types of FBN1 mutation. There's haploinsufficiency and dominant negative. And mutations leading to haploinsufficiency have production of mainly the wild type allele, resulting in less normal fibrillin 1 protein. And then for mutations leading to the dominant negative, there's an interaction between the wild type and mutated fibrillin 1 protein, leading to the disturbed folding or function of fibrillin 1. And the main thing to note is that there's a varied phenotypic expression. So more than 3,000 mutations in this gene have been published, and almost all are unique to an affected individual or family. So due to this large number of unique mutations and also clinical heterogeneity in individuals with the same mutation, genotype to phenotype correlations in Marfan syndrome are difficult to identify.
So while there is no cure for Marfan syndrome, there are possible treatments and therapies that can help with managing the symptoms and complications that come with the disorder. So, for example, cardiovascular treatments include beta blockers that are used to slow down aortic root enlargement that could lead to aortic aneurysms. Recently, there have also been more studies using Marfan mouse models that focus on the use of angiotensin receptor blockers to decrease overall TGF beta signaling as well. And this could also slow down the process of aortic root enlargement. So even with these two blockers, it's also possible that with the current treatments to slow down heart damage, there is often still a need for aortic repair surgery later in life. So that is also an option as well. For the musculoskeletal treatments, there is scoliosis bracing and surgeries as well in order to help realign the spine. And there's also breastbone correction surgery that can be done to alter the appearance of the chest area, often because there is a convex or concave appearance to the chest in individuals with Marfan syndrome. Lastly, for ocular treatments to manage Marfan syndrome, there are glasses and contact lenses as the usual procedure for myopia, and there's also cataract surgery in the cases where vision has been affected over time. And glaucoma prevention measures are also taken in patients with Marfan syndrome with treatments such as eye drops and laser treatments in order to keep vision intact over time.
So I'm going to go a little bit more in detail about the current treatments and therapies that were briefly mentioned in the previous slide. So currently, beta blockers are used the standard medical therapy for Marfan syndrome. And like we mentioned before, it's to prevent and delay progression of aortic dilation and aortic dissection. And this mechanism is based on the reduction of tension on the proximal aorta. So this relief of stress on the aortic wall can lead to decreased mean blood pressure and improvement of aorta stability by elastin and collagen cross-linking. And then after aortic root surgery, beta blockers are still continued to prevent complications distal to the graft. But however, the effectiveness of continuing beta blockers still needs to be studied, and further research is needed on this area. And then the second type is losartan, and it's an angiotensin II type 1 receptor antagonist, and this reduces arterial pressure and wall stress. And we
Just
mentioned
interrupt, just 5 minutes left.
Yeah. All right, thank you, Andrew. So in a Marfan syndrome mouse model, this has been shown to decrease the release of TGF-beta, resulting in nearly completely inhibited aortic dilation and normalization of histological changes in the elastic fibers of the aortic wall. And then lastly, we're going to talk a little bit about the outcomes. So the life expectancy for Marfan syndrome, patients actually live over 60 years old, and this is because patients adhere quite well to medication and physical activity guidelines. So they would be asked to not engage in intense exercise and just like do low intensity workouts instead. And also like adhering to the medication prescribed by their physician. And actually cardiovascular manifestations represent the major morbidity and mortality factors. So this is actually the main clinical focus. So patients need to look out for aorta dilation, predisposition for aortic tear rupture, mitral valve prolapse, tricuspid valve prolapse, and enlargement of proximal pulmonary artery. So you need constant cardiovascular monitoring to look out for these symptoms. And to address the ocular defects, there's also regular ophthalmic evaluation. And additionally, for women, pregnancy can potentially be dangerous, especially if the aortic root exceeds 4 centimeters. So complications can include rapid progression of aortic root enlargement, or aortic dissection or rupture during pregnancy, delivery, or the postpartum period. And that is it for our presentation on Marfan syndrome. Thank you.
Thank you, Emily. Thank you, Monsi. Once again, if there are any questions, feel free to post them in the chat.
Please.
Okay, so if there are no questions, thank you again, Emily and Monsi. We'll be moving on to the next presentation. Up next, we have Bruno Garcia, who's a student at the Universidad Señor de Cipan, who will be talking about hereditary non-polyposis colon cancer. Once again, feel free to post any questions in the chat at the end of the presentation.
Buen día, por favor, voy a compartir pantalla. Me comunican si es que se logra visualizar bien y si es que no hay ningún problema al momento del audio.
¿Se logra apreciar? ¿No hay ninguna dificultad al momento de visualizar las diapositivas?
Perfecto, perfecto. Ponla en presentación.
Ok. Buen día con todos. El tema a desarrollar se denomina hereditaria no poliposis colon cáncer. ¿Estoy bien? Mi nombre es Bruno García Ruiz, integrante del Círculo Internacional de Estudios de Genética, capítulo Chiclayo. El cáncer colorectal hereditario no poliposo, o también denominado síndrome de Lynch, va a presentar un patrón de herencia autosómico dominante. ¿Está bien? Este síndrome tiene como característica que es un trastorno hereditario que va a aumentar las probabilidades de padecer cáncer. ¿Está bien? Donde el principal es a nivel colorectal. Ahora, hay ciertos tumores extracolónicos que se van a asociar al síndrome de Lynch a lo largo de toda la vida. ¿Está bien? El principal, como hemos detallado, es el cáncer colorectal, que tiene una mayor incidencia en pacientes con síndrome de Lynch, pero no es el único. También encontramos el endometrio, uréter, estómago, páncreas, ovario, etc. Ahora, cuando hablamos del síndrome de Lynch, hay un cierto número de genes asociados a esta patología. ¿Está bien? Diversos genes y en diversos cromosomas. Como podemos visualizar, en el cromosoma 1 vamos a encontrar, por ejemplo, al gen EXO, en el cromosoma 2 al gen MSH2, MSH6, en el cromosoma 3 al MLH1, en el cromosoma 5 al MSH3, cromosoma 7 PSM2, cromosoma 20 PSNA. Pero ¿cuál es la característica de todos ellos? Que van a estar o van a formar parte de un grupo de genes que se les conoce como los genes reparadores del ADN. ¿Está bien? Hay un gen que se llama el gen PSNA que no forma parte de ese grupo de los genes reparadores, pero va a inactivar a uno, va a inactivar al MSH2. Por lo tanto, va a estar presente o se contabiliza dentro de los genes asociados al síndrome de Lynch. ¿Cuál es lo que queremos rescatar acá? Estos genes, al momento de que se expresan, se van a unir para formar ciertos complejos, ¿vale? El que debemos recordar es el complejo MUT-S-alfa, que se encuentra acá, también el MUT-S-beta y también el MUT-L-alfa, ¿está bien? Estos complejos son muy importantes por las diversas características que veremos a continuación. Hay una prevalencia en el cáncer colorectal donde el 1 a 3% de todos los pacientes que presentan cáncer colorectal van a tener síndrome de Lynch. Cuando hablamos de cáncer al endometrio, del 0,8 al 1,4%. Pero ahora, ¿qué pasa con estos pacientes con síndrome de Lynch? Cerca del 90% van a tener mutaciones en el MLH1 o en el MSH2. ¿Está bien? 7 a 10% en el MSH6, 5% en el PSM2 y 1% en el gen EPCAM. Ahora, hemos estado hablando de los genes reparadores del ADN. Estos grupos de genes van a estar dentro de un sistema, ¿está bien? Que se le conoce sistema MMR o en español se le conoce como sistema de reparación del ADN. Este sistema es netamente post-replicativo, ¿está bien? ¿Y cuál va a ser su función? Va a reconocer inserciones pequeñas por bases o ciertas inserciones o deleciones un poco más grandes, ¿está bien? Cuando hay un fallo en este sistema a nivel germinal, la tasa de error técnicamente es mayor de 100 a 1000 veces para poder expresar un fenotipo mutador. Este sistema MMR lo que hará es corregir errores en microsatélites. Está bien que serán introducidos obviamente por la ADN polimerasa. Como bien sabemos, hay cerca de medio millón de microsatélites en todo nuestro ADN, pero estos microsatélites no son secuenciales, sino están colocados de manera aleatoria. Por lo tanto, van a estar presentes también en regiones codificantes que van a servir para formar una proteína. Entonces, si estamos hablando que estos microsatélites van a presentar algún tipo de error, al momento de generar lo que es su transcripción, obviamente enviarán un mensaje o un código no establecido. Por lo tanto, al momento de la traducción generarán una proteína o no funcional o una proteína trunca. ¿Está bien? Ahora, como hemos detallado que son regiones codificantes de manera aleatoria, porque los microsatélites se encuentran de manera aleatoria, van a intervenir esas proteínas no funcionales o truncas en diversos procesos celulares, como es el caso de la apoptosis, preparación del ADN, regulación post-transcripcional. Entonces, cuando hablamos de una inestabilidad de microsatélites intragénica,
nos hemos podido dar cuenta que cerca del 80% con MMR deficientes van a tener alteraciones en el gen TGF-beta2 y también el otro 20% en el IGFR2. ¿Está bien? Y también ciertas mutaciones en los promotores de apoptosis VAX. Ahora, esta vía molecular del sistema, ¿qué pasa? Va a actuar por pasos, ¿está bien? Como hemos detallado en un inicio, los complejos que se forman, por ejemplo el MUT-S-alfa, que está formado por la unión de las proteínas del MSH2 y el MSH6, van a reconocer errores en la nueva secuencia del ADN menor a 10 bases. ¿Está bien? En cambio, el MUT-S-beta, formado por la proteína del MSH2 y el MSH3, va a reconocer errores mayor a 10 bases. Por lo tanto, el MUT-S-alfa va a reconocer pequeños errores o pequeñas deleciones o inserciones, y el MUT-S-beta va a reconocer un poco más grandes, ¿está bien? Mayor a 10 bases. Entonces, los complejos MUTs lo que harán técnicamente es reconocer el error en la cadena del ADN. ¿Está bien? Primero se va a reconocer el error en la cadena del ADN. Luego vamos a tener lo que es el MUT-L-alfa, que va a estar formado por la proteína del MLH1 y el PMS2, que lo que hará es reclutar enzimas de reparación del ADN. ¿Está bien? Y también aumentará la eficiencia de los complejos MUTs. ¿Está bien? Aumentará la eficiencia para poder reconocer los errores del ADN. Cuando ya, bueno, después, una vez de que ya se reconoció el error y ya se han reclutado las enzimas necesarias para poder reparar este ADN, empezará lo que es el corte de la secuencia incorrecta, ¿vale? Bien puede ser por interacción del MUT-S-alfa, va a intervenir el MUT-S-alfa si es menor a 10 bases, y si es mayor a 10,
5
va a
minutos.
intervenir el MUT-S-beta. Van a interactuar con la proteína PSNA, que va a ayudar a discriminar lo que es las cadenas del ADN. Y a su vez va a interactuar con la enzima EXO1, ¿no? Que es una exonucleasa, que ayudará a la escisión y a la resíntesis del ADN. Acá podemos visualizar, por ejemplo, el complejo MUT-S-alfa, que va a reconocer un error de bases. Acá tenemos, por ejemplo, también el MUT-S-alfa reconociendo errores pequeños, pero en un error grande ya no es el MUT-S-alfa, ahora es el MUT-S-beta. ¿Está bien? Y tanto como el alfa como el beta va a interactuar con el MUT-L alfa para poder discriminar las cadenas, determinar el corte y la resíntesis de la nueva hebra del ADN, ¿no? Ahora, hay una correlación genotipo-fenotipo, según la variante patogénica del sistema MMR. Por ejemplo, si hay una mutación en el MLH1, va a ser más frecuente en pacientes con cáncer a nivel colorectal. ¿Está bien? El MSH2 en neoplasias extracolónicas, uréter, estómago, páncreas, como hemos determinado. El MSH6 determina lo que es cáncer al endometrio, pero en una presentación tardía. El PMS2 va a tener una presentación tardía, pero va a tener un menor riesgo de padecer cáncer, ¿no? Entre todas las variantes patogénicas, esta se podría decir que es la de mejor pronóstico. El EpCAM, por ejemplo, va a desactivar a lo que es el MSH2. Por lo tanto, si desactiva este gen, va a tener un mayor riesgo de sufrir neoplasias extracolónicas y va a depender del tamaño de la lesión. Acá, por ejemplo, podemos visualizar lo que es un riesgo, una población general en cáncer de colon, por ejemplo, y acá en una variante patogénica en síndrome de Lynch con alteración en el MLH1. En una persona normal, 4,5%, y en una variante patogénica del MLH1 puede llegar incluso hasta un 80%. Y puede presentarse antes de los 50 años, ¿no? O hasta los 60. Por ejemplo, en el MSH6 va a estar predominando lo que es cáncer al endometrio. Y el que se determinó como mejor pronóstico, ¿no? El PMS2, en algunos no hay reportes o presenta un riesgo menor que otras variantes. Hay ciertos criterios que nos ayudarán a establecer un diagnóstico, como es los criterios de Ámsterdam. Ámsterdam 1, Ámsterdam 2. ¿Por qué se dejó de utilizar el Ámsterdam 1? Porque no englobaba a las neoplasias extracolónicas, no las englobaba. Por lo tanto, ya en el Ámsterdam 2 ya se englobaban. ¿Y cuáles son los criterios? Para poder determinar que una persona presenta lo que es síndrome de Lynch, debe presentar los 4 criterios. Debe tener los 4 criterios. 3 o más familiares con cáncer asociado a síndrome de Lynch, 2 generaciones afectadas, cáncer detectado antes de los 50 años, y en lo que es cáncer colorectal debe excluirse el diagnóstico de poliposis adenomatosa familiar. Ahora, La asesoría genética en el síndrome de Lynch, como bien sabemos, es una penetrancia incompleta. Cerca del 50% de la descendencia puede presentar este síndrome. Lo que se hace es referir al área de asesoría genética para poder determinar la evaluación del riesgo de cáncer, ¿no? Determinar si es un cáncer de tipo esporádico, si es hereditario, ¿está bien? Y también un cierto descarte con otros síndromes, como es el síndrome de Cowden o politosis colónica familiar. Lo que se hace son revisar los antecedentes personales y familiares a nivel oncológico a través de un árbol genealógico. Se utilizan ciertos criterios clínicos para poder establecer mejor un diagnóstico, ciertos estudios genéticos confirmatorios, y después de ya recopilar toda esa información, explicar los resultados, las causas y las implicancias tanto en el paciente como en los familiares. ¿Está bien? También se recomiendan colonoscopías a partir de los 25 años en familiares y pacientes que pueden tener una predisposición a esta enfermedad, cierto control ginecológico y en ciertos casos aspirado endometrial y ciertos marcadores tumorales como lo que es el CA-125. Muchas gracias. Si hay alguna pregunta o algo relacionado acerca del tema del síndrome de Lynch, me pueden comunicar sin ningún problema. Voy a detener
Gracias, Bruno.
compartir.
Antes de continuar con las exposiciones, queremos hacer la invitación al curso que estamos organizando, que es un curso de citogenómica que va a ser del día 26 al 30 de abril del presente año, de 2 a 7 de la noche, con un valor académico de 3 créditos, en total de 25 horas. El costo para los estudiantes es de 300 y para profesionales es de 500. Cualquier tipo de información que deseen, pueden escribirnos mediante el correo y con gusto vamos a responderles.
¿Puedo compartir ahorita? Rodrigo, ¿puedo compartir?
Sí, si me deja presentarle, doctor.
Ya.
A continuación va a exponer el PhD Carlos Tirado, quien es profesional investigador de Baylor Scott.
¡Pendejo! ¡Me robó mi línea!
Por favor, sus micros apagados. El doctor Tirado es el asesor principal del Círculo Internacional de Estudios de Genética y nos expondrá acerca de plasma cell myeloma. Si hay alguna pregunta, la pueden escribir mediante la plataforma virtual. Gracias, doctor.
Buenas tardes. Vamos a ver cómo sale mi español, ¿ok? Vamos a hablar un poquito sobre plasma cell myeloma, que es el nuevo nombre que se le da al mieloma. Y vamos a hablar un poquito de la citogenómica, un un update en citogenómica, ¿ok? En primer lugar, que no tengo ninguna relación con lo que estamos, voy a presentar, así es de que, por favor, no hay ningún conflicto de interés. El plasma cell myeloma es un desorden neoplásico de las células plasmáticas que más o menos abarca un 1.8% de todas las malignidades en Estados Unidos y el 18% de las malignidades hematológicas y es el 2% de las causas de cáncer. Se caracteriza por una gran heterogeneidad en la parte clínica, un espectro grande de anomalías genéticas y los tratamientos además, ¿no? Entonces se ha podido ver que ahora el FISH o el Fluorescence in Situ Hybridization tiene una sensibilidad muy grande en el pronóstico de esta enfermedad neoplásica, ¿ok? Como sabemos, es una enfermedad de las células linfáticas, ¿ok? De los linfocitos B, y lo cual es interesante porque es un desorden que abarca un quinto de las malignidades hematológicas y del 2% de las causas de cáncer en los Estados Unidos, ¿no? Como dije anteriormente, el problema está en las células plasmáticas. Tenemos varios desórdenes de células plasmáticas. Algunas son malignas y otras son no malignas. Tenemos, por ejemplo, la MGUS, que es el Monoclonal Gammopathy of Undetermined Significance, que no es un cáncer, pero que puede ser convertirse en cáncer. Luego tenemos el linfoma linfoblasmacítico, el plasmacytoma y luego el plasma cell myeloma, ¿ok? La monoclonal gammopathy of undetermined significa que es el MGUS, el MGUS, como le llamamos en el mundo latino. Tiene menos del 10% de la médula ósea está con células anormales, de células plasmáticas anormales, no es cáncer. Pero lo interesante es de que estas células pueden hacer una proteína M, ¿okay? Que a veces se encuentra en la sangre o en la orina. Y en muchos pacientes la cantidad de proteína se mantiene el mismo. No es cáncer, ¿ok? Pero empiezan a hacer síntomas y problemas de salud, ¿ok? En algunos pacientes luego se puede convertir en una amiloidosis y puede causar un problema en el riñón, en el corazón y en los nervios, ¿ok? Pero sin embargo puede convertirse en cáncer, ¿ok? O en un linfoma linfoplasmacítico o en un chronic lymphocytic leukemia, ¿ok? El plasmacytoma, ahora ya tenemos que las células plasmáticas están pueden estar en un lugar y formar un tumor y entonces se llama plasmacytoma. Entonces lo podemos encontrar como un plasmacytoma aislado del hueso, ¿ok? O también podemos encontrarlo como un plasmacytoma extramedulario, ¿ok? Y que a veces se encuentra en las garganta, en las amígdalas y en en los sinoides paranasales, ¿ok? También tenemos el plasmacytoma solitario, que es como el mieloma múltiple, pero causa un crecimiento de las células plasmáticas y puede pasar fuera del hueso también, ¿ok? Y puede terminar en un caso de cáncer neoplásico. Los síntomas los tenemos que tienen una fragilidad en los huesos, que es tienen un dolor en los huesos, una fatiga grande, rupturas en los huesos, problemas en los riñones, una hipercalcemia, lesiones de la piel y también infecciones, ¿ok? Entonces, tenemos que medir, el clinician va midiendo la proteína M. Si está menos de 30, dicen ellos que es un MGUS. Si está mayor de 30, puede ser un smoldering multiple myeloma. Y luego ya tenemos mayor de 30 está en el plasma cell myeloma, ¿ok? Entonces lo encontramos que no, que está en el hueso, ya es una neoplasia grande, ¿ok? Entonces podemos ver acá también que el hit empieza en el MCAS, es un hit, los hits genéticos que tenemos, ¿no? Porque sabemos que es todo una cadena de varios hits. Luego seguimos con smoldering y luego seguimos con el mieloma, ¿ok? Se tienen que tocar todos los genes. Ahorita estoy hablando así rápido porque se tienen que tocar los genes del ciclo celular, las ciclinas y las tirosinas quinasas ciclina dependientes, ¿ok? Que se involucran en todo el ciclo celular, ¿ok? Y lo más interesante además de que ya comenzamos, cuando ya estamos comenzando con los problemas del IgH, de la cadena pesada de inmunoglobulina, ya es que estamos haciendo ya la gravedad del asunto, ¿ok? Entonces, el plasma cell myeloma se encuentra más o menos que podemos encontrar unos cariotipos anormales en 30-50% de los casos, ¿ok? Pero ojo, es bien difícil conseguir que podamos ver el clon anormal, ¿ok? Y luego les voy a decir por qué. Entonces, ¿qué es lo que pasa? Que estamos usando FISH ahora con el SMART, que es la clasificación del SMART 3.0, que es la que da la clínica Mayo para todos los estándares de plasma cell myeloma. Entonces, está poniendo FISH como una herramienta que es la que da el pronóstico, el diagnóstico, ¿no? De cuán grave es la seriedad del asunto, ¿ok? En mi laboratorio usamos B-cell mitogenes, usamos para poder, desde que nos dicen que es una amiloidosis, una paraproteinemia o un mieloma, posiblemente mieloma, usamos mitógenos de tipo B, que somos el POGWIT o el LPS. También estamos usando el I4, ¿ok? Porque queremos sacar el clon anormal, que aparezca el clon anormal, ¿ok? La detección de las células clonales anormales, de la clona normal, es bastante limitada. debido a que el clon anormal a veces no crece, ¿ok? O la morfología de los cromosomas no es tan buena, ¿ok? Entonces, a pesar de toda de este trabajo de poder conseguir cromosomas, cuando los encontramos que los cariotipos son muy complejos y les voy a explicar por qué. ¿Ok? Encontramos luego, por ejemplo, anomalías involucrando el locus 14q32. ¿Ok? Encontramos también trisomías. Lo más interesante es de que encontramos hiperdiploidía en el 50% de los casos, con copias extras del cromosoma 5, 7, 9, 11, 15, 19 y 21, que están relacionadas con el good prognosis o el standard risk que le llaman ahora. Ni siquiera buen pronóstico, sino standard risk, como diciendo, aquí no pasa nada, la enfermedad sigue su curso, ¿no? Y luego también tenemos que, por ejemplo, que si hay hiperdiploidía, y además vemos anomalías como de lesiones del brazo corto del cromosoma 17 o una 414, por ejemplo, el buen pronóstico que podría tener la hiperdiploidía está nulificado por la presencia de una deleción del brazo corto del cromosoma 17, que luego por Fish se puede ver que hay una pérdida del TP53, ¿ok? Por ejemplo, tenemos acá una hiperdiploidía. Encontramos copias extras del 5, del 9, del 11, del 15, del 19, del 21. Hasta aquí estamos hablando de lo que antes decíamos buen pronóstico. Ahora ya lo cambiaron. Ahora se llama standard risk, ¿ok? Pero los cariotipos de mieloma múltiple son muy complejos. Pueden ver este de un paciente que hemos tenido donde hay una gran cantidad de cromosomas marcadores que no sabemos de dónde vienen y encontramos una variedad de problemas cromosómicos acá. Este fenómeno que es muy típico de plasma cell modeloma, ¿no? Por ejemplo, acá tenemos una copia del cromosoma 1q, lo cual ya nos está dando la respuesta y ahora voy a decir por qué. También hay hiperdiploidía, pero si pueden comparar, los que son citogenetistas, pueden comparar el brazo corto del 17 normal con el brazo corto del otro cromosoma. Entonces, hay un material adicional que está conllevando a que se pierda el P53, ¿ok? Entonces, encontramos que en mieloma múltiple hay un fenómeno que se llama cromotripsis, ¿ok? Que es una hecatombe. Imagínense ustedes un terremoto, como los terremotos que hay en México, donde un pedazo de un cromosoma se va para el otro lugar, el otro se va para el otro lugar, se hace una ensalada de rearreglos cromosómicos. Entonces, este fenómeno se llama cromotripsis, que fue descubierto precisamente en mieloma múltiple, ¿ok? Y este fenómeno de cromotripsis está asociado con genomic instability, la instabilidad genómica, y por supuesto con muy mal pronóstico. Entonces, a veces no encontramos hiperdiploidía, entonces encontramos una pseudodiploidía o una hipodiploidía. A veces encontramos monosemia del 13, del 14, 16, 22 y de los cromosomas sexuales, ¿no? Anteriormente decíamos que la pérdida del cromosoma 13, que la dirección del 13q sigue siendo, o la monosomía del 13 sigue siendo un buen marcador citogenético, pero ahora no está considerado dentro del grupo de los parámetros para poder decir el pronóstico. Entonces, lo que hemos hecho, nosotros lo hemos quitado del panel de Fish. Si lo vemos por cromosomas, lo reportamos, pero no lo hemos incluido en el panel de Fish, ¿ok? Entonces, por ejemplo, también vemos este cariotipo, ¿no? Que está bastante complejo, pero ahí el key del asunto es que estamos con una adición del cromosoma 1, ¿no? Luego tenemos anomalías, como por ejemplo la 414, que tiene un pronóstico pésimo, la 614, un pronóstico neutral ahora se dice, ¿no? Neutral. Antes se decía el buen pronóstico. 1114 también un pronóstico neutral. Anteriormente se decía que era muy buen pronóstico, ahora se dice un standard risk, o sea que es un pronóstico que la enfermedad sigue su curso. Luego tenemos la 1416, 1420, que son pésimas, son muy malas. Luego tenemos la hiperdiploidía, que si está sola, está muy buena. La dirección del 1314, que ya no se considera ahora en el nuevo SMART, que está como muy mala. Y luego tenemos las locaciones MIC, por ejemplo, que son neutrales o adversas, algunas. Y cuando tenemos copia del 1Q, eso sí es malo. Ok, eso está pésimo. Esto dentro de la categoría y la dirección del 1P, ¿ok? Pero entonces ahora lo que estamos haciendo, aparte de tener los mitógenos para células B en mieloma, En Europa se comenzó a usar una técnica que se llama la CD138 isolation, el aislamiento de la célula CD138. Entonces, esta técnica ha revolucionado el mundo, porque ahora la estamos queriendo usar en nuestro para AML, tratando de aislar los blastos. No hemos tenido suerte todavía, ¿ok? Pero en mieloma múltiple ya ese es el estándar risk, perdón, el protocolo estándar que se está usando. Usted no puede hacer, ¿cómo se llama? Fish en un pellet que le va a dar de repente un fish normal, ¿ok? Sino que lo hacemos en esas células que están, en las células CD138, se hace el aislamiento de las células CD38 y luego se aplica el panel, ¿no? Que tenemos para mieloma múltiple, que va a abarcar todas las anomalías cromosómicas que les he dicho. Y lo que es interesante es que a veces encontramos la sobreexpresión o copias extras del 1Q, porque en el 1Q se encuentra este gen que es el CKS1B, que está relacionado con que las copias extras de este gen están relacionadas con muy mal pronóstico. Se llama Adverse Prognostic Factor. Entonces, la nueva clasificación del mieloma múltiple nos dice que estas, por ejemplo, las trisomías, la hiperdiploidía, el 11-14 y el 6-14, estándar, se conocía como muy buen pronóstico. Y ahora tenemos la de high risk con 4-14, 14 y 16, de lesión del 17P, P53 mutation, si lo hacemos por genética molecular, y la ganancia del 1Q. O sea, que esto está completamente cambiando. Si usted quiere hacer la citogenética convencional, la puede hacer, no hay ningún problema. Pero el tiro, el key player acá es el FISH, ¿ok? Entonces, esta amplificación es muy, muy interesante porque te va a causar que pongas al paciente en un higher prognostic factor, o sea, que es un mal pronóstico, ¿ok? Esto se viene estudiando desde el 2014, 16 y siguen sacando, ¿no? La amplificación se puede ver también en monoclonal gammopathy of undetermined significance y en plasma cell myeloma, ¿ok? Luego tenemos, por ejemplo, que la dirección del 1P también es importante, ¿no? Y por eso es que la sonda cubre no solamente el 1Q, sino también cubre el 1Q, el 1P, porque hay un gen acá que es el P18 que está cuya delección está asociada con un mal pronóstico, ¿ok? Entonces aquí le digo, acá viene la sonda que les decía y acá viene la amplificación que podemos ver. Entonces este paciente está asociado con mal pronóstico. Nuevamente la tablita de SMART que la estamos usando, nosotros seguimos mundialmente la clasificación de SMART en citogenética y es muy interesante que los europeos, en este caso, España, Navarra, es la que el director de la clínica de Navarra, su especialidad es el mieloma múltiple y él es el que estandarizó todas estas técnicas, ¿no? Ahora también hay un trabajo muy interesante que estamos tratando de ver si incorporamos estudio poblacional, ¿no? Porque dice que hay una contribución del germline risk en genomic instability. Y esto es muy interesante. Uno de nuestros colaboradores nos dice que hay que ver estos genes, ¿no? Como el KRAS, el NRAS, que se encuentra mutado en 20% de estos pacientes, y que sigue por el P53, el LDS3 y otros genes, que está el BRAF, por ejemplo, también. Y también tenemos otros genes como el 3, EGR1. Entonces parece ser de que tenemos que estudiar cuáles son los que están mutados en estas germline mutations que tienen esta, ¿no? Porque acuérdense que la teoría de la hipótesis de que el cáncer se hace en el momento de la concepción. Y tengo muchos ejemplos para darles, pero no vamos a hablar de ese tema. Es un tema que estoy controlando mi tiempo porque estamos con un tiempo bien corto. Ahora lo que está diciendo este grupo de colaboradores con nosotros de que tienen que hacer un panel de Next Generation Sequencing para plasma cell myeloma, ¿no? Entonces nos están diciendo de que hay que hacerlo, de que nos va a dar y que hay unos genes putativos que están relacionados con plasma cell myeloma, como estos que no teníamos nada que ver anteriormente, como el TPN11, el PKR2, que son protoncogenes y que se encuentran activos como oncogenes, y los genes supresores tumorales como el VR5 y el HUWE1, ¿ok? Entonces ahora este grupo también está haciendo el landscape de lo que pasa, de qué es lo que pasa, y están escribiendo mucho en las colaboraciones que tenemos con ellos, de que todo empieza, ¿no? Todo empieza fríamente calculado, empiezan las ciclinas a activarse, las proteínas tirosina quinasas comienzan a activarse, y comenzamos con el 11-14 y todas las regulaciones del ciclo celular, o sea que todo está dado por el ciclo celular. Yo pienso que el plasma cell mieloma es un ejemplo muy bonito de cáncer, ¿no? Porque el cáncer es un problema en los puntos de chequeo del ciclo celular, ¿no? Entonces yo creo que con esto estamos haciendo estudios también de que puede ser familiar esta enfermedad, ¿no? Entonces hay un estudio muy bonito que se ha hecho en Suecia, que tiene el año 2019, que es un colaborador que se ha dado el lujo de analizar 13.896 pacientes. Yo no tengo ese lujo, pero bueno, ellos tienen mucho dinero por el gobierno, entonces han podido establecer de que parece que hay un estudio de asociación, ¿no? De familiar, ¿no? Entonces están incluyendo ahí los polimorfismos para ver como marcadores y ver cuáles son los genes de si se puede pasar o no se puede pasar, pero los genes que están ellos enfatizando son los de que reparan el ADN, los de la apoptosis y la de la respuesta inmune, ¿no? Yo creo que este es un estudio muy interesante. Voy a repetir que este es el estándar de care que se puede hacer para mieloma múltiple, ¿ok? Que tenemos que ver todas o sea, el cariotipo que lo puedo reportar o no lo puedo reportar es irrelevante porque lo que yo tengo que decir que está asociado con el fish finding que encuentro, ¿no? Y bueno, con eso tengo también un paciente, un caso muy bonito que se estudió hace poco, donde el paciente tenía plasma cell dyscrasia, ¿ok?
5 minutos, doctor Tirado.
Perfecto. Entonces tuvimos que hacerle todas las ondas y lo que encontramos que había una 11-14 que se encuentra en el 10 al 20% de mieloma múltiple y es el estándar risk, ¿ok? Pero lo interesante del caso es de que cuando le hicimos la citogenética al pellet que no había sido enriquecido, porque estuvimos estudiando primero el FISH en las células CD1-138. Pero ¿qué creen este cariotipo? ¿Qué es lo que tiene de anomalía este cariotipo? La morfología es pésima, ¿verdad? Entonces tuvimos que hacer un FISH porque no me gustaba el 9Q ni el 22Q. Y bueno, hicimos el FISH con el BCR-ABL y resulta que el famoso paciente, además de tener mieloma múltiple, que fue detectado en la línea linfoide, porque en las células CD138, plasma cell, también tenía 922, ¿ok? Entonces pudimos ver que había una inserción y lo que le dijimos al paciente, al clínico, le dije, ¿podrías ver la morfología de tu médula? Y encontró de que también tenía una parte de su morfología tenía un gran porcentaje de blastos. Entonces había desarrollado en paralelo una leucemia mieloide crónica. Entonces fue un caso triste, pero bueno, ahí van lidiando con el pobre paciente, ¿no? O sea que le ha tocado dos casos. Y bueno, con esto quería terminar. Muchas gracias, de verdad, y un placer haber estado con ustedes. Y los invitamos para nuestro curso de citogenómica que se va a realizar del 26 al 30 de abril de este año. ¿Okay? Y la Jornada Internacional de Genética que la tenemos en presencial del 10 al 12 de agosto del año 2022, que será la última vez que se realiza en el Perú y va a ser en la ciudad de Chiclayo, en el Centro Binacional de la Embajada Americana. Y los esperamos, ¿ok? No hemos podido tenerla este año por el problema del COVID, pero de verdad, si tuvieran alguna pregunta, muchas gracias. Ahí termino, ¿ok? Disculpen la velocidad, pero estamos contra el tiempo. Gracias.
Muchas gracias, doctor Tirado. Las siguientes expositoras son Stephanie Bottomley y Caitlin Tran, estudiantes de la Universidad de California y miembros del Círculo Internacional de Estudios de Genética, capítulo Estados Unidos, presentando el tema denominado enfermedad de Tay-Sachs. Si hay alguna pregunta para las expositoras, por favor escribir en el chat al momento de finalizar la exposición.
Hi, my name is Caitlin Tran, and I will be presenting with Stephanie Bottomley about Tay-Sachs disease. So first we'll start with an introduction. Here's a brief overview of what we will be going over today. We will first start off with an introduction of what Tay-Sachs disease is and its incidence. Then we will go into its mutations, subtypes, clinical manifestation, treatment and management, current challenges, and directions for future research. So first we'll start with an introduction into Tay-Sachs disease. Tay-Sachs disease is essentially an autosomal recessive disorder that leads to progressive lethal neurodegeneration as a result of a mutation in HEXA, which is a gene that codes for the enzyme beta-hexosaminidase A, and we will delve deeper into this later in the presentation. Something to understand first is the general population incidence of Tay-Sachs disease. So since it is an autosomal recessive disease, it is rare. Only 1 in 100,000 live births are affected by the disease, while 1 in 250 are carriers. However, Tay-Sachs disease does have different incidence in certain populations, such as the Ashkenazi Jewish population. Here, 1 in 3,500 live births are affected by the disease, while 1 in 29 are carriers. Other populations with a high incidence and thus a high risk of Tay-Sachs disease include the Cajun community of Louisiana, the Old Order Amish and Dutch communities of Pennsylvania, and the non-Jewish French Canadians living near St. Lawrence. Usually, if at least one person in a couple is a part of these populations, then a genetic screening test is often recommended since they are at a higher risk of having a child affected by Tay-Sachs disease.
All right, on to some mutations. So Tay-Sachs disease is caused by the loss of function mutations in HEXA, located at 15q23. It follows an autosomal recessive inheritance pattern and codes for the alpha subunit of enzyme beta-hexosaminidase A. Now, although this only causes one disease, it is actually root caused by over 120 mutations that result in either gene insufficient enzymatic activity or completely absent enzymatic activity. These are usually single base substitutions, but they can be caused by small deletions or small insertions. So a little bit more about beta-hexosaminidase A. Under normal circumstances, lysosomes in neural cells will have beta-hexosaminidase A to cleave waste products such as GM2 ganglioside. In a normal cell, this is cleaved and the body can excrete this as a waste product normally. However, in someone with Tay-Sachs disease, they either have completely insufficient enzymatic activity, very low enzymatic activity, or no enzyme at all. So these waste products, the gangliosides, are not cleaved and instead build up inside the neurons. Now these build up to toxic levels and the brain matter deteriorates and is damaged beyond repair. On the left here, you'll see a normal neural cell, and on the right, a neural cell represented affected by Tay-Sachs disease. And you'll notice decreased exocytotic activity and GM2 accumulation where observed. GM2 are gangliosides. Here we show an MRI of a 9-month-old male with Tay-Sachs disease. The brain here has already begun deteriorating beyond repair. So some subtypes. Due to the number of mutations that cause Tay-Sachs disease, it actually presents with clinical heterozygosity. So you have 3 subtypes: infantile, juvenile, and adult. And they are dependent on which mutation is present, which will in turn determine the amount of functional enzyme made, which will determine severity. So infants with Tay-Sachs diagnosed usually have almost no enzymatic activity at all, and their disease progresses quite severely quite quickly. Juveniles are similar, and adults usually have between 10 and 15% functional enzyme, which is why their disease progresses slower, although it's just as lethal, and they are diagnosed later.
So after understanding mutation, understand how it presents itself. As mentioned before, the autosomal developmental delays in regression, inattentiveness, exaggerated startle response, a cherry red spot on the retina, as seen in this image here, generalized hypotonia with sustained clonus, and hyperreflexia. If a patient displays any of these symptoms, then gangliosidosis due to Tay-Sachs disease is often suspected and tested for. However, as mentioned before, there are 3 different subtypes of Tay-Sachs disease: infantile, juvenile, and adult, that can present with slightly different symptoms, and we will delve a little deeper into each one. So for infantile Tay-Sachs disease, you generally see all of the aforementioned symptoms: mild motor weakness, irritability, hypersensitivity, especially with the exaggerated startle response, problems with the eyes, including the cherry red spot that eventually leads to blindness, narrowing of the retinal vessels, nystagmus, which is just repetitive, uncontrolled movement of the eye that can reduce vision and depth perception, and optic atrophy, which is the death of retinal ganglion cell axons of the optic nerve. Patients can also have developmental delays and regression by 4 to 6 months of age. They can become less responsive by 8 to 10 months of age. And they can have seizures, usually myoclonic seizures, by 12 months of age. They can also have ataxia, which is the degeneration of the nervous system, dyskinesia, the impairment of voluntary movement, sleep disturbances, episodes of screaming, macrocephaly, or the enlargement of the brain by 18 months of age, decerebrate posturing, dysphagia, or swallowing difficulties, unresponsive and vegetative state by 2 years. They can also potentially have cardiovascular symptoms, but it's rare, and they're also susceptible to. Patients are often resistant to mycobacterium infections because they lack the alpha subunit of the hex A enzyme but overproduce the beta subunit, which warrants immunity. But they are prone to infections like respiratory infections that often lead to death. And you can see some of the symptoms in this image here, like muscle weakness, decerebrate posturing, and unresponsiveness. Meanwhile, juvenile Tay-Sachs disease often affects children between the ages of 2 and 10 years old. Usually an earlier onset leads to a faster disease progression. Some of the symptoms are similar, such as incoordination, clumsiness, muscle weakness, ataxia, dysarthria, which is a motor speech disorder, dysphagia, and progression of spasticity. Other symptoms like the cherry red spot are not usually observed, but optic atrophy and retinitis pigmentosa can be observed later in life. By 10 to 15 years old, a vegetative state with decerebrate posturing usually occurs, and patients often die shortly after as a result of respiratory infections. And you can see some of the symptoms again, such as muscle weakness, decerebrate posturing, and reduced vision in this picture here. Adult Tay-Sachs disease is infrequent, so diagnosis is often delayed by at least 8 years. The disease is also less aggressive because of the smaller mutation and residual activity of the enzyme. Typical symptoms like muscle weakness usually begin in adolescence or early adulthood, but they can also appear later in a patient's life, like in their 20s or 30s. The phenotypes are usually different, but generally patients can have progressive motor neuron disease, dystonia, where muscles contract uncontrollably, and spinocerebellar degeneration. Mentation and verbal skills are often affected later in life, and 40% develop psychiatric manifestations without dementia, including psychotic depression, hebephrenia, schizophrenia with disorganization of thoughts, delusions or hallucinations, paranoia, or bipolar symptoms. And again, you can see some adult Tay-Sachs disease patients trying to improve muscle weakness in these pictures here.
So on to treatment and management. So unfortunately, there are no known treatments or cures for the disease itself. Current therapies aim to make the patient more comfortable as the disease progresses. Typically, this includes anticonvulsants, diet control, preventing blockage of airways, and managing complications such as infections. So the main way to actually prevent Tay-Sachs disease is by genetic screening before children. And one organization that has really spearheaded this is Genética. They test Ashkenazi Jewish couples before marriage to reduce the risk of conceiving a child with Tay-Sachs disease and cancel them on the possible risks associated with having a child with Tay-Sachs disease. So some current challenges. Targeting hexa and beta-hexosaminidase A directly is actually extremely difficult in lab. And this is made even more complicated by the significant number of mutations that can cause Tay-Sachs. There are around 120 that have been entered into the current literature. So future research, we've seen moderate success in mice models and some very small human models. Not human models, human trials. So we've mostly seen enzyme replacement therapy, bone marrow transplant, and gene therapy have the most promising results thus far. So with enzyme replacement therapy, somatic symptoms may decrease, but the blood-brain barrier actually does not allow enzymes to pass through with most delivery methods. So it's very difficult to actually synthesize both subunits of the enzyme in lab. With bone marrow transplants, so these did help. They showed an increase in enzymatic activity in leukocytes and plasma. However, the effects were temporary, usually less than 2 years, and disease progression is not slowed. Patients are slightly more comfortable, but the overall result is the same. And with gene therapy, the results of gene therapy are very promising. However, delivery of wild-type hexa has to be done intracranially to get beyond the blood-brain barrier, and they use adeno-associated virus-based vectors. The crux with this, though, is that there's a very, very narrow window in which the results can be seen because this needs to be administered regularly before the disease progresses. And typically by the time someone is diagnosed with Tay-Sachs disease, their brain matter has deteriorated too far for this to provide any relief or reversal.
Sorry to interrupt. You have 5 minutes remaining. Okay.
Those are my references. Thank you. Are there any questions?
Great. Thank you so much, Stephanie and Caitlin, for your presentation. If there are any questions, you can feel free to leave them in the chat below. Our next presenter is student Percy Zapata. He's a student at Santo Toribio de Mogrovejo Catholic University, presenting the topic called Sézary syndrome. If there's any questions for the presenter, please write it in the platform chat at the conclusion of the presentation. Thank you. Unfortunately, Percy can't be with us today, so I am going to share his presentation recording. Is everyone able to hear the presentation? Okay. Let me unshare.
Muy buenas tardes con todos.
Can
Mi
we hear
nombre es
now?
Percy Zapata, estudiante de Medicina Humana en la Universidad Católica Santo Toribio de Mogrovejo. Pertenezco al Círculo de Estudios de Genética del capítulo de Chiclayo. A continuación, para el workshop, voy a presentar el síndrome de Sézary. ¿Qué es el síndrome de Sézary? Este constituye la fase leucémica de la micosis fungoide. Va a estar caracterizado por presentar eritrodermia, poliadenopatías superficiales y un elevado número de linfocitos atípicos. En la figura de la derecha observamos un extendido de sangre periférica de un paciente con síndrome de Sézary. Es por ello que observamos células de Sézary. Estas células son células tumorales, son linfocitos de tamaño pequeño, mononucleares, de núcleo cerebriforme, riñonado, como podemos observar ahí, ¿no? Con un citoplasma escaso, una cromatina muy agrupada y núcleos discretos. Estos linfocitos tienen una característica de que van a tener un proceso o un fenómeno llamado epidermotropismo, y este epidermotropismo va a generar de que hay una formación de microabscesos, llamados microabscesos de Pautrier, y esto va a ser como la marca del síndrome de cesárea, y con esto lo vas a identificar así a ojos cerrados de que va a ser un síndrome de cesárea. Bueno, la prevalencia del síndrome de cesárea es de hombres con una proporción de 2 a 1 respecto a mujeres, afectando en un rango de edad de adultos entre 60 a 70 años. Las características de la enfermedad se puede dar tanto de nuevo o se puede iniciar previa dermatosis inespecífica. También se puede presentar en uno o varios componentes a la vez en la enfermedad, ¿no? Puede solo tener una eritrodermia, puede tener otro tipo de compromisos, ¿no? Tumores, otras manifestaciones, o solamente puede presentar una. Su evolución es mucho más rápida y agresiva que la micosis fungoide. Es por ello, a que es una leucemia, es un cáncer, entonces obviamente va a ser algo agresivo. Y una vez que va a estar comprometidos tanto órganos linfáticos pertenecientes al sistema linfático u órganos viscerales, puede ser fatal y eso va a ser determinado según el estadio en el que esté el paciente. En la imagen de la derecha podemos observar una eritrodermia, viniendo eritro de rojo y dermia de la dermis, ¿no? Entonces observamos acá máculas de coloración rojiza, pequeñas, en la piel de nuestro paciente con síndrome de cesárea. La etiología va a ser desconocida, va a estar asociada, pero sin embargo va a estar asociada al Human T lymphopic virus 1, al Epstein-Barr y al citomegalovirus. Para la citogenética, en un estudio se observó que se origina a partir de un linfocito TCD4 periférico y mediante la clonación se detectó un reordenamiento monoclonal en el receptor de células T, llamado también TCR. Este receptor, en estudios de hibridación in situ con fluorescencia, se observó de que no va a tener, de que hay una ausencia de rupturas que vayan a implicar alguna de las regiones de este TCR. Sin embargo, se encontraron pérdidas y ganancias de ADN. Las pérdidas de ADN se van a dar básicamente en su mayoría en el cromosoma 1, brazo corto, 38%, cromosoma 17 en su brazo corto, en el 21%, cromosoma 10, brazo largo en su 15%, cromosoma 19 en su 15%. También se encontraron ganancias de ADN en su cromosoma 4, brazo largo 18%, la mayoría, en el cromosoma 18 en 15%, cromosoma 17 en brazo largo en su 12%. Por último, en el cromosoma 1, brazo largo, se identificaron zonas de lesión común mínima en el cromosoma 1 de brazo corto, región 3, banda 6, y en el cromosoma 1, brazo corto, región 2, brazo 2. Debido a que el estudio de la citogenética nos detecta un amplio gama de alteraciones asociadas al síndrome de cesárea, es que no hay un tratamiento específico hasta la actualidad. Es por ello que el tratamiento suele ser muy general: fototerapia, radioterapia, interferón alfa, que básicamente esos tres son como los pilares que vamos a tener para el tratamiento del síndrome de cesárea. Sin embargo, también se emplea quimioterapias, pero sin mucho éxito, ¿no? O sea, el éxito suele ser muy limitado, dependiente del estadio del paciente, no es muy específico. Bueno, el fenotipo va a ser, otra vez, como mencioné, origen de un linfocito TCD4 periférico observado en estudios de clonación y reordenamiento monoclonal, células de receptor auticiva. Para el diagnóstico tenemos parámetros, entre ellos que por milímetro cúbico tenemos que encontrar alrededor de mayor de 1000 células de cesárea circulantes. También tenemos que encontrar más de 10 CD4 en relación con CD8 y expresión aberrante de marcadores PAN-T. También vamos a evidenciar células teclonadas, que son células anómalas, cancerígenas. Y este paciente va a ser clasificado según el sistema de estadiaje adoptado por Van y Humber, que es el siguiente, ¿no? Tenemos 4 estadios, desde el 0 hasta el 4, y cada uno tiene un significado, ¿no? El estadio 0 son células anormales diré, pero no hay cáncer, básicamente, ¿no? O sea, son células anormales, puede haber un carcinoma, van a estar diseminadas en tejidos cercanos, o sea, no van a estar diseminadas en tejidos cercanos, y estas no pueden convertirse en cáncer. Sin embargo, en el estadio 1 hay cáncer presente, pero va a determinarse si es 1, 2 o 3 en cuanto a mayor sea el tamaño del tumor, o haya más tumores, o se haya extendido a tejidos cercanos, ¿no? Y en el estadio 4 se va a determinar básicamente porque ya es un cáncer diseminado, es decir, una metástasis en el paciente. Es por ello que el pronóstico nos dice que mientras que el estadio sea mucho más temprano, un estadio 0, un estadio 1, va a ser un pronóstico mejor, ¿no? O sea, por eso es que el estadio clínico es un indicador muy importante. Los pacientes que presentan enfermedad cutánea limitada van a tener un pronóstico excelente. Y los pacientes con tumores cutáneos, eritrodermia generalizada, como observamos anteriormente, un síndrome cesárea o enfermedad extracutánea suelen tener una supervivencia corta, que va en un rango de entre 1 a 4 años, ¿no? Y bueno, esto es básicamente mi exposición para el workshop de genética del día de hoy. Muchas gracias con todos.
Okay, we give Percy a virtual thank you for his presentation that was recorded. Our next presenters are students Lena Nabipur and Ruby Tang. Lena and Ruby are students from the University of California, Los Angeles, as well as being members of the International Circle of Genetic Studies. They will be presenting on the topic of thalassemia. If there are any questions for the presenters, please write them in the platform chat at the conclusion of their presentation. Thank you.
Okay. Hello, everyone. So today, Ruby and I will be speaking about the topic of thalassemia. So with a quick overview over thalassemia, we'll be starting with an introduction. So the incidence, the clinical presentation, disease severity, general population, risk factors, as well as the types and subtypes, so alpha thalassemia and beta thalassemia, the genetic basis, so deletional and non-deletional mutations, so rearrangements, missense, et cetera, as well as the treatment and management, so current and future treatment strategies. So the thalassemias represent the most common monogenic disorder worldwide. The term thalassemia itself refers to disorders associated with the defective synthesis of either alpha or beta globin subunits of hemoglobin, which are inherited as pathologic alleles of one or more of the globin genes located on chromosomes 11 and 16, which we'll go into more on the following slide. Because thalassemia heterozygosity confers some immunity against malaria, there is a particularly high incidence in the Mediterranean basin, the Middle East, subtropical regions of Africa, as well as various locations in Asia. However, it is also seen that cases of thalassemia also occur sporadically in basically every ethnic group and geographic location as well. Although there is a difference in the co-inheritance of other genetic modifiers, the underlying pathology can be characterized by decreased hemoglobin production and red blood cell survival, resulting from the excess of unaffected globin chain, which form unstable homotetramers that precipitate.
Early precipitation in the red blood cell lifespan caused by red blood cell damage and severe hemolysis is associated with ineffective erythropoiesis and extramedullary hemolysis. With this severe ineffective erythropoiesis, chronic anemia, and hypoxia, there is a risk for increased gastrointestinal tract iron absorption. These cases lead to bad prognoses due to severe anemia. One of the only options known to date to reduce morbidities and prolong survival has been iron chelation therapy. The thalassemia syndrome is characterized according to which of the globin genes, the alpha or the beta, is affected. These are seen in the image on the right. In patients heterozygous for a globin mutation, the quantitative amount of mutant hemoglobin depends on a variable of factors, such as the number of functioning genes in the individual, the stability of the mRNA, the rate of synthesis and stability of the gene product, and the efficiency with which abnormal hemoglobin is assembled. In heterozygotes, the beta globin mutant hemoglobins comprise of about 50% of the total hemoglobin. And since normal hemoglobin is produced from 4 alpha globin genes, this means that the mutants usually comprise of about 25% of the hemolysate. This fraction increases also if the deletional forms of alpha thalassemia are also present in the same subject.
So there are two main types of thalassemia: alpha, which is characterized by defective synthesis of the alpha globin subunits of hemoglobin, as well as beta, which is characterized by the defective synthesis of the beta globin subunits. While the former is mostly caused by deletional mutations, the latter is mostly caused by non-deletional mutations. And both types have many subtypes, which are defined according to clinical severity. Thalassemia trait, which is the least clinically severe category of thalassemia, encompasses mostly asymptomatic carriers who generally do not require treatment. In contrast, those with thalassemia intermedia generally have moderate anemia and occasionally require red blood cell transfusion, with the alpha thalassemia form being called hemoglobin H disease due to the formation of hemoglobin H from excess beta globin. For the category of highest clinical severity, thalassemia major, those with the alpha form present very differently from those with the beta form. The alpha form is called hemoglobin Barrett's hydrops and it is characterized by severe anemia and hypoxia, fetal abnormalities, and as a result, death either in utero or shortly following birth. While the beta form is less severe, those afflicted require regular red blood cell transfusion and die within 10 to 20 years if not fully treated. There are also some rare cases of thalassemia inheritance. The first is dominantly inherited beta thalassemia, in which those who are heterozygous for dominant beta thalassemia mutations exhibit typical symptoms of anemia. However, this condition is relatively rare, and most of these mutations have only been reported once. The next is uniparental isodisomy, in which patients develop late onset beta thalassemia despite only having one parent who is a beta thalassemia carrier. The last we will discuss is de novo mutation. So although most thalassemia cases are the result of parental inheritance, there have been some cases in which de novo mutations in the alpha and beta globin genes have resulted in thalassemia, but all of these rare cases have yet to be fully characterized.
So thalassemias occur when mutations cause selective deficiencies in the synthesis of either alpha-like or beta-like globin. However, it is the imbalance in accumulation of the subunits that creates these inclusion bodies and thus pathophysiology of the disease. There are several hundred known mutations that can cause thalassemias, which can affect every step in the globin gene expression. For the purpose of these explanations, alpha and beta thalassemia will be considered together because of the genetic and molecular mechanisms being quite similar. Deletion of one or more alpha globin genes is the most common mechanism accounting for alpha thalassemia, while on the other hand, the complete deletion of the beta globin gene has been reported rarely. The classical clinically harmless forms of hereditary persistence of fetal hemoglobin seem to arise from deletional events that remove large regions of DNA from beta gene clusters. These deletions eliminate both delta and beta globin structural genes. The deletions of the locus control region, or the LCR region, are very rare but are informative forms of both beta and alpha thalassemia. These prototypical mutations were associated with the total absence of beta globin synthesis, even though the beta globin gene and its promoters and enhancers were normal. As far as gene arrangements,
gene rearrangements. The HB-Lepore syndrome arises from unequal crossing over and recombination events between the closely linked delta and beta globin genes. This event fuses the 5' end of the delta globin gene with the 3' end of the beta globin gene, which produces a fused globin polypeptide with altered electrophoretic mobility. This event eliminates any intact beta or delta locus from the chromosome. HB anti-Lepore is the reciprocal product of the unequal crossover events in HB-Lepore, in which the 5' region is encoded by beta globin gene sequences and the 3' end by delta globin sequences. This retains normal loci such that the thalassemia does not occur. The HB Kenya results from an unequal crossover and recombination event between the beta and A gamma globin genes. The only globin that can be encoded by this is fetal hemoglobin, containing only the G gamma form and the A gamma beta chain form. For alpha thalassemia, since each of the 2 copies of the normal alpha globin genes codes for the same exact sequences, it's not possible to create structural alpha globin protein variants that are analogous to the previously mentioned rearrangements by unequal crossover and recombination. However, it is possible that common forms of alpha thalassemia appear to arise from deletion of 1 copy of alpha globin genes are actually due to unequal crossover that fuse the 2 alpha globin genes together. Mutations affecting transcription only account for a small percentage of thalassemia cases. However, these point mutations are important to consider because of their ability to alter the efficiency of the promoter or the enhancer sequences. Many mutations are known to disrupt normal splicing of the mRNA precursor. Some thalassemia splicing mutations directly affect canonical splicing signals used to mark the beginning and the end of introns, which abolish the normal splicing or simply reduce it to a variable degree. Mutations that alter the consensus site reduce production of the alpha or beta globin mRNA, which appear to be catabolized quickly such that abnormal mRNA products are unable to accumulate. Another class of splicing mutations includes the activation of a cryptic site. Base substitutions, small deletions, or small insertions of DNA can convert a site within an exon or intron that normally bears a slight resemblance of a splice site into something with a much stronger signal. Accumulation of mRNA products depends on the efficiency with which a cryptic site is used and the stability of the abnormally spliced product. Mutations that create premature nonsense codons account for the most common forms of beta thalassemia, which form a truncated fragment of the globin. The physiological consequences of translation termination have a protective phenomenon also that serves to eliminate abnormally translated mRNAs. However, this is seen to only occur for premature stop codons in the 1st or 2nd exons.
Although the phenotypic spectrum for thalassemia is quite wide, there are a couple of clinical manifestations that are very common across a majority of thalassemia patients. These include mild to severe anemia, which is related to the quantity of functional globin chains the patient has, jaundice, and skeletal changes such as facial deformities, osteoporosis, body masses, and bone pain, all of which are more severe and more likely to occur in patients with more severe types of thalassemia. There are also cardiovascular complications, such as heart failure, arrhythmia, hypertension, and thrombosis, as well as metabolic abnormalities like hypogonadism, hypothyroidism, growth impairment, and diabetes. Lastly, patients often suffer from iron overload, which is a result of the patient receiving frequent blood transfusions and not being able to excrete the excess iron properly. And many patients may also suffer from hepatosplenomegaly and an increased risk of cancer. So the well-characterized structure and function of the alpha and beta globin genes provide a good framework for researching thalassemia treatment. Currently, there are a variety of standard treatment and management strategies for thalassemia patients. So for diagnosis, thalassemia can be diagnosed prenatally using PCR and hybridization assays, using fetal DNA obtained using chorionic villus sampling or amniocentesis. After birth, transfusion therapy may be beneficial for children with growth impairment, facial or bone abnormalities, or low hemoglobin levels. With transfusion therapy, confounding factors that may affect the severity of anemia, including blood loss, folic acid deficiency, and co-inheritance of the glucose 6-phosphate dehydrogenase deficiency must also be addressed. Spleen removal can similarly improve the level of hemoglobin as well as decrease the transfusion requirement and lessen iron overload. Additionally, since blood transfusions introduce excess iron that the body may not be able to get rid of, iron chelation therapy can be used to mitigate this effect and therefore prevent subsequent cardiac complications. Lastly, hematopoietic stem cell transplantation and cord blood transplantation both aim to introduce healthy stem cells in patients with thalassemia, but the latter is mainly used in pediatric patients because of the relatively small number of stem cells in the umbilical cord blood. Lastly, some future therapies include fetal hemoglobin inducers, which can decrease the severity of anemia by modifying fetal globin gene expression to increase gamma globin chain production, as well as gene therapy, which could entail transferring a wild-type beta globin gene sequence and its promoter sequence into stem cells. However, more research has to be done on both of these therapies before they can widely be implemented in thalassemia treatment. So these are currently not used. Everywhere. And that concludes the end of our presentation. So if you guys have any questions, please feel free to type in the chat or unmute and we'll be around.
Muchas gracias. A continuación, o el siguiente expositor será el estudiante Kevin Bonilla, estudiante de la Facultad de Ciencias Biológicas de la Universidad Nacional Pedro Ruiz Gallo, quien nos expondrá acerca del tema de hipercolesterolemia familiar. Si hay alguna pregunta para el expositor, por favor escribirle en el chat de la plataforma al finalizar la exposición.
Hola. Hola, buenas tardes, ¿me escuchan?
Sí.
Sí, ya, porque como estoy transmitiendo en Facebook, a ver, ya. Muy buenas tardes, mi nombre, como ya dice Bruno, mi nombre es Kevin Bonilla León, soy estudiante de Biología de la Universidad Nacional Pedro Ruiz Gallo, integrante del Círculo de Estudios. El día de hoy voy a tratarles de manera general lo que es hipercolesterolemia. Siguiente diapositiva, por favor. Bueno, la hipercolesterolemia es la alta concentración de colesterol malo, el cual mayormente no presenta síntomas y generalmente se detecta cuando aparece una enfermedad del corazón o cerebrovascular, también hipertensión arterial, obesidad o diabetes. ¿Entiende? Según el Centro para el Control y Prevención de Enfermedades, alrededor de 73 millones o el 31,7% de adultos en los Estados Unidos tienen niveles altos de colesterol malo, del cual solo el 48,1% recibe un tratamiento para reducir estos niveles de colesterol malo. En los Estados Unidos, el nivel más alto de colesterol malo ocurre en hombres hispanos, seguido de los hombres afroamericanos, y también como dato se conoce que la enfermedad, la hipercolesterolemia, es más común en mujeres que hombres. Siguiente. Con respecto a la genética de esta enfermedad, los genes asociados a la hipercolesterolemia son LDLR, APOB, PCSK9, LDLRAP1, de los cuales el primero, el LDLR, es el más frecuente, ya que se encuentra asociado aproximadamente al 90% de los casos, seguido de la APOB, que, bueno, este representa entre el 5 al 10% de los casos. En la imagen vemos que el gen LLR se encuentra ubicado en el brazo corto del cromosoma 19 en la posición 13.2. Siguiente. Vamos a hablar un poco más del gen LDLR, ¿no? Este gen, como dije, se encuentra en el brazo corto del cromosoma 19, ¿verdad? Es el encargado de codificar el receptor colesterol malo o el receptor LDL. Tiene un tamaño de 5.3 kilobases. Este receptor se une a una lipoproteína de baja densidad que van a ser los principales portadores de colesterol malo en la sangre, que también son los principales portadores de colesterol en la sangre. Estos receptores juegan un papel importante en la regulación de las concentraciones de colesterol, siendo muy abundantes en el hígado. Con respecto a las mutaciones, este gen cuenta con 5 mutaciones, de las cuales algunas reducen el número de receptores de lipoproteínas de baja densidad, otras alteran la capacidad de los receptores, que como consecuencia van a generar altas concentraciones de colesterol en la sangre. Siguiente. Siguiente. Ya. El cuadro clínico. En el cuadro clínico vamos a observar niveles altos, niveles elevados de colesterol plasmático, presencia de una enfermedad cardiovascular prematura y la presencia de xantomas, que es una afección cutánea caracterizada por la formación de placas o nódulos amarillos. Siguiente. Para el tratamiento. Acá viene a ser muy importante los cambios en el estilo de vida, tanto en la alimentación como en la actividad física. Esto es como principal tratamiento para la hipercolesterolemia. Pero además de eso se deben incluir fármacos como las estatinas, las resinas, el ácido nicotínico, ezetimibe y anticuerpos monoclonales. Bueno, eso sería todo. Bueno, mis conclusiones. Bueno, como conclusión respecto al tema, podemos concluir que esta es una enfermedad que padecen en su mayoría mujeres de países hispanos de carácter genético, hipercolesterolemia familiar, y está relacionado a mutaciones en el gen LDLR, en mayor frecuencia, ¿no? Que se manifiesta con síntomas como dolores de cabeza, fatiga, xantomas en la persona que padece la enfermedad. Muchas gracias.
Muchas gracias, Kevin. Las siguientes expositoras son las estudiantes Vanessa Chia y Hannah Robinson, estudiantes de la Universidad de California y miembros del Círculo Internacional de Estudios de Genética, capítulo de Estados Unidos, presentando el tema denominado distrofia muscular de Duchenne. Si hay alguna pregunta para el expositor, por favor escribirle en el chat de la plataforma al finalizar la exposición.
Hannah, can you please share?
Yeah, I'll try to share.
Okay, yeah, I can see that. I'm Vanessa.
And I'm Hannah.
And we'll be presenting Duchenne muscular dystrophy.
So Duchenne muscular dystrophy, abbreviated DMD, is a genetic condition that causes muscle weakness. It is the most common type of muscular dystrophy, with an incidence of 1 out of every 5,000 boys. The Duchenne type dystrophy is an X-linked genetic disorder affecting primarily boys with extremely rare exception,
and a problem in this gene is known to result in a deficiency of the protein in muscle fibers called dystrophin. The symptoms first present in early childhood, and they increase in severity as the child gets older, and untreated children become wheelchair-bound by the age of 12 years and die of cardiorespiratory complications in their late teens to early 20s. So for the clinical presentation, DMD presents usually around 3 to 5 years of age. The presenting symptoms include gross motor delay, gait abnormalities, difficulty rising from the ground, and frequent falling. Most boys gain strength and motor skills, albeit to a lesser extent than their peers, until around 6 years of age. And then after this stage, progressive deterioration in strength begins to occur. Chronic respiratory insufficiency secondary to restrictive lung disease is universal amongst DMD patients. The vital capacity increases until around the 12 years of age and then decreases by 4 to 8% per year. Additionally, orthopedic complications such as scoliosis are frequent, and heart failure and respiratory complications become the most common cause of death in DMD patients around the second or third decade.
So now we'll be talking about the genetic basis of DMD. The dystrophin gene is located on the X chromosome, and DMD is an X-linked recessive disease affecting mostly males, and it is maternally inherited as males only have one X chromosome. It is reported that female carriers are usually unaffected, except for rare cases of abnormal X chromosome inactivation. And for symptomatic females, they can display symptoms such as muscle weakness, increased creatine kinase levels, also known as CK, which is an enzyme found in the heart, brain, or skeletal muscle, and occurs with increased muscle damage and hypertrophy of calf muscles. And according to the literature, 2/3 of DMD cases result from an X-linked recessive pattern, while 1/3 of cases are a result of spontaneous mutations where affected individuals did not inherit the mutation from their parents. Since Duchenne muscular dystrophy is caused by mutations in the DMD gene, here is some background information on it. The DMD gene, also known as the dystrophin gene, is the largest known human gene, and it spans 79 exons, and it is approximately 2.2 megabases in size. It is located on a short arm of the X chromosome, specifically at XP21.2. And the DMD gene encodes for the protein dystrophin. So what is dystrophin? Dystrophin is a structural protein, and its main function is stabilizing muscle cell membranes and acting as a shock absorber during muscle fiber contractions. Essentially, muscles are made up of long tubular cells called myocytes, as pictured in the top panel, and these can contract to cause flexing of your muscles. And this protein dystrophin that is illustrated in the red chain in the diagram is located near the membrane of the myocytes. And this dystrophin acts like a chain that links the actin cytoskeleton of the myocyte to the extracellular matrix, and it is made up of an actin binding site end that attaches to the cellular skeleton, and the central rod and the dystroglycan binding, and that attaches to the dystroglycan complex, and that is anchored to the extracellular matrix itself. And this linkage prevents myocyte membrane damage when the muscle contracts. So in the bottom panel, we see a shortened or non-functional dystrophin protein caused by mutations in the DMD gene. And every time the muscle contracts, small rips will appear in the myocyte membrane, and this leads to chronic muscle damage, inflammation, and eventually replacement of muscle fibers by fat and fibrotic tissue, and this will cause a loss of muscle function. So now we'll be talking about the different possible mutations in the DMD gene. Thousands of different dystrophin mutations have been identified in DMD patients, and various mutations can occur in this gene, such as deletions, which make up around 68% of cases, and duplications, which make up around 11% of cases, and also point mutations, which make up around 20% of cases. And both large and small deletions and duplication mutations lead to the incorporation of aberrant amino acids into the protein during translation, and this leads to a frame shift mutation at an exon level. And often the resulting incorrect reading frame contains many stop codons, and this leads to the premature termination of translation and production of a shortened protein. And the resulting dystrophin proteins lack the crucial domain that connects to the extracellular matrix and are therefore not functional and are unstable. And point mutations can also convert a codon or an amino acid into a stop codon, and which will result in a premature termination of protein translation. In addition, point mutations or small deletions or insertions can disrupt the splice site. And during the splicing process, introns are removed by the splicing machinery, and generally an exon for which the splice site is mutated will no longer be recognized by the splicing machinery, and that leads to the exon being excluded from the mRNA. And so thus splice site mutations generally cause a single exon deletion at the mRNA level, and this will also lead to a partially functional or non-functional dystrophin protein. And lastly, for patients with Duchenne muscular dystrophy, there is no known genotype or genotype-phenotype correlation identified, as the disease severity can vary for the same mutations, sometimes even within the same family, and there's also variation in the extent to which the heart is affected. Now we will talk about the diagnosis of DMD. It is said that the timely and accurate diagnosis of DMD is crucial, and diagnosis typically begins in early childhood, and it is performed after certain characteristic symptoms arise. These symptoms include impaired muscle function, which is weakness and clumsiness, a Gower's sign, which is weakness in hip muscles and difficulty with stair climbing. And first, individuals that are being tested for DMD will undergo a serum analysis, which will reveal the presence of elevated muscle enzymes due to leakage in the bloodstream. And the most notable enzyme in this case, it's creatine kinase, also known as CK. And upon these findings of the individual displaying delayed muscle function, speech delay, and high CK levels, patients are generally referred to a neuromuscular specialist, and they will perform a genetic analysis of the DMD gene to confirm whether the patient has DMD. Since 70% of patients or individuals with DMD have single exon or multi-exon deletion or duplications in the dystrophin gene, the first confirmatory genetic test will be using MLPA, or comparative genomic hybridization assay, to assess for dystrophin gene deletion and duplication. And if this deletion or duplication test is negative, next generation sequencing will be used to screen for remaining mutations attributed to DMD, which include point mutations, nonsense or missense, small deletions, and small duplications or insertions. And if this genetic sequencing does not confirm diagnosis, then a muscle biopsy sample will be taken, as an immunohistochemistry of tissue sections or a Western blot of muscle protein extract to test for the presence of the dystrophin protein.
So for treatments and therapies, current treatments such as corticosteroid treatment, surgical management, cardiac and respiratory intervention have improved muscle function, survival, and quality of life for those with DMD. However, the progression and eventual outcome of the disease has yet to be modified with these sort of treatments. Regarding gene therapies for DMD, the clinical development of gene therapy has largely centered on dystrophin gene replacement therapy. So the goal of the gene replacement is to restore the contractile capacity of the skeletal muscle by introducing a functional copy of the dystrophin gene in muscle fibers. But for this method to work, the delivery of a new dystrophin gene to all muscles in the body is needed, and the size of the full dystrophin gene poses a challenge. If you refer to the picture on the right, the vector for transferring the dystrophin gene to muscle, called adeno-associated virus, abbreviated as AAV, is a non-enveloped virus that could be engineered to deliver DNA to target cells, but it cannot be used for the large dystrophin gene. So in response, researchers have created microdystrophin genes with the size of 6 to 8 kilobases, along with dual AAV vectors, which are seen in the bottom right of the picture. However, there are some limitations to this method. AAV might be depleted over time, and microdystrophin might not be able to completely halt the dystrophic process. So although gene therapy mediated by AAV vectors appears able to effectively treat muscle pathology, the restoration may not be long-lasting beyond a few years, which may not be suitable for treatments intended to last a decade or longer. Another therapy is the suppression of the premature stop codon, which would allow for the generation of a full-length dystrophin protein. However, this is only applicable to patients with the nonsense mutations, which is around 10 to 15% of all DMD patients. However, this method does have inconsistent results. In some studies for a medication called adalurin, which uses the suppression of the premature stop codon, it has demonstrated inconsistent functional improvements of the heart and skeletal muscles in DMD patients and failed to indicate dystrophin expression. But one of the more promising therapeutic approaches for DMD is exon skipping therapy. Exon skipping therapy targets the splicing of dystrophin pre-mRNA by employing synthetic specific antisense oligonucleotides, abbreviated AON, which is seen in the picture on this slide, thereby skipping at least one exon and restoring the reading frame. This results in a truncated but still functional dystrophin protein, and it can improve the severe DMD phenotype into a more mild symptom of Becker muscular dystrophy. And 83% of DMD patients with deletions in the dystrophin gene can be targeted and improved by the exon skipping. And then also the exon skipping therapy can target different exons to skip, which can also target various DMD patient subpopulations. This method also has some limitations, such as the low efficacy in cardiac muscle, poor cellular uptake, and fast clearance from the circulation system. So these limitations basically mean that the AON-mediated exon skipping has short-lived effects. However, the relatively new CRISPR-Cas technology may offer a more permanent alternative for the AON therapy. Diseases caused by a single gene mutation, such as DMD, are good candidates for CRISPR. And the correction approaches for DMD include permanent exon removal, exon skipping, exon reframing, and exon knock-ins. However, CRISPR is still in its early stages of development. It has only been tested on animal cells and induced pluripotent stem cells, so extensive preclinical investigation is still required at this point. However, this technology has nevertheless opened up new opportunities for correcting monogenic neuromuscular diseases, including DMD. These are our references.
Thank you for listening. Please enter your questions into the chat if you have any.
All right, so if there are no questions, the next presenter is Fabian Grales, a student at William Patterson University in the Biological Sciences Department, and he will be presenting on xeroderma pigmentosum. So if there are any questions for the presenter, please write them in the platform chat at the end of the presentation.
Hello, everyone. My name is Fabian Grales. Thank you, Emily, for the introduction. Today, I'll be presenting to all of you on the topic of xeroderma pigmentosum. Xeroderma pigmentosum is a rare recessive autosomal cancer-prone disease, and it is a genetic skin condition that is characterized by freckling, redness, and blistering of the skin that is exposed to the sun. XP affects 1 in 1 million people in the US and Europe, but it is far more common in places like North Africa, Asia, and in the Middle East. This condition is inherited in an autosomal recessive pattern, meaning that both copies of the gene in each cell contain mutations. The parents of an individual with an autosomal recessive condition each carry one copy of the mutated gene, but they typically do not show signs and symptoms of this condition. So before going over the genetic abnormalities and causes of XP, I would like to quickly go over the effects that it has. So the effects are fourfold. They include cutaneous, ocular, neurological, and neoplasia. So the first is cutaneous. Given the hypersensitivity of XP patients to UV light as well as radiation and other toxic chemicals, this disorder is characterized by its cutaneous effects, in particular the dryness of skin. In terms of ocular effects, photophobia is noticed in XP within their first decade of life. And the damage to the eye can include keratitis, corneal opacification, as well as vascularization. Repeated sun exposure can potentially cause atrophy in the eyelids and can also contribute to vision loss. For neurologic effects, 25% of XP patients are known to develop a progressive neurodegeneration. Such conditions include acquired microcephaly, diminishing deep tendon reflexes, regressive high-frequency sensorineural hearing loss, progressive cognitive impairment, spasticity, ataxia, as well as seizures. The last category of effects is neoplasias. And again, like it was mentioned before, it is a very cancer-prone disease. The risk of non-melanoma cancers are 10,000 times greater, and squamous cell carcinomas potentially develop within the ocular areas, as well as the oral cavity and tip of the tongue. The average XP patient is diagnosed with their first non-melanoma cancer by the age of 9, and their first malignant melanoma by the age of 22 years of age. Okay. So the next category is genetic abnormalities within XP. So the genes that are involved in XP are part of a process, a pathway known as nucleotide excision repair. The proteins that are produced by these genes function to recognize DNA damage, unwind regions of DNA where damage has occurred, excising the abnormal sections, and then replacing the damaged DNA with corrected segments, as seen in the schematic. Since DNA can be damaged by UV rays from the sun and by such toxic chemicals, radiation, as well as free radicals, the job of a gene network like this one, NER, is incredibly important. Seven different genes named from XPA to XPG are also involved in this step process. The NER complex process is aimed at correcting the DNA that is damaged by the UV rays, specifically cyclobutane pyrimidine dimers and pyrimidine 6,4 pyrimidone photoproducts. Okay, this slightly more sizable schematic shows the same sort of process. So the first step is lesion recognition. Then we have unwinding of the DNA duplex by a respective helicase enzyme, the excision, which involves the three genes XPA, XPF, and XPG. We also have the DNA repair synthesis, and then the ligase that rejoins the DNA strands. Okay, so as far as the genes that are involved in XP, we have a mutation in the gene XPC, which is the first one seen in this table. It is the most common subtype within the United States, and it is located on the short arm of chromosome number 3. The proteins which result from these genes in normal expression are part of the NER pathway and function to recognize damaged DNA segments, excise them, and fill the gaps with corrected DNA segments. The mutated endonuclease is unable to recognize damaged DNA, and this results in severe sun sensitivity, as well as malignant tumor formation of the skin and mucous membranes. The second gene that we see here is XPA, located on the long arm of chromosome 9, and this gene codes for DNA damage binding protein 1, also known as DDB1, and it is on chromosome 9. DDB1 normally recognizes damage in DNA and assists in the unwinding of DNA, and patients with this subtype present with both skin as well as neurologic manifestations of the disease. The other following genes, from XPE to XPB, are rare subtypes, but they still play a role in the pathway. XPV is the variant, also known as POLH, and it is located on the short arm of chromosome 6. Okay, so as far as diagnosis and prognosis of XP, XP is initially diagnosed based on signs and symptoms, primarily considering that most patients do not have a family history of this condition. Molecular genetic testing can be done on the XP gene specifically in order to detect the condition earlier on. Given the early age at which patients are diagnosed with malignant melanomas, their life expectancies are unfortunately low. Life expectancies also vary based on presence of neurodegenerative effects as well as subtype. That being said, without neurodegeneration, the life expectancy is on average 37 years of age, and with neurodegeneration, it is significantly lower at 29 years of age. So for the last category on XP for today, we have that the pharmacological treatments that are available to XP patients all target the dermal manifestations of this disease. And that being said, there is a lack of effective treatment for the neurodegenerative effects. But a 2019 study done by Betty et al. Outlines potential treatments like antioxidant therapy with coenzyme Q10, as well as neurite development therapy with amitriptyline, which targets more than just the symptoms associated. These are the resources used for my presentation, and that concludes the presentation for today. Thank you. And if you have any questions, I'd be more than happy to answer them in the chat.
Great job.
Okay, if there are no questions, then we can move on. So the next presenters are Yuri Lin and Arlif Lozada, and they're students of the University of California, Los Angeles, and members of the International Circle of Genetics Studies USA chapter. They will be presenting on sickle cell disease, and if there are any questions for the presenters, please write them in the platform chat at the end of the presentation.
Okay, today Arlith and I will be presenting about sickle cell disease, and we'll be focusing on emerging drugs and therapies within the last few years. So Arlith will start us off with some background.
Hello, everyone. First up, with a little bit of background, sickle cell disease refers to a collection of inherited blood disorders that feature the ability for erythrocytes to change into a crescent or sickle shape. It is caused by the inheritance of abnormal beta globin alleles carrying the sickle mutation on the HBB gene. The most common form is homozygous HBSS, which is sickle cell anemia, with inheritance of beta S from both parents. There are also other forms of the disease, such as hemoglobin C and HbS, all of which express sufficient HbS to cause sickling of the cell. Diagnosis of the disease can occur through techniques such as electrophoresis by looking at hemoglobin and analyzing the presence of HbS and variant hemoglobins. As we mentioned here, as we can see, this is a visual representation of what I mentioned earlier about sickle cell disease being a variety of blood disorders. On the left, we have not having sickle cell disease, which shows us homozygous HbA, which is normal. Then you might have the sickle cell trait, but you don't have necessarily the disease. Then on the right, we have HBSS, which is sickle cell anemia, and that is the most damaging. And then we have the other forms, which also have sickle cell disease, but it's not as extreme.
All right, I'll briefly go over the clinical presentation. So the main clinical features of sickle cell disease are pain crises, which is a hallmark feature and essentially just means intense episodes of pain. There's also anemia, infection, and acute chest syndrome, which includes chest pain, coughing, and difficulty breathing, and also splenic sequestration, which means rapid enlargement of the spleen. And then other clinical features include swelling of the hands and feet, stroke, and leg ulcers. There are a lot more, but these are the most common ones. All right, we're also going to go over pathophysiology, which is going to be very important when we talk about the drug and gene therapies, as those target specific mechanisms that we'll cover today. So there are 4 key components, and the first is probably the most important one. hemoglobin S polymerization happens in low oxygen environments, and this is the root cause of sickling. And sickled red blood cells are rigid and liable to bursting open, which causes not only anemia, but also a whole host of other problems. And so to go over how this happens, we'll take a look at the genetic mutations. So normally, hemoglobin should have glutamic acid as the sixth amino acid, but there's a single nucleotide substitution that turns GAG to GTG, and that substitutes glutamic acid with valine. In this case, we then have HBS. And then we can see here that HBS actually behaves normally in an oxygenated environment, but as soon as it enters a deoxygenated environment, it turns into a fiber, and that is the polymerized bundle form, which causes the characteristic sickling shape. And if you can see from the diagram, the sickling shape is very irregular, and it causes blockages in the blood vessels, and that is called vaso-occlusion, which is a second key component. So vaso-occlusion results in ischemia, which is tissue damage from hypoxia, and it's actually the key cause of acute pain crises in most patients, and it's also responsible for most of the hospitalizations in people who have this disease. All right, so the third key component is endothelial dysfunction, and this essentially just means that there's impaired function in the lining of the blood vessels. If you take a look at this diagram over here, the endothelial lining is the innermost white portion. So what happens is sickled red blood cells are very likely to burst open, hemolysis, and that releases hemoglobin bundles into the blood. Cell-free hemoglobin is very reactive. So if you can see in this diagram down here, it undergoes oxidative redox reactions and forms things like free radicals and reactive oxidative species. And also you can see over here, NO turns into NO3 minus. So the depletion of nitric oxide is very damaging, since nitric oxide is an important gas that regulates vasodilation. And all of this comes together to cause oxidative stress and damage to blood vessels and organs. Finally, we have sterile inflammation, and this just means inflammation in the absence of microorganisms. And this is actually a result of the previous two components. So both endothelial dysfunction and vaso-occlusion come together to release inflammatory signals, which causes inflammation. And inflammation goes on to promote adhesiveness of neutrophils, platelets, and other cells. And what happens is there's a positive feedback loop with vaso-occlusion. And to put the four of these into perspective with each other, we'll just take a look at the arrows of this diagram. So HBS polymerization is the key cause of everything else in the diagram. And these two arrows go down and cause endothelial dysfunction and vaso-occlusion. Then if we see over here, vaso-occlusion and endothelial dysfunction both contribute to sterile inflammation. And the purple arrow just shows that there's a positive feedback loop between these two. Okay, now I'll talk about the drug therapies. So sickle cell disease drug therapies fall broadly into two categories. First are drugs that inhibit hemoglobin S polymerization. This essentially prevents or decreases the sickling of red blood cells. And the second category of drugs target symptoms or after effects of sickle cell disease. So you can think of one as a more upstream approach and one as a more downstream approach. So under the first category, it can be further subdivided into how the drugs mechanistically work. So the first are drugs that induce fetal hemoglobin. And one example of this is hydroxyurea, which was approved in the FDA by the FDA in 1998. And while this isn't a newer drug, it's incredibly important because it's the most commonly used drug to treat sickle cell disease. So to take a brief look at how this works, we actually don't know the mechanism of how it induces fetal hemoglobin, but we do know that it does. And if you can see from this partial pressure diagram over here, fetal hemoglobin is able to bind oxygen at a lower partial pressure than adult hemoglobin. That essentially just means that it's better at binding oxygen. And this results in a higher oxygen environment in a red blood cell and just prevents the sickling event from occurring. Second class of drugs under these are drugs that allosterically modify HBS and turn it into its high oxygen affinity R state. One example of this is voxelotor, which was recently approved by the FDA in 2019. So to go over how this works, we can see that HBS actually exists in two conformational forms. There's the T form on the left and the R form on the right. And if you look at this diagram over here, HBS behaves normally when there's a mix of both conformations or when there's R. However, if there's only the T conformation, it forms the polymers, which are the cause of sickling. So what voxelotor aims to do is to bind to HBS and induce the R state, which can essentially prevent the sickling. And if you can see, this diagram over here is very similar to this diagram, since the drug binding causes HBS to have a higher binding affinity to oxygen. Okay, next we'll talk about drugs that target the after effects or the symptoms of fetal hemoglobin. So the first is antioxidant therapies. One example of this is L-glutamine, which was approved by the FDA in 2017. So L-glutamine is a precursor molecule to NAD, which you'll be familiar with in your bio classes as an electron carrier. So over here in this diagram, you can see that NAD exists in two forms, NAD plus and NADH, and they undergo redox reactions to switch between the two. And so what NAD does is it scavenges free radicals and reactive oxygen species from the blood and from the body, and this essentially reduces endothelial dysfunction by reducing oxidative stress. And then next we have anti-adhesive therapies. One of these drugs is crizanlizumab, which was recently approved by the FDA in 2019. And how this drug works is it binds competitively to P-selectin. So P-selectin is a cell adhesion molecule. You can see in this diagram over here, it facilitates the binding of leukocytes, of abnormal red blood cells, platelets, and a bunch of other different cells. And when crizanlizumab competitively binds to P-selectin, it reduces this adhesiveness. And that also reduces vaso-occlusion, which, if you remember from before, just means blockage of the blood vessels.
Awesome. So now we'll be talking a little bit about genetic therapies in sickle cell disease. So these genetic therapies tend to follow a model of one, taking hematopoietic stem cells, which are stem cells that produce other blood cells from the patient. Then they're manipulating them genetically ex vivo, and then they're returning them to the patient. The first one will be globin gene addition, which is the transfer of an additional globin gene in HSC. As of now, this is the only available curative option for patients with severe disease. Still, there are many risks that come with the procedure, such as graft rejection, which occurs when the recipient's immune system attacks the donated tissue and begins destroying it. Next up, we have globin gene correction, which is the most straightforward approach of correcting the SED mutation. As we mentioned earlier, A becomes T, resulting in GLU to VAL by HDR. This correction of the beta S mutation has been achieved with various nucleases, including zinc finger nucleases, which are artificial restriction enzymes generated by fusing a zinc finger DNA binding domain to target specific desired DNA sequences, and DNA donor templates. Lastly, we have HbF induction, which relies on the potent anti-sickling effects of fetal hemoglobin, HbF, and seeks to enhance endogenous gamma globin gene expression through manipulation of normal factors and pathways responsible for HbF silencing in an adult. And as we can see here, this is more of a visual representation, because we really like visuals, of gene addition, gene correction, and HbF induction. As one little thing about globin gene correction is that a homozygous cell correction of just one of the two alleles will lead the patient to only have the sickle trait instead of SED. And as I mentioned earlier, having the trait would be better than having SED, because then you're not going to show the characteristics. Additionally, here we can also see that there are limitations. There's low transduction efficiency, there's low correction efficiency, and there's toxicity or variable outcomes for chemical inducers. So it is important to not only see that although there have been many successes with genetic therapies, there's also limitations. Now, we're going to finish up with the impact and the global significance of this disease. Although there have been many positive impacts on clinical care and research of sickle cell disease, most of these observations have occurred in first world countries. Places like sub-Saharan Africa, India, the Congo, and Nigeria observe over half of the cases of newborns being born with HBSS, with a total of 312,000 in 2010. So we can see there are a lot of newborns with this disease in these places, but yet not much research is being done in these countries. If these babies don't receive proper newborn screening and treatments like penicillin prophylaxis and pneumococcal immunizations, they're more likely to die from SCD before they turn 5. Fortunately, there are environmental and genetic factors that cause phenotypic expressions of the disease that can allow for diagnosis if there aren't enough resources to look further. Therefore, still, it is critical that we strategize how to stop the continuous mortality rates by analyzing where SCD is more prevalent. And I attached on the right an image of what we can do to help get more research going in these third world countries.
All right, we're going to wrap up our presentation by recapping all the key points that we covered today. So, sickle cell disease is a collection of diseases that's defined by the sickling of red blood cells. And the hallmark feature of sickle cell disease is unfortunately pain crises. A lot of other common symptoms include anemia, infection, and a lot of other complications. And recent advances have been made in sickle cell disease drug and genetic therapies. Some of the notable emerging drugs that I talked about today include L-glutamine, crizanlizumab, and voxelotor, all of which came out within the last 6 to 7 years. And then gene therapies include gene addition, gene correction, and HbF induction, as Arlith covered earlier today. And to put it all in perspective, so as most of us here are either future physicians or physicians already, we have to be cognizant of the fact that research and all these therapies don't happen in a vacuum. We have to take into account the global impact. So as Arlith mentioned earlier, most of the advances have occurred in first world countries, but sickle cell disease is actually the most prevalent in Africa, the Middle East, and India. There's limited research and access to these new therapies, which are often very expensive in the places that actually need it the most. And so to just end everything, we want to emphasize that research and access to treatments must be increased in the parts of the world that need them the most. So thank you so much for listening today, and that is our presentation. If you have any questions, please leave them in the chat box.
Muchas gracias a las ponentes por su excelente presentación. Bien, ahora los siguientes expositores son los estudiantes Wilson Ye y Wendy Su, ambos estudiantes de la Universidad de California, UCLA, y miembros también del Círculo Internacional de Estudios de Genética, capítulo de Estados Unidos. Vamos a presentar el tema denominado Long QT Syndrome. Y repetimos que si existe alguna pregunta, por favor, para los expositores, escribirla al final de la presentación y se pueda dar respuesta. Muchas gracias. Adelante.
All right, hello everyone. So today we're going to be presenting on long QT syndrome. And so Wendy will take us off with the introduction.
All right, hello everyone. My name is Wendy, and today we'll be learning a little bit more about long QT syndrome. Long QT syndrome is a cardiac disorder that affects the QT interval on an ECG. So the Q wave of the ECG represents ventricular depolarization, which initiates the ventricle to contract and empty. The T wave then represents the repolarization of that ventricle, which then initiates another cardiac cycle. LQTS often shows itself with a longer interval from the start of the Q wave to the end of the T wave when seen on an ECG. Or it can also present as an abnormal T wave. So long QT syndrome is estimated to be seen in about 1 in every 2,000 births, according to a 2009 study. However, there are some limitations to this study because it was done on all white infants, and there is little understanding of how this statistic extends to other races as well. In long QT syndrome, it has an average age of presenting itself at about 14 years of age, and this cardiac disorder it also presents the risk of sudden death if a cardiac event is experienced. And the probability of this event occurring, as well as its fatality, its deadliness, varies based off of a person's genetic mutations, the patient's sex, and the patient's QT interval length. There are some various forms of treatment for LQTS, but different forms of LQTS have varying responses to those treatments, and certain forms respond better to certain treatments.
And to finish off, one of the complications often faced by LQTS patients is torsade de pointe. So that's French for twisting of the points. And this is when you have the ventricles beating fast and out of rhythm, resulting in a twisted ECG reading that can result in sudden death. So to kind of better visualize what Wendy was talking about earlier, we here have an electrocardiogram, ECG for long QT syndrome. And as you can see here, the QT interval is denoted by the range from Q to T. And so the QRS complex is where ventricular depolarization takes place. And this is when there's an influx of sodium ions through voltage-gated channels, which allows for the depolarization of the ventricles. So you have sodium going in, depolarization, and then followed by ventricular repolarization at the T wave. And so this goes through the outflow of potassium ions through voltage-gated channels. And we'll be exploring this a bit more in depth later on in this presentation.
So as seen on the previous slide, one of the distinguishing clinical features of LQTS is the prolongation of that QT interval, as well as any T-wave abnormalities. Some other features of it as well are syncope, dizziness, palpitations, and seizures. There is an age-related increase in the risk of LQTS because the QTC interval generally increases with age. Other factors that also influence LQTS are abnormalities in extracellular electrolyte levels, such as potassium, calcium, and magnesium. And when there are changes to any of these potential gradients, that can alter the cardiac action potential, impacting the onset of LQTS. Quinidine therapy is one of the most common cases of acquired LQTS because it can alter extracellular levels of potassium, and then that can affect the QTC interval. Another factor that influences LQTS as well is certain medical conditions such as diabetes or epilepsy. For most patients of LQTS, they tend to be asymptomatic for the disease, and it remains unknown that their QTC interval is longer than the standard QTC interval unless an ECG is done on them. And the majority of one of the most common types of this syndrome do not have cardiac events, and almost half are asymptomatic as well.
So diagnosis is usually made through clinical and familial, so family history. So if you have a pattern of relatives or related ancestors, individuals in your family that have LQTS, then you would be at risk. And ECG is usually done to using this corrected QT interval, QTC, to account for the heart rate, and it's calculated according to Bazet's formula in order to account for the heart rate. And so prolonged QTC intervals is generally around 0.47 seconds in women and 0.45 seconds in men.
Because LQTS has to do with heart, primarily with that QT interval, we'll be looking at the key currents and corresponding ion currents associated with ventricular depolarization and repolarization. And mutations that occur that are common for LQTS are often increasing or decreasing ion channel function, affecting the current and resulting in the onset of LQTS. So the first one we'll be talking about today is this slow delayed rectifier current, and it is a potassium current which counteracts the influx of calcium ions. And the importance of the calcium ions is to aid in those depolarization efforts. This potassium current is a slowly acting outward potassium current that plays a key role in repolarizing the cardiac action potential. In some cases of insufficient activation of this ion current, typical in some forms of LQT, it's unable to counterbalance the calcium, prolonging the action potential because it's unable to repolarize. The second one is a rapid delayed rectifier current, and it has an outward potassium current that aids in repolarization as well. And it occurs at the end of the plateau and is responsible for sort of that sharp repolarization on sort of that middle image. And here, the HERG protein is one of the main players here. And errors and imbalances in this current also impact the QT interval, and causing mutations will extend that QTC interval. And then the last one for today is the depolarization current. This current is associated with sodium levels and electrical impulses in the heart. In certain forms of LQT, we see we see that the QT interval is extended. And it extends the ventricular action potential because the sodium current is unable to inactivate or remain inactivated during the repolarization process.
So there are several forms of LQTS, and so we're going to go over a couple of them. So the main form is the autosomal dominant form, and that is Romano-Ward syndrome. And Romano-Ward syndrome, LQT 1 through 3 make up 90% of genotype positive cases. So just those three forms, LQT1, LQT2, and LQT3 make up 90% of genotype positive cases. And then LQT 4 through 16, excluding 7 and 8, have a frequency of less than 1% for each type. And so you might be wondering, 7 and 8, then what is that? So LQT7 is actually associated with, it's Anderson-Tawil syndrome, which is associated with muscular weakness, developmental disabilities, and LQT8 is Timothy syndrome, associated with syndactyly, which is a webbing of the hands and fingers, and you also have an association with autism. And so you also have Jervell and Lange-Nielsen syndrome, which is JLNS, and this is also not very common, less than 1%, and this is the autosomal recessive form of LQTS. So today we're going to be mainly covering the Romano-Ward syndrome, since it is the majority of patients that have it. So LQT1, LQT2, and LQT3 make up approximately 90% of genotype positive LQTS cases. And how are they subdivided? LQT1 is the most common, 30 to 35%. LQT2, 25 to 30%, and LQT3, 5 to 10%. So pretty easy to remember. It goes in order of the way the numbers are labeled for the specific types. So LQT1. What gene is it? So it is, the responsible gene is KCNQ1, located on the locus is 11p15.5. Codes for the protein, the voltage-gated potassium channel 7.1. So here the K stands for the potassium, and then the effect is a loss of function. And so the aberration is a loss of function mutation of the KCNQ1 gene, which codes for the potassium channel 7.1. And potassium channel 7.1 is a voltage-gated potassium channel alpha subunit found within the cardiomyocyte cell membranes, which mediates the rectifier current, IKS, as Wendy touched on earlier. So just a reminder, so this I is the current, and K is the potassium, and the S is the slow rectifier current. So in this case, we're talking about the slow rectifier current. And this is the most common, present in 30 to 35% of LQTS cases. So understand words might be a bit hard and confusing to understand. So here we have this diagram, which I labeled the specific ions and what happens at each stage of an ECG. So this is normal so far. We're looking at a normal depolarization and repolarization process. You have an influx of sodium and calcium ions, which allows you to depolarize, followed by fast inactivation at the top. So at the very tip, you have inactivation of your voltage-gated sodium channels that keeps it from depolarizing any further. Then you have opening of these calcium and potassium voltage-gated ion channels, and the potassium ions kind of overpower the calcium one. So it leads to this steady decline in the plateau, followed by a closing of the voltage-gated calcium channels, which leads to this rapid repolarization process. So what happens in LQT1? So in LQT1, as we touched upon earlier, right, we have a loss of function mutation of the KCNQ1 gene coding for a potassium channel. So we're be losing a potassium channel function right here. And so what happens is you have a disruption of the repolarization process, leading to prolonged QT interval because it can't repolarize as quickly. And so as you can see here, I didn't, there's still the potassium channel open at the far right repolarization process because that's what is the known as the rapid rectifier currents, which we'll talk about later. But for now, you can see that there is this disruption of the slow rectifier current, which causes a longer repolarization process. So LQT1 individuals with QTC, so corrected QT interval of 500 milliseconds or greater, are high risk, while all others are low risk. So generally, this is not specific to LQT1, but generally, the longer your interval is, the higher the risk. And so use of beta blockers, fortunately, are pretty effective for LQT1 patients, and it's more effective than in LQT2 and LQT3 cases. It's the most effective one. And nadolol is the most effective beta blocker treatment, followed by, in this order, atenolol, propranolol, and metoprolol. And they all also exhibit protective influence from cardiac events, but nadolol is the most effective one. So what do we have next? We have LQT2, and LQT2 is encoded by the gene KCNH2, located on 7q3536, codes for the protein, the potassium voltage-gated potassium channel 11.1, and it's also a loss of function. So a loss of function mutation of the KCNH2 gene, also known as the HERG gene, which codes for potassium voltage-gated potassium channel 11.1, leads to a reduction of the rapid current amplitudes and prolonging of cardiac repolarization. The second most common, present in 25-30% of cases. And so as you can see here, you have only the rapid part, the rapid repolarization process is affected. So this disruption of repolarization leading to prolonged QT interval as a result of the rapid repolarization process being affected in this situation. And so now we have risk factors and treatments. So like I mentioned earlier, longer the interval, the worse, the higher the risk. And so for the difference between LQT2 and LQT1 though is for individuals under 500 milliseconds, females are generally intermediate risk while males are low risk. There's also a reduced efficacy of beta blockers in comparison to LQT1, though nadolol remains the most effective beta blocker treatment, but there's an absence of cardiac event reduction from the other beta blockers that worked in LQT1. And so now we have LQT3. And so this is the SCN5A gene on the 3p21-24 locus, and codes to the protein sodium, the voltage-gated sodium channel 1.5, and it's a gain of function mutation. So as you can notice, there's a difference here, right? We have a gain of function mutation this time rather than a loss of function. And this is due to the fact that we're focusing on the sodium channel now rather than the potassium channel. And they counteract each other in this depolarization, repolarization process. So there's a gain of function mutation, and that leads to a disruption of the cardiac sodium channel fast inactivation process. So it's easier to visualize it. So here, as you can see, what it does is you have an inactivation of sodium channels at the very tip, which allows you to repolarize. But when you have this gain of function, it doesn't, you slow and you decrease the inactivation process, which prolongs your repolarization process. So you can just imagine at the tip, it takes longer for the sodium channels to shut down. So you have more sodium channels coming in and keeps it depolarized and takes much, much longer to repolarize. And so risk factors, male sex with QTC greater than 500 milliseconds indicates high risk, while all other groups are intermediate risk. So for this one, you get mixyla 2018, a voltage-gated sodium channel blocker has been shown to decrease QTC length and reduce cardiac events. And so that is not a beta blocker. And you have reduced efficacy of beta blockers in comparison to LQT1 and LQT2. So the ones we listed earlier, which is not mexilatine, mexilatine is a voltage-gated sodium channel blocker. And there hasn't been beta blocker efficacy shown until 2018 due to just the rarity of LQT3. But it was demonstrated in 2018 that there is a slight efficacy of nadolol as well.
So with LQTS, there are two types. There are acquired and congenital LQTS. So congenital LQTS means that the disease is present from birth in the individual. Acquired LQTS is the more common form, and it occurs from triggering factors such as electrolyte abnormalities, drug use, and certain medical conditions. Acquired LQTS also has a significantly shorter QTC interval of an average of 453 milliseconds, whereas the congenital form of LQTS has an average QTC interval of 487 milliseconds. And there's still a genetic basis to some acquired LQTS patients. And a study found that 28% of the acquired LQTS patients have a mutation on major congenital LQTS genes. So this diagram right here shows the distribution of genetic variations amongst acquired and congenital long QT patients. The key takeaway from this diagram is the finding that in LQTS, in the mutation on the gene KCNH2, which represents the long QT2 form of long QT syndrome, is more common than the LQT1 mutation on the gene KCNQ1. It is not known exactly why this occurs. Furthermore, there is a higher prevalence of KCNE1 and KCNE2 mutations in acquired long QT syndrome. This could be due to their roles as accessory proteins in cardiac potassium channels, so their dysfunctions aren't detrimental enough to cause congenital LQTS.
So moving on to therapy and management, how do we manage LQT cases? So you generally avoid triggers for cardiac events, and that could be physical exercise, startling events such as loud noises, alarms. So what they'll say is don't set alarms, and emotional stress. So it's actually specific to which type of LQT you have. So for example, if you have LQT1, physical exercise is the primary cause of cardiac events. So if you have LQT1, you really, really want to avoid physical exercise. But LQT2 and 3, it's not so much physical stress as it is emotional stress and startling events, whether that's from startling events from rest and sleep. Beta blockers is the primary treatment, first line of treatment for long QT syndrome, with nadolol being the most effective. Alternatively, you have implantable cardioverter defibrillator (ICD) implantation for high-risk patients, as well as left cardiac sympathetic denervation (LCSD) for ICD patients with arrhythmia storms. And it's basically when beta blockers don't really work, so you got to resort to this. And then finally, you have mexilatine for LQT3 patients, as mentioned earlier.
So in conclusion, LQTS is a cardiac disorder where we see a longer than normal interval between the beginning of the T wave that kicks off ventricle depolarization or contraction to the end of the T wave, which represents ventricular repolarization or relaxation. It can also present with T wave abnormalities. Oftentimes, it manifests from mutations that affect the potassium and sodium channels, which play a large role in the depolarization and repolarization process. There are three most common forms of LQTS: LQT1, LQT2, and LQT3. They are classified according to their mutations and effects on mechanisms resulting in a prolonged QT interval. The most common type is LQT1, characterized by a loss of function mutation of KCNQ1, which codes for the voltage-gated potassium channel 7.1. The second most common type of LQT syndrome is LQT2, also characterized by a loss of function mutation, but this time on KCNH2, which codes for the voltage-gated potassium channel 11.1. The third most common type is LQT3. Unlike the other two, it is characterized by the gain of function mutation on the SCN5A gene, which codes for the voltage-gated sodium channel 1.5. If these currents are not operating properly, there is a prolongation of the QTC interval, which is an indicator of LQTS. Proper treatment is extremely important with LQTS. Those who do not receive proper treatment have a high risk of dying within their first year of syncope. Those who do have an extremely low mortality rate within a 15-year period. The two most common forms of treatment are lifestyle changes and beta blockers. In certain cases, ICDs or LSCDs are also treatments prescribed to patients as well. Hopefully this presentation brought you all more insight on this disease, and thank you all for listening. If you have any questions, please drop them in the chat box below. And these are our references.
Bien, muchas gracias a Wilson y a Wendy. En esta oportunidad, para finalizar ya nuestro workshop, invitamos al último expositor, el estudiante John Mamani, quien es estudiante de la Universidad Nacional del Altiplano, y en esta oportunidad va a presentar su tema denominado fibrosis quística. Y reiteramos de igual manera que si existe alguna pregunta, al finalizar la presentación, la ponencia del presente expositor, la pueden hacer en el chat de la plataforma. Bien, adelante, John.
Ya. Ya, bueno. Muy buenas noches, soy yo, y en este caso voy a exponer fibrosis quística. Esta enfermedad de fibrosis quística es una enfermedad multisistémica que altera el flujo de iones y por lo tanto del agua. Bueno, altera el flujo de iones de la célula y por lo tanto del agua, lo cual produce secreciones densas y secas. Usualmente esta fibrosis quística se da en los pulmones. La obstrucción de las mucosas en las glándulas exocrinas es la principal causa de morbilidad y mortalidad en pacientes con fibrosis quística. Y existen 6 clases de fibrosis quística. Aquí se puede observar. Bueno, esta fibrosis quística tiene en particular, es debido a una mutación, bueno, la más común es la del gen CFTR, que es el regulador transmembranal de fibrosis quística. Este gen se encuentra en el cromosoma 7, en el brazo largo, en el lugar 31.3. Todos al poseer este gen, pues no padecemos de esta enfermedad. Sin embargo, las personas que tienen mutado este gen, como es en el caso de la fibrosis quística, pues presentan alteraciones, como las mencioné anteriormente. Bueno, la mutación más común es de la delta F508, que es la más común entre las fibrosis quísticas, ya que existen más de 2013 mutaciones enumeradas en la base de datos para mutaciones del Consorcio de Análisis Genético de Fibrosis Quística. Esta mutación de la delta F508 crea la falta de la fenilalanina, que cerciona cerciona o corta la porción de la citonina y la timina y timina para que no dé la fenilalanina y así está más común. Esta fibrosis quística, esta mutación, a ser una enfermedad autosómica recesiva, pues se va a, a heredar en los hijos. Como aquí podemos observar, aquí el padre es portador, pero sin embargo no presenta, ya que solamente la tiene en un brazo del cromosoma y aquí en la madre también la tiene en un brazo del cromosoma. Sin embargo, aquí vemos su hijo afectado al poseer ambos brazos de cromosoma afectados. Aquí el 50% de sus hijos pues puede o no poseer la enfermedad y el 25% pues puede o no poseer la enfermedad. Seguidamente aquí tenemos las mutaciones de fibrosis quísticas más comunes, la que es la G542X, que es la más frecuente en España, que pertenece a la clase 1, que se llama defecto de síntesis. Esta síntesis defectuosa de la proteína por contener señales de terminación prematura. En la clase 2 observamos el defecto de procesamiento y tráfico intracelular. Aquí hay otras mutaciones que es la N1303K, la 1507, y pues esta es la proteína del CFTR. No, esta proteína no puede plegarse correctamente y asumir su configuración terciaria apropiadamente. En la clase 3 tenemos el defecto de activación y regulación. En esta mutación es una mutación sin sentido, que es la mutación de G551D, y produce un defecto en la actividad reguladora de la proteína CFTR o canal de cloro que se encuentra en cada célula. En la clase 4 pues encontramos el defecto de conductancia, que aquí tenemos tres mutaciones, la R117H, la R347 y la R334W. Esta es la presencia de una cantidad normal de la proteína en la membrana apical, pero presenta una reducción en el transporte de cloro en este canal. Aquí en la clase 5 observamos la reducción de la cantidad. Pues esta presenta una reducida síntesis de la proteína y un tráfico, por lo tanto, un tráfico ineficaz. Aquí se encuentra con frecuencia relativa alta, como la mutación 2787 más 5G. Y en la clase 6, pues tenemos la renovación acelerada. Afectan las propiedades reguladoras de CFTR sobre otros canales de iones como la ENAC. Seguidamente, pues tenemos una tabla de mutaciones más frecuentes del gen CFTR y en poblaciones con alta prevalencia. Este estudio, pues, aquí nos hace una pequeña lista y aquí nos presentan, bueno, esta es la Delta F508, que es la más prevalente en el mundo con un 66%. Seguidamente tenemos la G542X, que poseen un 2,4% en España, y así sucesivamente todas las mutaciones alrededor del mundo. Esta enfermedad, al ser hereditaria, pues es una enfermedad progresiva, ya que no se va a presentar cuando sea bebé, cuando sea niño, o tal vez sí, pero al ser progresiva, pues esta enfermedad va a ir, va a ir afectando poco a poco los pulmones. Y a la edad de los 20 años, 25, a esa edad, pues el adulto ya se va a encontrar en unas condiciones muy malas para seguir con la vida. Y pues aquí es donde se ve más la consecuencia de esta enfermedad. Seguidamente, pues tenemos este defecto genético de la proteína CFTR, ya que es la falta del funcionamiento de la propia proteína por una de todas las mutaciones que se dividen en las diferentes clases. Después tenemos la pérdida de la secreción del cloro. Aquí podemos observar una célula normal y de buen estado. Y aquí al costado una célula con fibrosis quística. Aquí podemos observar un normal transporte del cloro y el sodio. Sin embargo, aquí pues ya no se observa que el cloro salga, sino que solamente el sodio entra y al cloro al no poder salir el cloro debido a esta enfermedad, pues lo que hace las mismas células permiten que el sodio ingrese en mayores cantidades hacia las células y así haciendo que la mucosidad de lo que se encuentra en un estado normal pues se reduzca y haciendo que las microvellosidades ya no puedan, ¿cómo se dice? Ya no puedan trabajar normalmente. Bueno, aquí lo que les dije de aquí, la absorción dominante del sodio, aquí observamos, luego la depresión en líquido en superficie de la vía aérea, después sigue la mucosa deshidratada y estática, aquí se puede observar, el deterioro en aclaramiento e inflamación e infección. Bueno, pues esto es un círculo vicioso, por así decirlo, ya que todas estas mutaciones, pues, nos van a dar una proteína CFTR anormal que no va a trabajar adecuadamente. Después vamos a tener el defecto del transporte iónico y va a hacer que el sodio entre en mayores cantidades. Seguidamente vamos a tener el aclaramiento mucociliar alterado. Después vamos a tener la obstrucción o tapones mucosos y así sucesivamente se va a crear una infección, después una inflamación y de nuevo la obstrucción o tapones mucosos y se va a hacer un círculo de daño crónico. Y finalmente va a ser la cicatrización y vamos a tener una enfermedad pulmonar terminal. Y bueno, eso sería todo por mi parte. Gracias por escucharme y gracias.
Bien, muchas gracias John y gracias a todos los presentes por permanecer en esta ponencia, por estar acompañándonos en este workshop. Y no sé si es que se puede apreciar en pantalla, pero igual queremos invitarlos a, nuevamente a este curso virtual de citogenética que se va a realizar del 26 al 30 de abril del presente año, de 2 a 7 de la noche, en este caso con un costo para estudiantes de 300 nuevos soles peruanos y para profesionales 500 soles. Y también les invitamos a nuestra jornada internacional de genética que se va a realizar el próximo año, es decir, en el 2022, del 10 al 12 de agosto de ese año mencionado. Para mayor información en ambas presentaciones, tanto para el curso y para la jornada, nos pueden encontrar en nuestra página de Facebook como Círculo Internacional de Estudios de Genética, o también nos puede escribir a nuestro correo electrónico, el cual está presente y escrito también en la página de Facebook. Ahora sí, damos por culminado nuestro evento y agradecer a todos ustedes por su participación. Muchas gracias.
Gracias, gracias.
Muchas gracias.
Hasta luego, gracias, encantado.
Hasta luego. Muchísimas gracias.
Gracias, hasta luego.
Que estén muy bien.
Usted también. Diana María Pedillo, ¿desde dónde es Diana María? ¿De dónde estás conectada, Diana María?
De Cali, Colombia.
Ah, Diana, ¿cómo estás? Encantado. Mucho
Mucho
gusto, ya nos
gusto.
estaremos viendo. Nos vemos en Bogotá o en Colombia. Tú nos invitas nomás,
Claro que sí.
pero nada de virtual. Ya generalmente estos congresos virtuales, es difícil,
Son
pero
complicados.
bueno. Gracias a todos, de verdad, gracias a todos por estar con nosotros. Como ven, este es un esfuerzo de los estudiantes son estudiantes de pregrado, ¿no? Que están en todo el mundo. Estamos en Buenos
Muchas
Aires, estamos en Perú, en España, en Estados Unidos y por supuesto ¿cómo se llama? En Perú, ¿no? Bueno, encantados. Si tuvieran alguna pregunta que quieran hacer, veo que está David Trejo. ¿Qué te pareció, David, el simposio? David. David, ¿estás ahí? Bueno, si quisieran presentarse, alguna persona que quiera presentarse, por favor, aprovechen porque en 5 minutos nos desconectamos. Ok. Rodrigo. Thank you, Catherine. You did an excellent job.
Thank you. It was really great to be here, and I really enjoyed listening to everyone else's presentation.
Yes, very good. They are undergrad kids, but they are really ahead of us. Wilson, thank you very much. You did an excellent job. Okay, good job to everybody. Okay, have a nice weekend, and thank you very much for being with us. Okay, thank you. María Cristina Tello López, ¿de dónde estás conectada? Se fue, no escucha. María Cristina, ¿dónde estás conectada? María Cristina. Bueno. ¿Qué dice Rodrigo? No te entendí.
Buenas noches, doctor. ¿Cómo está?
María Cristina Tello, ¿no? ¿Quién habla?
María de Guadalupe Enríquez, doctor, de la región de
¿Cómo estás? Hola, María de Guadalupe, ¿cómo estás? Resucitó.
realmente muy buenas ponencias. Al mismo tiempo, se estaba viendo que se pueden aplicar bastantes herramientas computacionales para poder hacer simulaciones y pues poder inhibir algunos factores que alteran el proceso genético y no tener mayor patologías en adelante.
Así es, así es. Está también con nosotros el doctor Jorge Manuel Vázquez Cerezo. Encantado de conocerte, Jorge. Nos vemos en México, ¿ok?
Vale, nos vemos en México.
El
Espero
10 de
poderlo
junio estoy
conocer.
llegando, Dios mediante, porque tú sabes que uno propone, pero Dios dispone. El 10 de junio
Exactamente.
estoy llegando, voy a estar en el hotel María Isabel Sheraton, ¿ok? Por si acaso. Si no me
Ok, por si
encuentras,
acaso.
ahí me llamas por teléfono al hotel, ¿ok? Cualquier cosa nos estamos
Bueno,
comunicando.
no
¿Ah?
tengo su número, pero sí, claro.
Pero agrégame, ¿tienes Facebook?
Claro.
Ya, agrégame en Facebook, Carlos Tirado. Estoy con mis dos sobrinos ahí al lado, ¿ya?
Ok,
Un abrazo.
Carlos.
Carlos ha tirado, ¿ok?
Ok, perfecto. Muchas gracias.
Encantado. Nos vemos en México, porque a mí
Encantado. Nos
me encanta
vemos.
México. Ok, adiós, adiós, bye bye.
Vale.
Oye, un abrazo a todos. David Trejo está sí, con nosotros, David. David, buenas noches. Creo que está en el autobús porque no nos contesta. Muchas gracias, Francis. Muy buen trabajo has hecho. Gracias. Andrea
Muchas
Chávez,
gracias a
¿desde
usted también,
dónde nos
doctor.
llama? Andrea Chávez, ¿desde dónde nos llama? ¿De dónde está conectada? Andrea. Andrea, buenas noches. No dice de dónde son. Bueno, está Fabián. Fabián, thank you very much. You did an excellent job, okay?
Thank you. Thank you,
Thank
Dr.
you.
Tirado.
Thank you very much. Everybody, all my students from America and all my students from Peru, great job, okay? Andrew Reyes, are you there? He's already gone, no? Okay. Is Andrew Reyes there? No, he's already gone. Okay, that's fine, no problem. Well, thank you very much to everybody, and I will see you, okay, next time. Thank you. Cerramos, Rodrigo, ¿verdad? Rodrigo.
Sí.
Ya, perfecto. O nos quedamos en FODA. 10 minutos,
Sí.
¿puede ser? 10 minutos, que está fresco todo.